because I have selected only one in this case, and DKK binding assay using the immunofluorescence, and also artificial analysis of the expression of the gene related to the Wnt signaling. So this is the MTT assay for the evaluation of cytotoxic of these drugs. It actually IC50 of 456 plus or minus 17.95 micrograms. And also, we have confirmed the LRP plasmid insertions by using these Western blotting things. And Western blotting results of LRP6 overexpression of HETK cells were demonstrated. And the MTT assay for the evaluation of cytotoxicities of rosidronate is actually performed in these overexpressed cell lines. And finally, the last part of my presentation is about the conclusion. So I have not included complete because of the lack of time. I have done only the in vitro and in silico part. So in silico part is the selection part, and second one is I have performed only a few tests with respect to this in vitro. So finally, to conclude this presentation, the exact pathophysiology involved in the Alzheimer's disease is still a topic of debate and challenge for the many researchers. The amyloid hypothesis, neuroinflammation, oxidative stress, insulin resistance, and mitochondrial abnormalities have also been associated with the Alzheimer's disease. Late diagnosis is, as well as lack of therapeutic treatment, add another hurdle for the patient suffering. Shown an increase in the Wnt antagonist DKK expression. So that was my hypothesis. So my target was you reduce or block this DKK activities and dysfunction of the Wnt co-receptor LRP and activation of GSK3 beta. And currently, by utilizing
Hello.
yeah,
Thank you, sir. Thank
thank
you,
you.
Dr. Pai. It's a nice presentation. And I have, if any queries from the audience, any questions? So, drug repurposing is very important
Yes.
because it's why it's important because if we use the one drug for two diseases, it's a good like aspirin. Where previously it was used for the antipyretic, nowadays for the antiplatelet. So, sir, use the recent techniques like in the old pharmacology, we use the testing of drugs directly on the animals. But nowadays, we first use the in silico, then in vitro. Nowadays, network pharmacology also added, then in vivo. So by using these techniques, we decrease the use of animals in the research. So thank you, sir.
Yes.
Thanks a lot.
But ultimately, we cannot replace the animal. Animal work will be there only because, and I want to make it one thing very clear. Please remember, in silico is not ultimate one. You will see that you may be having very good interaction, and in the actual part, you might not get that. So don't think that in silico is the ultimate one. You will get 100% result. Okay, so that what are the advantage of this particular in silico is you can reduce the time. See, 1 lakh compound, I can screen 1 lakh, 2 lakh, 3 lakh, one tenth of this many compounds. So within 2 3 days, I can sort down. Okay, that is one of the advantage of this particular in silico. Okay,
Yes, thank you, sir.
thank you, thank you.
So please wait. Thank you, Dr. Pai, for the wonderful session and the talk. We would request our chairs to kindly felicitate Dr. Pai.
Without wasting time, I'm calling next speaker, Dr. Ashwini S. Patil. He is an assistant professor at the DYP DPU School of Pharmacy and Research, Pune, and a dedicated pharmacology researcher with expertise in preclinical pharmacology, translational research, and medical scientific writing. She earned her PhD in pharmaceutical technology from the Institute of Chemical Technology, Mumbai, where she gained specialized research experience at premier institutions including ICT Mumbai. Her research focused on neurological disorder, cancer biology, and cardiovascular pharmacology, with technical expertise in stereotaxic brain surgery, carotid artery cannulation, and advanced behavioral and biochemical techniques. Dr. Patil has authored impactful research papers in reputed journals such as Biomedicine and Pharmacotherapy, Helion, Applied Biochemistry and Biotechnology, Inflam- Inflammopharmacology, and Molecular and Cellular Biochemistry, with a cumulative impact factor of 38 plus and increasing research citations. She received the prestigious DAE ICT Fellowship in 2019 to 2023 and has been recognized with first prize at the 7th International Conference on Emerging Trends in Drug Development and Delivery, along with presentations at major scientific platforms, including the Society for Neuroscience, San Francisco 2023, and Indo-Gulf WANPS 2024. Her notable research contribution includes a high impact publication impact factor that is 7.5, elucidating cannabinoid modulating phytomolecules for metabolic syndrome. Now I am inviting Dr. Patil for valuable.
Thank you so much, sir, for the kind introduction. First of all, very warm good morning to all of you. May I know the audience? All are UG, PG students? Yeah? Yes? UG students. Okay, fine. So my presentation is towards the what we can say higher education. What all are the opportunities you have in pharmacology? What career you can build to start with? The title of the presentation is Catalyzing Change, Young Minds Transforming Pharmacology for a Developed Nation. And I would like to thank the entire organization committee to invite me for this session. So, to start with, the definition of health, as we are a pharmacologist, we are dealing with the health of human, animals, right? So, the basic definition as per WHO is a state of complete physical, mental, and social well being, and not merely the absence of disease or infirmity. So, why it is very critical to know what is pharmacology? So, pharmacology is a branch of medicine, you can say, where we study the effect or the therapeutic potential of drug, maybe a new drug or repurposing of existing drug, right? So, to have a strong knowledge of the basic pharmacology is very, very essential. So, in this, I would like to highlight three major points. Those are healthcare foundation, treatment and cure, cross field innovation. So, in healthcare foundation, as I mentioned, pharmacology, the knowledge of pharmacology is very, very essential to know in depth what is the mechanism of a disease so as we can find the right drug for its cure. Next point, that is treatment and cure, right? So to find a right treatment for a disease, for that we need to understand by which mechanism that drug will act, right? So first of all, we need to understand the underlying mechanism of a disease, and then we can target a new drug for curing that disease. Third point, that is cross field innovation. So as pharmacy is, as we understand or we imagine that it is a paramedical field, right? But nowadays it is expanding with medical devices, isn't it? So we have to be in touch with other fields or other departments as well. For example, the biomedical departments. So that is the importance that we should do the research in collaboration. So it should be a multidisciplinary research that will lead to a cross-field innovation. Okay, I think it should be a two-way communication. Everyone should focus towards the presentation. Yeah. So to take some examples of recent game changers in pharmacology, right? So the first one, so as we are discussing about now repurposing of drugs, okay? So paricalcitol, which was previously very well known for curing renal diseases, now it is been found in researches that it is a vitamin D receptor activator, which is showing a promising results in cardiac remolding. Next, the drug repurposing. Then next is chronopharmacology. So as we all know, time is very, very important. We can't wait something will fall from heaven. We have to work on things. Okay. So as we all know, in the scenario when it was a COVID situation, right? So it was a urgency of the society, entire nation, entire world for the drug which will cure the situation. That was a COVID virus. It will kill the COVID virus, right? So time was very, very important at that time. So which was the company which discovered the vaccine? Anyone would like to answer? Yeah, it was India or abroad? Yeah, it was serum, isn't it? So India was the one to discover the vaccine, even though there were many players in the market who were trying to give the treatment for COVID, but Bharat Serum was the one who discovered the vaccine. Okay, so time is very, very important. We should utilize the time for the what we can say, the right drug for a best delivery. Then the protacs are now recently being widely used in the therapies of cancer. So what are protacs? Okay, so these are remolded proteins which are now being used for anti-cancer therapies. Then next is AI-driven drug discovery. As you all know, AI, that is artificial intelligence. To term about it is not artificial intelligence, it is aerial intelligence. Okay, so we can use many of the AI tools for detecting new diseases, new targets, and with the help of that, we can find a new drug which will act on that specific target. And accordingly, we can work on animal models or maybe cell line studies and further on clinical patients. Then the very, very recent, those are biosimilars, organs, organoids, and organ on a chip. Okay, so the entire organ. So previously, you must be knowing that we used to do cell culture studies. Okay, then after cell culture, whatever the results you get, that we further do into animals. Okay, now instead of using animals, even though in the last lecture, sir was mentioning that we cannot refuse the use of animal, we have to do animal studies. But then even there are now models on with the help of which you can do the research on the entire organ, which is based on chip. Okay, so that will reduce the amount of drug as well as the accuracy. It will increase the accuracy of your experiment, and you can do multiple experiments at single time. So again, reducing the time and energy. So it this slide also again is resembling the previous slide. Okay, so emerging frontiers in pharmacology, it includes AI driven drug discovery, forensic applications, precision medicine, microbiome pharmacology. Nowadays gut microbiota, it is playing a vital role in every disease pathology. So gut when related to brain, gut in related to cardiac disorders, it is playing a vital role in every diseases. So to find a drug which will act via gut, like which will improve your microbiome, will effectively cure the other targeted diseases. Then as I mentioned, drug repurposing, to give you example of three important drugs which are being repurposed. So that is a thalidomide, okay, repurposed for multiple myeloma and leprosy. Sildenafil, from antihypertensive drug to treatment for erectile dysfunction and pulmonary hypertension. Third one is metformin, that you all very well know. It's an anti-diabetic drug. Okay, nowadays it's a very beautiful molecule. It is having many potential, and nowadays it has been the scientists are exploring it for many other therapeutic efficacies. For example, cancer uh, and metabolic disorders. Then the last one is the nitric oxide. As you all know, nitric oxide is very, very toxic. But then it has been found that nitric oxide has multiple physiological roles, that is vasodilation, immunity, and neurotransmission. It has broad antimicrobial and antiviral properties. It is easily delivered via inhalation or topical formulation. So the intention of mentioning these points to you all is you all can think in that direction. What are the possibilities on which we can work in future? As you all are in UG right now, further if you are interested in pharmacology, MPharm in pharmacology, so during your academic projects, you can choose such projects for finding multiple roles of any XYZ drug for its different activities. So next thing is translational breakthroughs. So as you all know, rare diseases are very rarely seen in India, okay, because of the strong immunity we can say we have. Okay, in abroad, these diseases are commonly seen. Okay, so to find a cure for rare diseases is very, very important nowadays. So as I'm mentioning again and again, to find a cure, that is meaning to start with a research from in vitro, then preclinical studies. Okay, so to find the right model for a study and to start study the molecule for a specific disease. Then pharmacology also deals with biomarker validation. Okay, so nowadays you must have heard about early detection kits. Okay, so these kits are nothing but these are based on the biomarkers. For example, the metabolites after once the disease is induced in a patient or in animals. So ultimately something is getting converted to something. So that way the metabolite can be identified and how it can be detected, that process can be identified and that it can patent and can be converted into a detectable device. Okay, then next thing is to take a molecule from preclinical to clinical phase is also very, very important, because many a times research are done and they are kept limited to the lab itself. Okay, they are not taken to the clinical level. Okay, so it's very, very important whatever we are doing, it should not be a simple process just for the sake of doing something. It should come with some uh, output. Okay, it should come with some product. Okay, so that is the entire benefit of a research. Otherwise, just for the sake of thesis or publishing a research paper, it's not enough. Okay, it should be finally with something that can be used clinically. Okay, so why the change is so important? As I was mentioning, uh, the title was catalyzing change. So why there is a need of change? As now you all know, there are so many medicines. If you talk about antimicrobial resistance, okay, it is increasing widely. Okay, so there is a need of new antibiotics, isn't it? So we need to work on new areas, we need to work on new drugs. Okay, so to work on a molecule which is totally you don't have any idea of that molecule, that's very blank. Okay, instead of that you can start working on a molecule which is already is there in the market, that we called it as a repurposing. Okay, so that is you can say comparatively is more easier, because 50% data you will get for that molecule in the website, right? And even the molecule, if it is already there in the market, then its safety profiling, ADR reporting, everything is there already. So you don't have to go back and then again research on it. You can work further for its new efficacies, right? So that is a very important area you can work on.
Sorry for interruption, ma'am. A little faster, please.
Yeah, sorry. You have 2 3 slides only. Okay. Yeah. So these three slides I have, last three slides, those are very, very important for all of you. So yeah. So I think you all must be having the Institutional Innovation Council at our institute, right? So so giving the facilities to the students is very, very important. So you all must be having all the facilities at your end. Okay, to utilize all these facilities is your part. Okay, so while choosing your projects at your UG level, choose in such a way that it will lead to some product, as I was mentioning. Next thing, whatever you are working on, you should be able to present that work at some international or national conferences. That will give you a different confidence that you are working something and you are able to present that work in front of people. Okay. Next thing is, if you think of, I have many ideas in my mind, but I don't know how to execute it, right? There is a part of funding as well. Okay, so I've mentioned a few things. I'll share this PPT if you need. Okay, so there are Atal Innovation Mission, Startup India Mission, Nidhi Prayas. These all are government agencies which do fund the startups for giving them from seed funding, so that is a pre-incubation phase, and then incubating that product to a next level. And those fundings are not refundable, or they do not charge for those funding. Okay, you can explore these as I'm mentioning. Yukti, IIC, that is Institutional Innovation Council. Then next is, you all must think, as you all are young blood, okay, you should always think for higher education. Nowadays, it's very, very important. Don't just stop after your graduation or PG, okay? It's very, very important to go for higher education, that too from a very good institution where all the research facilities you will have. And I would suggest that while doing your research, as I mentioned already, that the research should be something which will get converted into a usable product. Okay. So I have mentioned in the last point that for higher education as PhD, so don't think that PhD is a full-time study. Okay, you can think it as a full-time study with a salary. Okay, so government is paying you a very good amount of salary. Now it is been increased. Okay, so you get as a junior research fellow after completing your masters. For the initial 2 years, you get 45,000 in hand, and after that you get a good amount of money. Okay, so go for higher education, that all we need for our developed nation. So what should be the visions of our future? So scientific discovery, youth led innovation, so all you have to work together and give innovation that will develop our nation, self sufficiency and global contribution. Thank you all, as we are running short of time, so I had to wind up it early. Thank you so much.
Very nice presentation by Dr. Patil. Any question from the audience? You may ask.
So I have my contact details as well. If you wish, you can contact me later on as well. That's not an issue.
Dr. Patil explains very well critical role of pharmacology, recent game changer of pharmacology, importance of AI, emerging frontiers like AI-driven discover, forensic application, precision medicine, and translational breakthrough. Any questions? Thank you, Dr.
Thank you so much.
Thank you, Dr. Patil, for the insightful talk. We would like our chairs to kindly honor Dr. Patil. Request Dr. Pai to please come on the stage. So can you please? We request Dr. Pai and Dr. Patil to kindly felicitate our chairs. Dr. Ved Prakash. Ma'am, please. Okay. तो दूसरा भी लाएंगे नीचे डॉक्टर थैंक यू वहाँ पे डॉक्टर पुनीत बंसल We thank worthy chairs for gracing this occasion. We will rejoin at 10 a.m. हाँ हाँ sure sure I will just ask the student to बेटा इनके pen drive में कर Request all the students to be back in hall 4 at exactly at 10. You have a talk? Yeah. What?
Here, this. CP This light is not occurred.
सर वो भी फ्रीज कर रखा है ना
हम्म?
फ्रीज कर रखा है लाइव में चल रहा था ना उसके लिए फ्रीज किया आपको दिखा
पेंट
देता
का जो प्रेजेंटेशन है ना
हाँ जी
वो थोड़ा क्लियर नहीं आता देख रहा था
क्लियर नहीं आ रहा तो ये इनके प्रोजेक्टर में है सर ये ऑडी
वही मैं बोल रहा प्रोजेक्टर का प्रॉब्लम है
प्रोजेक्टर का ही
लाइट का पावर कम है
ये
एक बार दिखा लीजिए पीपीटी
आपको भी सुबह मैंने देखा तो मैंने लगा मैं डॉक्टर जय के साथ कल खेलूंगा
सेकंड टॉपिक फर्स्ट है टोटल लोकेशन उसके बाद तो यही रहेगा ना
हाँ जो अगर सर हर आप ही आ रहे
हाँ अगर वो नहीं आएगा तो वो नहीं आया क्या
ये आई नहीं
किसी ने ना कॉपी किया था बट hopefully पता नहीं क्लास का कहाँ था क्या नहीं था ये कुछ किया because the number was not there in the search person list भी है पर actually आपको कुछ के बाद आप देखिए
ठीक, नहीं लग रही, नहीं लग रही काउंटिंग।
सेकंड चेयरपर्सन कब तक आ रहे हैं? I don't know. Understood. नहीं इसका मैं कर तो नहीं है अगर पता क्या अगर आ रहे हैं 5 मिनट वेट कर सकते हैं हाँ हाँ सर तो उसके कल मेरा ये है उसके पढ़ने पहले हमारे की शायद हाँ संभव भी आज अच्छा नहीं
इनका लैपटॉप
सुनो हाँ उनकी हो गई है देन डॉक्टर मनोज कुमार की नहीं डॉक्टर राजाराम
सर ये भी फ्रीज कर रखा है।
तो लाइट और इनमें थोड़ा लेट शुरू हुआ आज लेट आए थोड़ा सा हाँ
सर वो कैमरे के लिए ऑनलाइन कर रहा है तो इसके लिए फ्रीज कर रखा है, आपको चेक करना है?
अच्छा देखते कैसे दिख रहा था अगर एक स्लाइड दिखता है ऐसे दिखेगा अच्छा ठीक ये तो शायद डॉक्टर शंभू सिंह yes, it's true she's there already. Madam, A warm welcome to everyone for the Symposia 12, which translational insights on cardiovascular disease and therapy. I have the honor to invite Dr. Adepalli and Dr. Ashutosh Kumar to chair the sessions. Sir, please. Dr. Adepalli is a distinguished pharmacologist with a career spanning over 4 decades in academic leadership, industrial drug discovery, and clinical research. दे दिया. Dr. Ashutosh Kumar is an assistant professor in the Department of Pharmacology and Toxicology at NIPER, Mohali, with over 15 years of research experience in pneumopharmacology. I request our both chairs to kindly introduce the speakers.
So, good morning. Welcome to this session where we have the first speaker as well known Dr. Harlokesh Yadav. So, he's a highly regarded pharmacologist and additional professor in the Department of Pharmacology at AIMS, New Delhi. And he has like research interests covering areas like molecular pharmacology, cardiovascular pharmacology, nanopharmacology, and he specializes in cardiac ischemia, reperfusion injury, cardiac hypertrophy, and so many. He has been honored with many accolades and awards, to mention few, like Dr. Manjeet Singh Award for Molecular Pharmacology, and then Datta Prize for outstanding published work in IJP. And beyond his research and teaching, responsibilities, he is also active member of scientific community. He serves as secretary of the IPS and fellow of both National Academy of Medical Sciences and National Academy of Sciences, India, NASI. So with this brief introduction, we would like to invite Dr. Harlokesh to deliver his.
Thank you, my dear friend Asutosh and Dr. Adapalli. And if you are talking about the vitamin D, and here we presented, we will present two research work, one on the animal research work and one is the clinical trials, and both are related to the vitamin D. And one thing is that in one animal study, what we reported that uh, uh, we correlate the severity of disease. The patient we are coming in our OPD, most of the this pulmonary hypertensive patient, we are having the very very low level of the vitamin D. And the you see the expression of the things which are responsible for the severity of disease, that is the hypertrophy, that is the TGF beta, and nitric oxide, that is the having the totally opposite effect of the nitric oxide. TGF beta, both are governed by the vitamin D. So this is the our published work where we evaluated the you see the effect of the vitamin D on the pulmonary hypertension by an animal model. And if you see the oh hey compared point here. You see the you look inside the heart, right and left, the right ventricle which develops from the other type of the ventricle, and left ventricle is developed from the other type of ventricle. And if pressure get increase on the left ventricle, you see left having the inner right off. That's why the heart failure case, it means a lot having the very long time. But in the case of the right ventricle, like I previously said, you see, severity of diseases when pressure get increased on the right ventricles get hypertrophied, get sudden cardiac arrest, and not only it limited up to the right ventricles, it get radiated toward the pulmonary artery and it get further radiated toward the lungs. And most of the you see the pulmonary hypertensive patients having the um, lungs fibrosis and severity of disease is very very high. If patient is not treated, the total life span of patient is 2 years. 2 साल में मरीज को मर ही जाना है. After treatment of the all the drugs, you can increase only up to the 9 years. That's why this is the totally fatal disease. And most of the patient of the pulmonary hypertensive patients having the severe deficiency of the vitamin D. So we try to correlate and okay, and the when deficiency of vitamin D is there, TCF beta expression is more. When the deficiency of vitamin is there, inos expression is less, and both are acting oppositely to each other. So what we do here in first study, we कोई बात. So we divided our animals in two sets. One is the normal, one with a vitamin D deficit, and both were exposed to the same dose of the monocrotaline. And in normal group, one group before giving the monocrotaline is agent which is responsible for the induction of the disease, and one set of the animal were pre-treated with the vitamin D to find out the very clear cut effect. And one group of the animals having the normal vitamin D subjected to the monocrotaline. And other hand, one set of the animals which were the uh, vitamin D deficient, and they were exposed to same dose of the monoprotein, and we correlate the severity. Okay, and we try to validate our study by in cell line, and to mimic the vitamin D deficient model of the rat, what we do, we silence the receptor of the vitamin D, and both one set of the cells is the normal cells, endothelial cells, one set of the cells of the smooth muscle cells. Okay, and to mimic the vitamin D deficiency model, we silence the vitamin D receptor, and both cells were exposed to the TGF beta, and one set of the cell were pre-treated with the vitamin D because active form of the vitamin D is the calcitrol, and so we used calcitrol, and one set of the cell were treated with the calcitrol, and one set of the cells were treated with the calcitrol before exposure of the TGF beta. And you see here the expression of, you see, the serum level of the vitamin D uh, is very very poor, and the expression of the receptor in the silent cell were significantly decreased. And here, and this is the experimental protocol. One, it is the normal rat were exposed to the monocrotaline. We use two doses of the vitamin D. One is the 100 international unit. One, one is the 200 unit. You see, and one set of the rat were pre-treated with the vitamin D before giving the monocrotaline, and because it is mediated by the nitric oxide, and to evaluate the effect of nitric, we give it the inose inhibitor, that is the LNAM. It is the same model of the deficient rat, because and we use same dose of the monocrotaline. If you see the body weight were significantly decreased here in the case of the diseased rat were exposed to the TGF beta, and we use the sildenafil as a standard drug, and treatment of the sildenafil where the body weight were restored, and it is the 100 in the lower dose, it is the higher dose, and it is pre-treated. It means the rat were pre-treated with the vitamin D, where the falling of the body weight were minimum. And same case in the case of the here, uh, we used, point is not working. Okay, in the deficient rat, we use the treatment of sildenafil having the equal effect and the lower dose of the vitamin D, that is the 100. And here, lower dose is unable to significantly increase the, you see, the body weight, and is the same effect in the case of the lungs weight to the body weight. This is the how we diagnose the pulmonary hypertension in the rats by the echocardiography. Here, you see the this is the right ventricle flow tract. In the normal rat, this tract is, when you compare the aortic tract, it's very very lower in diameter as compared to the aortic tract. But in the disease rat, you see the diameter of right ventricle airflow tract is significantly higher. This is the right, the same thing, the right ventricle flow tract by the aortic flow tract and normal and deficient tract, but we did not find any significant effect on the left ventricle. Means left ventricle is not totally affected. Okay. हाँ. Slide कैसे बढ़ेगी?
स्लाइड सर शेयर करके ये पॉइंट
Slide only बढ़ेगी. So anyway, he okay. In the case you see here is the we measure the right ventricular pressure in the normal rat, it is near about 24, and in the disease rat, it is shoot upon near about 40, and in the treated, it get fallen near about 20 to 25. And if you see the right ventricular systolic pressure in the case of the disease rat, where it get increased from 20 to 40. We use same dose of the monocrotaline, but in the case of the vitamin D deficient rat, the right ventricular pressure get increased from 20 to 60, more than 60. We use same dose of the monocrotaline. In one set of the rat, in the normal it get increased 20 to 40, but in this deficient rat, same dose of monocrotaline increase the pressure near about 75% more. It means vitamin D deficient subjects are more susceptible for the severity of the disease. This is the first time we have reported in literature. In the there is no any significant effect on the left ventricle. Same things in the right ventricle hypertrophy index that was significantly lower, decreased by a higher dose of the vitamin D. And in the case of deficient rat, if you compare, here is the lower dose of the, that is the vitamin D is unable to decrease the right ventricle hypertrophy index, and same dose of the this vitamin D were significantly decrease the right ventricle hypertrophy index. Same effect we have observed in the, and this is the histopathology, and here you see the diameter of the pulmonary artery in the normal rat, and in the disease rat where the clear cut thickening of the pulmonary artery, and that gets subsided by the treatment of the either the standard drug or by the vitamin D. And in the expression of the enos where significantly decrease in the disease rat, and TCF beta expression were increased, and same thing in the alpha SMA where get increased. Same thing say as well as the normal and deficient rat. This is the expression of the mRNA level. He the expression of the mRNA enos expression, mRNA of this enos were significantly decreased in the disease rate, and get reversed by the treatment of the higher dose, and this in the lower dose were unable to significantly increase the expression of the enos in the normal rat, but in the disease rat it significantly increases the expression of the enos. You see how the vitamin D deficient subject are susceptible for the vitamin D. And we found the same effect on the TGF beta. You see the expression of TGF beta were significantly increased, and that get reversed by the treatment of the vitamin D. And this is the expression of the alpha SMA, and where it gets significantly increased in the disease rate and get significantly
Okay,
decreased
you
by.
can conclude in 1 minute, Dr. Lokesh.
And you see the expression in the cell as we find the similar effect in the isolated cells. It means the vitamin D deficient subjects are more susceptible to the severity of the disease, and this uh, effect of the vitamin D is mediated toward the by enos and alpha. This is the animal study, and we conducted one trial. This is the first trial in India in ACS patient. We have evaluated the effect. Just give me 3 minutes, if you permit. 3 minutes. Just we try to correlate our animal finding toward the directly to the clinical trial.
2 minutes, go ahead.
बहुत कम टाइम है इसके, मैं आधा घंटे के लिए मून माइंड बना के आया था
अरे बढ़िया.
और मैं ही आके
Go
घबरा
ahead.
गया.
No, I'm just testing you. Go ahead.
You see this effect of the vitamin D, how this how the you see the severity of disease get increased in deficient patient, and lower dose were unable to effect reduce the pressure, reduce the expression, but in the deficient it's highly susceptible. And we try to correlate this. It is our team And this is our clinical trial where we did trial in first time in aims in remote aortic preconditioning. What is happening in ACS patient? When patient come to the rush to the emergency, and there's only one things we have to implant the stent. And before implantation of the stent, there was ischemia. And during the implantation, after the implantation of the lethargy perfusion, and the unavoidable injury. This is the ischemia perfusion injury. What we did, we just occluded this 5 minutes of this one of the upper arm and repeated 4 cycle. And when you occluded this, when occluded this one of the arm, some mediators get released, which get circulated in all over the body. When it enters the heart get adapted to, to tolerate the ischemia perfusion injury. And this is the first time of the, we have a lot of the animal work, but is the first trial. And we what we did, we when the patient came into the emergency, we just took the sample. And what we did, just evaluate the correlate cardiac injury markers with the vitamin D level. And most of the deficient patients were having the higher level of the CKM, higher level of the LDL, high level troponin, not in the cardiac marker, inflammatory markers will get increased. And when we, you see, when we put at the fourth cycle of the ischemic preconditioning, by including one of the arm, the this marker will significantly decreased. And this if effect of the remote ischemic preconditioning were correlated with the vitamin D. I think it is the first type of the trial in literature we conducted in AIMS. And we are, आपने हमको टाइम नहीं दिया,
नहीं दिया ना
and this is our work.
तो अब भी दे रहे हैं go ahead
So, so,
Skills are generous enough.
so this is our team.
very
And after the instant implantation, when in one subject, a patient were just only control with the instant, and other subject patients we given the 4 cycle of the ischemia preconditioning, and in this treated ischemic RIPC group, all the markers, cardiac injury markers, inflammation, we measure. These are the markers. You see, the CKMB, um, LDS, myoglobin, whole blood glucose, troponin weight gets significantly lower. And these are the inflammatory markers where get auto decrease. This means remote ischemic preconditioning not only decrease the cardiac markers, it also able to decrease the inflammatory markers. And this effect of the RIPC is directly related to serum limit of the vitamin D. This is our team. I'm the PI of this trial, and this is the head of the cardiology. This is our team. Thank you very much.
thank you very much interesting study Dr. Harlokesh. I'm sure it is a very interesting study and one point very clear before I leave it. In fact, as we know, it's a global health problem, cardiomyopathy.
Yes.
And that is acute cardiomyopathy. And truly speaking, as sir was talking about vitamin D, pericalcidae,
Hmm.
has been proved to be in kidney diseases already. Now, the trend is coming how it can modulate, it can reset, remodeling mechanism. In that sense, this paper is very much interesting. I am sure your study in the clinical setting will be very fruitful and all the best for your.
Thank you, sir. Thank you, sir.
So we would like to take one or two questions.
Pharmacological, as you know, you might have done the vitamin D level 125 dihydroxycholecalciferol levels in volunteers and basic. You'll find surprisingly, although we have ample UV light and sunlight, 99% of our population is vitamin D deficient.
deficient. So what is your recommendation in normalizing the vitamin D levels?
देखो इसका just adapt to the your natural setup. Do do exercise. Try to expose yourself in the sunlight. If you can't do anyway, your personal problem, you can take vitamin D 60,000 unit near about per week up to the 6 weeks minimum. And by the time to time, you must not only vitamin D, you also you can measure your vitamin B12. These two things, vitamin D3 and B12, this deficiency is the common problem. And both not only affecting the UC, vitamin D is affecting the calcium level, vitamin B12 affecting the blood level. No, it can regulate the whole of the UC body. All most of the cardiovascular and even the CNS function get impaired by the severity of this.
Congratulations for all your great work. The thing is that even you talk about exposure to sunlight, uh, we had done one small study amongst the footballers. They were all professional footballers, always doing practice under the sun, but they are having almost 30% of them having vitamin D deficiency. And so this is not only the sunlight exposure, but I think the pollution that also matters, what type of ray we are getting. And another important aspect is the magnesium. Uh, Magnesium is an important cofactor with vitamin D, and I think this is why now the combination of magnesium and vitamin D is very much a popular drug for all of us who are practicing in the field of diabetes or cardiovascular disease. We are using this, they had shown certain effects on blood pressure reduction also. So I think you may find out certain new experimental thinking on
Evidences.
with this
Yes.
magnesium and vitamin
Dr. Shambhu, one thing. No, no.
Crucial role.
Thank you, Dr. Shambhu, for your very good and information. We measure the level of most of the element. Dr. Asutosh, magnesium, uh, zinc, selenium, aluminum, iron in anemic patient. You will surprise. You will surprise patient. Doctors are frequently prescribing selenium, but at the certain level of selenium, it is toxic. Magnesium, copper level, most of the Indian population having the severe level of copper, but aluminum level is the 100 times greater than the Indian toxic level. अल्युमिनियम लेवल वी फर्स्ट टाइम, और आपको बताऊं मेरी हिम्मत नहीं हो रही कि इसको मैं पब्लिश करूं. First time we measure the aluminum level in the population.
Sir, one question. Sir, like you showed this vitamin D3. So there are many, I just want curiosity out of curiosity. Vitamin D3 induced, there is a lot of preclinical models of inducing cardiovascular calcification. If you give too much of vitamin D3 in animal models, you can induce heart and the vessel calcification. So, in my curiosity, people use dosing vitamin D3, although I understand the dose is little higher, in which probably you have used a study. But long-term vitamin D3 taking by the human subjects and all those things, do you think that it may cause cardiovascular problems,
No, it's
calcification?
very toxic. Problem is that it's a lipophilic drug. Don't take vitamin D 60,000 unit. Uh, it is recommended to the continuous for the 4 week and followed by uh, 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 per month, and don't exceed beyond the 2 months. Total dose is 8 dose. If you increases the dose, it is a lipophilic drug. It gets accumulated. What doctor is saying, it is a very toxic drug. Thank you.
Before that was introduced in the next six, I just wanted to say to them, youngsters, those who are interested, even you can try in that, in that. In fact, it is also very good. One of the methods we
Yeah. So, yeah, thank you, Dr. Harlokesh, for discussing and bringing it this important, translational studies, and which will open definitely like normalizing the things, looking about this like calcium level along with other macronutrients. So that is very important and integral part. So thank you all.
So we would like the session chairs to kindly felicitate Dr. Yadav. So can you stand against the background? So मेरे को लगा उनको बार-बार ऊपर आना पड़ेगा. Can you burn your hand? Can you ready? Can you cheat?
So now we have our second speaker of this session, Dr. Shambhu Samazdar. So he is a distinguished clinician researcher in Kolkata and associated with Allergy and Asthma Treatment Center at Kolkata. And he is MD in pharmacology. He has also like postgraduate diploma in clinical endocrinology and diabetes from Royal College of Physicians, UK, and fellowship in respiratory and critical care from WBUHS, and he has published extensively, contributed to studies in prescribing practices, patient safety, and presented at national and international forums. So, with this brief introduction, we would like to invite Dr. Samazdar to deliver his talk. And, sir, one small request, please try to finish it in 15 minutes, yeah, so that we can
I had a
session. Thank you.
knowledge that we have a 20-minute discussion. So, okay, no problem. You please let me know when to stop. The thing is that this is an important concept now, the clinical pharmacology OPD, to ensure better patient care. So, as we want to talk about allergen immunotherapy more precisely, we need to understand clinical pharmacology, the discipline could be bedrock of any precision drug therapy, but also to allergy therapeutics and allergen immunotherapy. So let's first understand what is clinical pharmacology. So it is nothing but the study of drugs in humans. But what I want to emphasize, according to NMC, who is a clinical pharmacologist, they should have a knowledge of pharmacological principles and a good knowledge of clinical medicine. So there should be a good fixed dose combination of two things. One is clinical medicine and another is pharmacological principles. And for that, yes, in our country, we have the DM degree, DM in clinical pharmacology. But at the same time, when we have MD in pharmacology, MD in general medicine, MD in psychiatry, MD in anesthesia, MD in pediatrics, if they are being trained sufficiently with clinical pharmacology, they could be considered as clinical pharmacologists. Uh, this is very important to understand first, and then what are the services we can provide to clinical pharmacology OPD, whether it is actually required or not. As we are moving towards the precision era and we want to reduce this burden of inappropriate prescribing, polypharmacy is sometimes legitimate, there is a legitimate need of polypharmacy, but sometimes we need to avoid those potential inappropriate prescribing. We need to promote those evidence-based decision making while providing drugs at the point of care, and how we can bridging the prescriptions with patient physiology. We often think that prescription is nothing but a piece of paper, but we forget that it is a prospective relationship between me, the prescriber, and my patient. And that is where clinical pharmacology plays critical role. And this could be an important platform for pharmacovigilance, adherence, and drug rationalization. So these are the few important role of a clinical pharmacologist at OPD. And I think the most importantly, what we want patient oriented evidences that matters, that poems need to be given more emphasis than the disease oriented evidences. And these are important nowadays because we are having multimorbid patient, they are being exposed with irrational drugs, and we are moving towards patient-centric pharmacology. And clinical pharmacology, OPD, could be considered as the nucleus of this precision care in real-world clinical medicine. This is what we had proposed with my guru, Professor Shantanu Kumar Tripathi, that CPRRF, Clinical Pharmacological Reconciliation Review and Feedback. This could optimize the patient care by balancing the risk versus benefit of ongoing therapy and at the same time improving the adherence to the drug. So as today we are going to discuss about allergen immunotherapy, little bit we need to understand the basic pathophysiology of allergy. Allergy is nothing but this type 1 hypersensitivity. All the other type of allergy I am not going to discuss because allergen immunotherapy is for allergy that is type 1 hypersensitivity. So most important defect in allergy is the epithelium, the defect in epithelium. So allergen will enters through those defective epithelium, and this will be captured by the antigen presenting cell, the dendritic cells, and they will bring those allergen towards the lymph node. And in the lymph node, there the T cells, the naive T cells are waiting, and they will have a handshake with antigen presenting cell carrying the allergen, and there will be a play of those co-stimulatory ligands who are actually doing those handshakes. Depending on the nature of the handshake, the cytokines will come out, will have the interleukins 4, 5, or 13, and they will make the T cell, which was naive T cell, converted into Th2 type of cells, and those Th2 type of cells will ultimately tell the B cell to produce IgE instead of IgG. And we will have IgG, sorry, the reverse, the class switching should be there. Normally what happened, we are getting IgG from our B cell, but here by the virtue of this handshake, the IgG will not be produced. There will be class switching and there will be IgE production, and those IgE will go to the mast cells. The mast cell, you can see the Fc epsilon receptor on top of the mast cell, and the Ig will sit on top of the mast cell over those Fc epsilon receptor. This is the stage where we called as sensitization. Now again a new allergen will enter, and that allergen will be clasped by those IgE who are sitting on top of mast cell on those Fc epsilon receptor, and there will be mast cell degranulation. We will have histamines and other mediators. Now, whatever pharmacological agents we have, they will act here only. They will neutralize histamine. They will neutralize those leukotrienes, the inflammatory mediators. So we will have antihistamine, we will have steroids to treat. We want something more. And for that, the allergen immunotherapy matters because we are repeatedly administering some specific allergens here, which is going to provide a long-term benefit. This is nothing but a hyposensitization and modulating the patient immune response. So they are targeting the T lymphocytes. In allergy, who is the master cell? The T lymphocytes are the master cell. So allergen immunotherapy is the only therapeutic option we have which target the T lymphocytes, and therefore we will have more Treg cells and they will suppress those allergic mediated reactions. So ultimately there will be an immunological shift. The Th2 to again we are getting back those Th1 responses. So all the cells you can see, this is in a nutshell the mechanism of action of allergen immunotherapy. They will increase the Th1 activity again. So the class switching, which was previously from IgG to IgE, again we are getting back IgG. We are getting back the blocking antibody IgG4. So ultimately it will translate in improving the quality of life, decreasing the symptom score, medication score, and objectively we can find the skin prick test reactivity will be decreased. These are important indication of allergen immunotherapy. We can see starting from allergic rhinoconjunctivitis to asthma to atopic dermatitis, the venom hypersensitivity, and sometimes
In atopic dermatitis, it could be considered as first line treatment. These are few important contraindications that we need to remember, especially when the asthma is uncontrolled. We are having markedly compromised lung function, some heart issues, some organ dysfunction. For an example, the end stage kidney disease or Child-Pugh C score, chronic liver disease. There we need to avoid this immunotherapy. And if the patient is on beta blockers, we need to be cautious, especially if the patient is on non-specific beta blockers, then we need to sometimes avoid using allergen immunotherapy. So the risk benefit analysis should be done. And sometimes in case of SE inhibitor as a concomitant medication, we may need to consider whether we will start or not start allergen immunotherapy. So these are important points when it works: proper patient selection, proper antigen selection, proper composition and administration. Generally, the dictum is if I am dealing with a young allergic patient, early form of his disease and compliant, they could be an ideal person for allergen immunotherapy. But when we have long-term disease, non-allergic disease, wrong antigen selection, poor quality of antigen, and there are multiple comorbidity, we are not getting the result. So these are the schedule of subcutaneous immunotherapy. First three vials we need to give as a build-up phase. Then we start the maintenance phase vial. Initially, it is 1 to 2 times per week. Then it gradually the frequency become 1 to 2 times per month, and we need to continue that for 3 to 5 years. This is according to our guideline, allergen immunotherapy guideline, by which we should prescribe subcutaneous immunotherapy. This is just an example by how we are practicing allergen immunotherapy, the subcutaneous immunotherapy. These are few important factors we need to consider which increase the clinical efficacy of allergen immunotherapy. Again and again, good compliance and adherence with 3 to 5 years of allergen immunotherapy is extremely important. And when to discontinue the therapy, if I am not getting benefit after 1 year, we need to think to stop it. And there are no specific biomarkers available, but sometimes we use to estimate the IgG4 ratio, and therefore we can continue think of discontinuing the immunotherapy. These are few important local reactions we need to think, and local reactions are not going to predict. Even if it is large local reaction, it is not going to predict severe anaphylaxis or like that. But for subcutaneous immunotherapy, we need to be cautious on anaphylaxis. The efficacy data already it had been proven that, and that is why all the global guidelines starting from GINA to ARIA all are recommending this allergen immunotherapy. Now, what could be the role of clinical pharmacologist in allergen immunotherapy? First of all, we need to understand the drug-allergen interaction oversight, how the allergen immunotherapy introduction could reduce the medication score, and therefore it will translate into unnecessary adverse drug reactions with multiple drugs. For an example, we all know there is a black box warning now with Montelukast, but our poor allergy patient, they are having to continue Montelukast for years. So if we use this allergen immunotherapy rightly, that can reduce the medication score, and therefore we will have less adverse drug reactions. Monitoring therapy response and the counseling on adherence, how they should adhere to that 3 to 5 years protocol. So as lack of time, I think I am just going to this care loop. So first you have to detect the patient properly, and history is the most important part to select patient. And then you have to do the sensitization test, the skin prick test. After doing the skin prick test, you have to again take a second history, and that second history will confirm the sensitization status. And then you have to select the allergen. You need to start allergen immunotherapy, then monitoring and adjustment. You need to measure the outcome, and you need to ensure the long-term adherence and precision reassessment. These are few studies we had done with allergen immunotherapy, and in ocular allergy also this is recommended this meta-analysis, we had shown that sleet and skeet reduces symptoms in allergic rhinitis better than sleet. But in terms of medication score, in terms of overall quality of life, there is no much differences. But of course, in our country, the sleet is very costly compared to skeet. And this is the future scope. I think scientists need to work more on genomic biomarkers because that will help us to have a predictive allergen immunotherapy responses. This could be some important targets for genomic markers. So this is a future that with patient DNA we can understand the predictive monitoring of allergen immunotherapy. One of the very important part while doing allergen immunotherapy, the shared decision making. And that is also another important duty of clinical pharmacology OPD. We need to ensure that the shared decision making should be properly done. So these are few important uh, s- points need to be mentioned during the informed consent process.
So Dr. Shambhu, please
Yeah, I have, I think, one or
you.
two slides. So these are the counseling things, and interdisciplinary approach is important while we are talking about allergen immunotherapy. We need to have the other specialty in our team like ENT, pulmonology, pediatrics, and the pharmacovigilance team. So there is an important role of clinical pharmacology OPD who could have this initiative of starting allergen immunotherapy, because when we talk about clinical pharmacologist, it's not just an expert in medications. They are the conductor of collaborative allergy care. So we want from referral to relief, and that is why this is what I am not going in this case, but yes, this is one. This by these steps we can achieve better outcome. And now the AI is coming in a bigger way. We have chatbots who can ensure the adherence processes,
Yeah.
and
So now we
we
will,
can.
yeah, please conclude, sir.
So this is my concluding slide. I shall stop here only. The history is very important. While allergen immunotherapy need to be considered, the clinical examination, you need to be master on skin prick test and second history taking. These are the points. So I can finish here. So if we have some questions or
Yeah.
comments, we can
So,
take those.
so thank you, Dr. Shambhu. So we will take upon one or two questions.
Yes.
Maybe you could put this, we can face you. You know, that is enough. Yeah. See, rightly you mentioned biomarkers need a hour as a translation. That is being seen. Wonderful. When you wanted to suggest AIT therapy, what would be the preferred, user-friendly method of using?
If
Preferred.
we had mentioned in one of our slides, can I have my?
No, no, forget about
You can.
it.
So so the thing is that the subcutaneous immunotherapy, as of now in India, this is the better choice. Why I am telling this? The SLEET, the sublingual immunotherapy, is also available, but standardization is a big question mark. Now, one company, the Dr. Reddy, they had bring one Sensimune tablet that need to be given daily, but the cost is very high. So when we talk about the cost of subcutaneous immunotherapy, it is around 400 rupees per month. Whereas the SLEET, the sublingual immunotherapy, 9,000 rupees per month. So that is what we need to decide.
Any further questions from the audience? Young minds?
Yeah. Actually, the translation cardiovascular chemical. I think young minds should find out.
So still, I think after breakfast, glucose has not reached to the brains.
But
Okay.
I had a doubt because when the this session being named under that cardiovascular banner, so I was a little bit surprised. But there is one thing that when we talk about allergen immunotherapy, actually we are going to reduce the medication score. And as we are going to reduce the medication score, I think many of us in the who are doing pharmacovigilance practice also, they will find out the lava ICS combination, they are leading to different type of arrhythmia. There is maybe
I don't know whether that is the thought or somehow they want to accommodate this lecture.
So thank you, Dr. Shambhu. So
Thank you, Dr. Shambhu, for the insightful talk. Request the chairs to kindly give a token of gratitude to Dr. Shambhu.
So next speaker is Dr. Manoj K. Bartwal. So Dr. Manoj is a senior principal scientist at CSIR CDRA Lucknow, and his research focuses on molecular mechanisms underlying cardiometabolic disorders, inflammatory pathways, which are linked to atherosclerosis and metabolic diseases. And he has published extensively in peer-reviewed journals and has certain patents to his credit. And his work at CDRI has helped to bridge the fundamental immunology with the translational discovery aimed at inflammation mediated metabolic condition. So, with these few words, we would like to invite Dr. Manoj to deliver his talk.
Okay, thank you. First of all, I would like to thank the organizers for this wonderful event and for the invitation for delivering the talk. So today, what I am going to do is that I will take you through a journey which our lab undertook briefly, and then also tell you the importance of inflammation and innate immune signaling in cardiovascular diseases and especially heart failure also. So, what does immunotherapy or inflammation, what promise does it hold in heart failure and cardiovascular disease, and what research has been done, and where do we actually stand at present? So, some slides will be of academic interest for the students, and some will cover our lab research. So, this chart, it is mostly for even for the students to see, and it is a timeline of major milestones of research connecting inflammation with cardiovascular disease, the topic of focus today. And here you can see that historically from the 1850s, where inflammation, cell death, proliferation in atherosclerotic plaques was described by Rudolph. And everybody knows the NHR induces atheroma in cholesterol-led rabbits. This was a classical experiment done in the 1913. Right from there, if you travel the path through the early 1960s, the Framingham study, everybody knows about that. In the 1944, that's so early, the approval of the statins. And when you come to the like the inflammation, you see in 1994, CRP was found to predict bad outcome in unstable angina. That was also a landmark study. And then subsequently CCL2 and CCR2 receptors, the chemokine receptors for the leukocyte migration, their importance in cardiovascular diseases is 1998. And then subsequently PCSK9, this is a lipid lowering LDL receptor degrading enzyme. Its importance came up for LDL uptake and atherosclerotic cardiovascular diseases and the blockage of the vessel to the heart. So this was also an important event. Then clonal hemostasis, where you have more immune cells in 2014, and they were found to be linked to the high risk of cardiovascular diseases. Then we had approval of PCSK9 inhibitors, and you have monoclonal antibodies there for the lipid lowering and preventing the cardiovascular events. Then in the 2016s, we had GLP-1 receptor agonist. And more importantly, there from the inflammation point of view, there was a CANTOS trial. And here it established the anti-inflammatory therapy in atherosclerotic cardiovascular diseases.
And this was found that the anti-inflammatory therapy was independent of lipid lowering. So this connected directly the inflammation with the cardiovascular diseases. And in 2019, the Colcott study shows anti-inflammatory effect of colchicine, and subsequently single cell immune landscape of human atherosclerotic plaques was also generated. And the most important landmark was in 2023 when colchicine was approved by FDA as a first anti-inflammatory therapy for atherosclerotic vascular diseases. So this was just to take down you the memory lane and show how the field of inflammation evolved in the cardiovascular diseases and what is where it stands today from the treatments perspective. And this is a map which usually 2016, and it shows the prevalence of the cardiac diseases. And this is something which was there in the literature from before, but this is the latest stats which we have. And here you can see that there are a lot of reds in the ischemic heart diseases, cardiovascular diseases, stroke, and you can see the most red part. Presently, we are doing the presentation here, but you can see that the Punjab is like is occupying a huge it's a dark red color. So we all need to think like what are the reasons where like lot of incidences of these diseases are happening in this part of the region of India. So this is a country-wise statistics, and here you can see that everybody is well aware that cardiovascular diseases leading to the morbidity and mortality. And then again, as you see that inflammation and heart failure, you can connect that there are several aspects and inter-organs speak, which makes this more complicated, like from humoral immunity and skeletal muscles, GIT is involved, leaky gut leaks the LPS, and then you have rheumatic heart disease. So, and trimethylene N oxide is there, and bacterial products. Adipose tissue also secretes and from the spleen and pro-inflammatory. So this becomes a multi-organ crosstalk where the heart is affected by inflammation. And you can see that already there are several biomarkers which are already established with respect to heart failure, like the CRP, and then the roles of neutrophils, the cytokines, the galectin 3, non-natriuretic peptides, and the RAS biomarkers. So the point to show you all this slide is to build you and show that how the inflammation has taken also an important role in the patients of heart failure. And this is something, colchicine story I already told you, but this is something where anti-inflammatory summary of clinical trials of anti-inflammatory drugs for heart failure. And here you can see that there have been successes, there have been failures, but if you can see that with prednisolone or pentoxifylline, you can see that there is some improvement in symptoms of left ventricular ejection fraction and thalidomide. So there is some improvement. So some people have tried various drugs for the clinical trials, and some promising effect have been seen. Unlike like TNF, like an attached 2003 trial, or again Intercept 2004 trial, there was not much significant outcome. You can see the outcome was not significant. So this has been anti-inflammatory therapy has been a mixed bag. There has been some protection has been seen, and somewhere it is not. Now, Cantos trial, as I showed that the FDA approval for atherosclerotic cardiomyopathy, you can see that this antibody alpha beta has significant trend for dose-dependent reduction in the heart failure patients of hospitalization. And IL beta, this signaling, I would also emphasize in some of our data that it is a signaling mediated by IL-1 beta receptor, and there it leads to the hyperinflammation. So basically, quenching the IL-1 beta provides cardiovascular protection. Some examples are also here. So this is the thing that where people have conducted trials and seen that what is the outcome in the heart failure patients. So specifically what we are discussing today are some hypertension associated inflammatory changes and hypertensive heart failure, and today's some data will revolve around that. So for this, I will skip some introductory slides, which have again. So here you can see that the activation of innate immune system by DAMs and PAMs is very important for the amplification of the inflammatory response. And innate immune signaling is often activated and after detection of pathogens by pattern recognition receptors, And these pattern recognition receptors recognize the PAMs or the DAMs, that is that activate these receptors and lead to the downward signaling. And here on the right hand side, you can see that TLR or interleukin 1 receptor associated signaling, it activates a family of kinases which are called IRACs. And these IRACs are the interleukin 1 receptor associated kinases which are activated by upon the stimulation of IL-1 receptor, which can be an IL-1 body, IL-1 beta, or TLR also receptor. These are some of our previous work where we have shown that oxidized LDL is also a kind of PAM because it activates TLR and generates an inflammatory response. Some of our previous laboratory published work will not go into the details. And in fact, this PAM, you can say oxidized LDL through TLR, it leads to the accumulation of lipids and contributes to the formation of atherosclerotic plaques. And this is one of our work where we had shown that in new intimal hyperplasia induced by a balloon injury in animal models, if you inhibit the IRAC, that is an innate immune signaling kinase, you see protection in new intimal hyperplasia. And here we had identified a mechanism. So presently, what we have done that we have studied the role of this kinase by using a pharmacological inhibitor IRAC14, and we have taken a angiotensin induced cardiac hypertrophy model where we have infused angiotensin into these animals. And after 28 days, we have, after the continuous infusion of the angiotensin and treatment with the IRAC inhibitor, we have evaluated the all the cardiac parameters in this animal model. So we have tested this inhibitor both in preventive mode and the therapeutic mode in both. And here you can see that this slide summarizes that the angiotensin is inducing a very high hypertrophy. And you can see the reduced ejection fraction and the fraction shortening. And in fact, with the IRAC inhibitor, when we have pre-treated the IRAC inhibitor, there is protection in these hemodynamic parameters. Similarly, if you see cardiomyocyte cross-sectional area, if you see the cardiac hypertrophy gross heart pictures, if you see the fetal gene expression of hypertrophy, all are kind of inhibited when you treat these animals with IRAC14 inhibitor, meaning to say if you inhibit innate immune signaling kinase IRAC, there is inhibition in cardiac hypertrophy. And then subsequently, we have done this in the therapeutic mode also, where we have first infused angiotensin for 15 days, and after 15 days, we have infused IRAC14 inhibitor. Until 28 days, we have done the treatment. And there also we see the protection. So meaning to say, inhibiting IRAC in both preventive and therapeutic mode, we do see protection in cardiac hypertrophy. And this is the same heart size and the cardiomyocyte cross-sectional area. The protection is obvious from on all these aspects. This is now SMA staining and the picrocyte staining for the collagen deposition. In the polarized right also you can see that there is there is lot of fibrosis which is happening, and then you can see that with the IRAC inhibitor there is inhibition in the cardiac fibrosis, indicating that a protection is seen with this inhibitor. This is the data which shows the active. Yes, so 3 minutes is like says 15 minutes. So yeah. So this slide shows the activation of the RAC pathway. The TLR is upregulated on the angiotensin treatment, and you can see the I kappa B degradation is there, which is an indication of NF kappa B pathway activation. With IRAC inhibitor, you see a restoration indicating that yes, IRAC pathway is activated. And here in this slide, I don't know whether you are able to see or not, but you have NF kappa B translocation to the nucleus, which is inhibited in presence of the IRAC inhibitor, indicating that yes, indeed IRAC pathway is involved. Again, TGF beta SMAD, we evaluated the fibrosis pathway, and IRAC inhibitor does inhibit the TGF beta SMAD pathway. Like this, we have demonstrated the inhibition of fibrotic signaling. We have seen the activation of IRAC by after addition of angiotensin in the cardiac myocytes, showing that yes, indeed, cardiomyocyte signaling is affected. We have seen the cardiomyocytes in the cell lines, the hypertrophy and the inhibition by the IRAC inhibitor, and the inhibition in the hypertrophy gene expression is seen with these inhibitors, IRAC inhibitor. And this is the overall summarization of the data. And here you can see that angiotensin induces the TLR4 and the NF kappa B pathway
inducing inflammation, and the TGF-beta SMART pathway inducing the fibrosis, and this is overall leading to the cardiac remodeling. But in presence of the IRAC, if you inhibit the IRAC both in preventive and therapeutic mode, you do see a protection. So this study again highlighting our like kind of line of work where we see that inhibiting innate immune signaling does have some promise in heart failure. So this shows an overall summary of the word that ARAG mediates angiotensin-induced cardiac hypertrophy, and its inhibition both preventive and therapeutic mode is helpful. We have identified the NF-kappa B pathway and the TGF-beta 2 SMART pathway, which are involved in the inflammation and fibrosis respectively. And we speculate that this innate immune signaling kinase is involved in cardiac hypertrophy. Lastly, we will just acknowledge the support of the students. The unpublished work was basically done by Shreya Gaurav, and the facilities, echocardiography, intravital, Dr. Kumar, Pankaj, Anil, they all helped with the work, and funding from UGC, CSR to the students, Department of Biotechnology, Ministry of IOS, Center of Excellence, and Swiss National Science Foundation also we have. So thank you to all. Thank you.
Yeah, thank you, Dr. Manoj. So now the forum is open for questions.
Thank you, sir, for your this extensive research. The thing is that when you were talking about that IRAC pathway inhibition, so mainly you were talking about that RASH pathway.
Which one?
RASH pathway. So
RAS.
angiotensin that you had shown that na, and you after angiotensin 2 that
Correct, correct,
mediated
correct.
the
Right, right.
reaction.
Right.
So you are modulating the RASH system, right? So the thing is that already we have drugs which had shown a clinically significant benefit in terms
Mm.
of even now we have the sacubitril valsartan, then we have pinrenol,
Mm.
everything we have. And
Mm.
if you talk about the relative risk, residual relative risk in a heart failure patient is much decreased there.
Mm.
But what extra advantages on top of those drugs?
Mm.
Have you done any study that,
Correct.
for an example, if a patient is already on those
Correct.
RAS
Correct.
blockers,
Correct.
on top of that you are using
Correct.
another
Correct. Correct.
IRAC blocker. So what
Correct.
will the extra benefit be?
So, very nice question. So, the point is here that what we have shown that the angiotensin also upregulates the TLR pathway, because we have shown that this pattern recognition receptor TLR4 is also upregulated by angiotensin. And we believe that since IRAC pathway also induces the interleukin 1 beta, and which is interleukin beta also acts to the TLR interleukin 1 receptor pathway. So, an IRAC is also involved in interleukin beta production. So, in a way, independent of the RAS, this immunological pathway is also activated during the heart failure, which our present studies showing. So, as you are saying that angiotensin 2 and the RAS pathway is also involved in one type, but by using a innate immune signaling specific inhibitor, you can actually take care of the TLR or IL-1 beta signaling. And in fact, this signaling is also operational in the immune cells. So in fact, for inhibiting it, you can actually inhibit the infiltration of the immune cells to the heart also. So in that way, we think
Yeah.
that this can be a immune cell specific
So
intervention, which
maybe
we
it
have
is
some
like
for
through
protection.
like non-RAS pathway, they are trying to, take care of it.
Thing is that when we are again on top of RAS pathway, you are using this, how much benefit those group of patients we are going to
So yes, the point is that this if you have in heart failure specific population where you have cardio-inflammatory phenotype, that is what the emphasis. It cannot be generalized. So when you are screening the patients with different kind of biomarkers, you will see that subset of patients having different kind of biomarkers, and there are specific populations where you have a overactivation of the inflammatory pathway. So we believe that this kind of pathway will be beneficial for
Yeah,
patients have cardio-inflammatory type of phenotype.
yeah.
Mhm.
It's one niche area that is in the rheumatoid arthritis nowadays we are using tofacitinib.
Correct, correct, correct.
And we know this citinib group of drugs
Hmm.
generally cause thrombosis. Whether you may think about this in those rheumatoid arthritis.
I will tell you, like rheumatoid, RAG inhibitor is in clinical trial for rheumatoid arthritis also.
So that
It is in the clinical
Yeah.
trial.
Yeah, yeah.
So Before we go to the, maybe just I want Sure, to know, in the interest of youngsters.
Sure.
Is there any way a pluripotent stem cell derived mechanism like cardiomyocytes,
Correct.
stem cell death,
Correct.
in the process of this system?
Correct.
Because today, translational and people are looking even stem cell.
Correct. So activation of this pathway in the pluripotent stem cells generating a more of anti-inflammatory kind of phenotype. That can be helpful because it will subside the inflammation and promote the anti-inflammatory phenotype. So that can have some kind of benefit.
Yeah. In one of your slides, there was nuclear translocation you told of NF-kappa B.
Correct.
So, how come like with the, I think IC you have done,
Hmm.
you can actually measure the nuclear translocation?
So what we have done, we have stained with the DAPI and NF-kappa B, and then we have done the co-localization where we have seen the co-localization of the nucleus and the NF-kappa B, showing that it is
So
going
with
inside the
with
nucleus.
that magnification, it would be really difficult to trace out the
We
nuclear.
have used confocal microscopy and the color change, the usual way it is shown that the localization there, that's the same.
Have you also confirmed with Western blotting having a nucleus like fraction, cytoplasmic,
No,
and
that
what's
we have not. Yeah.
that will confirm it because NF-kappa B
Sure, sure.
nuclear
Cytosolic and nuclear.
translocation, or you
Sure.
can do the like cell migration assays are there to find out that nuclear translocation. That will
Sure,
be
sure, sure.
substantiate.
That
Yeah,
can be an
yeah,
additional method to show do
yeah.
that.
So
Sure,
thank you.
sure.
So thank you, Dr. Manoj.
Thank you.
Yeah. Will facilitate.
Thank you, sir, for the talk. Request the session chairs to kindly honor him. The fourth speaker is Dr. Balaram Ghosh. I think he's not there in the department. Request all the speakers to come on the dais and bestow a token of gratitude to our chairs.
The speaker is not there.
Oh, the fourth is not there.
We are in time.
So please come on the dais. I request you.
Well, sir, yesterday I heard you were nearly working on it.
Dr. Adipalli. Kindly join the hands. So actually we were not sure that Dr. Ashutosh.
Yeah.
There's a small tea break. We will join back at 11:40. Please note that the tea and the coffee is being served here only, outside the Audi. अच्छा जाता है। हाँ हाँ हाँ, तेरा खुल जाएगा बेटा।
चेंज करनी पड़ेगी।
कैसे करूँ?
तो अपने लैपटॉप में बोला जैसे हेलो 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 पास तो बोलो फिर कर ठीक है दूर तो बोलो फिर ठीक है क्या प्रेजेंट किसी ने नहीं ना किसी की भी नहीं आई अभी हेलो ज्यादा इको कर रही हेलो 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 सर सारी लाइट्स ऑफ होंगी सर नाम की हॉल का हाँ ठीक है मैं तो चाहिए 1800 जाके कॉपी मार फोल्डर के बाद आखिरी है पेस्ट कितने करनी है ये कॉपी कर ली बात बात कर डेस्कटॉप पे फोल्डर बनाया आपका टी आर है कुछ पाना था ले ला टी आर इसका इसका हो जाते हैं दो और दो ही है बस और दो ना ये कर दो पेन ड्राइव कर दो पेन ड्राइव कर दो क्वेश्चन डिजिटल सेंटर ऑल और एक प्रोफेसर होता है उसका नहीं देवशी से उतना फोन करेगा आई नहीं लैन तक गए फ्री में शायद फोन करिए सूरत सर देवशी छोटा का मैं उनको बुला लूँ अंदर से कोई लाया था नहीं पेन ड्राइव चक के जी तू तो चक के लाया मैं मेरे को ना ये तो साइड पे लग रहा है हाँ जी बोलो वो पांच जगह के स्पीकर वो बोला ना पांच मैं फाड़नी पैनी थल्ले आके करना वाला की रेस्टोरेंट में नहीं कर नहीं हो जा शिरेश हो जा डॉक्टर शिरेश हो जा ना पूछो अगर लग कर रहा है तो बोलो ਇਹਦੇ ਵਿੱਚ ਲਾ ਦਿਆ ਕਰੋ ਠੀਕ ਆ ਮੈਂ ਤੁਹਾਡਾ ਲਾ ਕਰੂੰਗਾ ਇਹਦੇ ਵਿੱਚ ਲਾ ਦਿਆ ਕਰੋ ਇਹ ਤਾਂ ਹੋ ਗਈ ਸਵੇਰ ਦੇ ਟਾਈਮ ਠੀਕ ਹੈ किस तरह क्या ले चल रहा है करके देख ले चेक कर आगे बैठ करके किया था
very warm welcome to everyone. We start with 16 symposia integrating informatics, information, innovation, and community for a healthier future. This session will be chaired by Dr. Anil Gaikwad, who is a professor from BITS Pilani, and Dr. Ranju Bansal, who is a professor at University Institute of Pharmaceutical Sciences. Request the respected chairs to kindly come on the dais. Request our chairpersons to kindly introduce the speakers.
So, a very good morning to all of you. So, in this session and symposium 16, we have four speakers, and the time allocated for each speaker is 20 minutes. And I'm Professor Ranju Bansal from UIPS Punjab University. And sir is
Professor Anil Gaikwad from BITS Pilani.
so okay. So we have with us first speaker, Dr. Olga Terasova, and we welcome you, ma'am. Dr. Olga, it's my pleasure to introduce Dr. Olga, and uh, she graduated from Pirogov Russian State Medical University in 2008 and earned her PhD in mathematical biology and bioinformatics from the Institute of Biomedical Chemistry, Moscow in 2012. She currently serves as head of the laboratory for big data analysis in digital pharmacology at IPMC, established in 2024 under the National Program for Basic Scientific Research. Her laboratory focuses on big data analysis in medical virology to identify safe and effective antiviral compounds and optimize infectious disease therapies. Dr. Terasova has authored and co-authored over 70 scientific papers and several book chapters. So a big round of applause for her. And she has 48 publications indexed in Scopus. She has led major research projects funded by Russian Foundation for Basic Research and the Russian Science Foundation, focusing on drug resistance and HIV host interactions. Her team developed the HIV host web platform and a computational platform for antiviral compound discovery. In 2024, she received the first degree prize for best investigation at the Young Scientist Forum of the Russian Academy of Sciences for her innovative work on HIV host interaction analysis. So mainly she works on bioinformatics, cheminformatics, virology, antiviral agents, and big data analytics. So we welcome you, Dr. Terasova. So you may start with your presentation.
Thank you very much. First of all, I would like to thank all the organizers for the opportunity to present our study. And it is a great honor for me to be on this conference, IPSCON. It's a very honored conference, as far as I understand. So let me talk about the prediction of adverse reactions of paired drug combinations and drugs based on structure-activity relationships and network approaches. So it is well known that the drug administration is associated with the risk, sometimes with a high risk of adverse drug reactions, and some of them may lead to the serious life-threatening consequence. And it is well known that use of multiple medications, sometimes polypharmacology, may induce drug-drug interactions, which may increase the possibility of adverse drug reaction occurrence and may increase their severity. And of course, it is important to analyze all these drug-drug interactions and to predict the possibility, the probability of such occurrence of such drug-drug interactions and adverse drug reactions. So we are now working on the computational approaches for predicting activity of chemical compounds, activity of drugs, and this work is focused on the development of computational approach for predicting adverse drug reactions for drugs and drug pairs. A few words about machine learning and artificial intelligence. They have the high role in the drug discovery. More and more, they became more and more powerful for drug discovery, for computational drug discovery. And in this review in drug discovery today, the authors analyzed various drugs, mainly biotechnology, produced by biotechnology. And they found that artificial intelligence method can help to increase the probability to discover chemical compounds that became drug on the latest phase of clinical trials. This is especially important because the cost of fault of wrong wrongly found the drugs, the chemical compound that can be active but can fail on the last drug clinical trial phase is especially important. So that's why it is important to find safe and safe drugs and drugs that can have the lowest probability to have adverse drug reactions. So let me start from our approach to predict adverse reactions of single drugs based on structure-activity relationships. There are a lot of sources that describe drug reactions. Some of them are arranged in the databases of adverse drug reactions, and the main the most important that these sources should in the adverse drug reactions in these sources should be standardized using MedDRA dictionary. And we use two database, the CIDR database and on-site database, which will be used in this presentation, this study. That contains adverse drug reactions described in the box warrant and warrant precaution sections. And they are standardized with a MedDRA dictionary, and some of them, most of them are manually cured. To predict, we use this database of the source of the adverse drug reactions, and negative examples are those drugs which do not have adverse drug reactions in their list. So we use this set of positive examples, the drug with the known adverse drug reactions, and negative samples, the drug without these known adverse drug reactions. And then we use well-known machine learning procedure described, implemented in past program that is based on the Bayesian classification, and that presented the structure of chemical compound in the set of substructure descriptors. So then we can build statistics, and then we can predict the probability of the presence of a particular adverse drug reactions and the probability of its absence. So we used this program, we trained it using the set of drugs from the on-site database as positive and negative examples. And we use two thresholds with P minus PI higher than 0.5 and higher than 0.3 to select the drugs that can have particular adverse drug reactions. And there is a accuracy receiver operating characteristics under curve. that is about for the mean accuracy is over 0.8 for well-known and for rare adverse drug reactions. So there are some of them that we may predict using our computational approach based on past. In our um, computational platform that is developed for prediction antiviral chemical compounds, we also have a feature to predict adverse drug reactions. This is an example of prediction such reactions for rilpivirine. It's the reverse transcriptase inhibitor, which also used in combinations with other drugs, but there is it is characterized by the depression of CNS sometimes, and it may cause depression and sometimes even suicidal ideation. So we use it as example in our some of our applications to understand how it may be caused. Uh, Also, we have an approach to predict adverse drug reactions for drug pairs because it is important due to the indications of several drugs and, for several diseases. So to predict the adverse drug reactions that is caused by drug pairs, we used drug bank for positive examples of drug reactions for two drugs and two size database that was used as a source of negative reaction negative examples for drug reactions so that we know that these two drugs, these drug pairs, do not cause the particular adverse drug reactions that were included in the positive in the data set of positive pairs. So we formed the training set, and then we use the combinational description that give us the opportunity to describe the pair of drugs based on the combinations, all the combinations between MNA for one drug and for another drug. So we can have all these combinations of MNA and use the same Bayesian approach to predict the probability to have an adverse reactions for the drug pair. So we use these values of accuracy and sensitivity specificity. It's important to know that when we validate our approach, we excluded both drugs, both of the pair, from the training set to predict their possibility to have an adverse drug reactions, and then compare to the uh, mm, well-known reactions for them to calculate the accuracy. We also analyzed how we can use our approach to understand the most common adverse drug reactions that can be predicted and new drug reactions that can be predicted for pairs in our analysis. So this is a heat map that rows of this heat map contains the various combinations, and the columns describe the various adverse drug reactions with the PAPI values, so computationally predicted probability to have the particular adverse drug reactions. So we found that for most combinations of antiviral drugs with antineoplastic drugs, we don't have any adverse drug reactions, probably QT interval prolongation, and also rhabdomyolysis will be and sometimes hemorrhage will be in the top of the predicted adverse drug reactions for such different such various combinations. There is the heat map with particular combinations of antiviral, anti-HIV drugs and various types of drugs with the PI minus PI value in the particular cell. So we also may have an view how they can be combined and which particular adverse drug reactions they may have. Also, when we find particular drug reactions for a drug and drug pair, we would like to analyze the causes of these drug reactions, the potential targets that may be associated with this reaction. What we do in our computational approach to find such mechanisms, such molecular mechanisms, we analyze the results of prediction for a particular drug. This is an example for real pivirine. For particular molecular mechanisms predicted by PAS or that are not associated initially with adverse drug reactions. So this is a simple list of any targets that are predicted for which this compound is potentially active. Then we use two computational approach to find the drug targets, the genes that are associated with the so-called seed genes for the studied diseases that were associated from the particular adverse drug reactions. For instance, for CNS depression, we use the particular disease from this genetic database, such as depression. So we compared each adverse drug reactions with a particular pathological condition, such as disease, for instance, from this genet, to find the particular networks and genes that are associated with the known genes for these pathological conditions for this disease. And we used the network that were published previously to find these subnetworks and find important targets, important genes associated with these seed genes and potential involved in the adverse drug reactions. We used two algorithms, random walking with restart and diamond algorithm. First one can help us to find the potential targets that are associated, that are connected with the seed genes, but are not directly interacted with them. And the second one can help find the so-called functional models that are connected with the seed genes and can be involved in the particular pathological condition. So when we obtain the results of our computational procedure for real pivirine, then we ranged the targets during as a result for prediction by PAS PA minus PI, then the prediction of the random walking with restart algorithm score, and then it was ranked using these two algorithms. And also we took into account the involvement of a particular target in the model according to the diamond algorithm. So we may range these targets, and then we may use them to analyze a particular network, signal network, to make a signal network analysis. So then we used pathway enrichment analysis. So based on these targets selected for such type of analysis for this particular study, and we found several pathways that some of them include more than 10 genes associated with these adverse drug reaction with this pathological process, and then we analyzed them and found that there is phosphoinositol 3 kinase pathway. We found that some of these targets are known to be rilpivirine targets. Some of them are potential targets of rilpivirine, And some of these targets in this pathway is the known targets for regulation depressive effect, depressive conditions. Another one pathway also we found several targets that are associated with the action of rilpivirine, and some of targets are associated with depression. So using this computational approach, we can understand whether this drug can be involved in the particular adverse drug reactions, and what are the mechanisms of these kind of drug reactions. Then when we found these potential targets, we also used the network approach to find potential master regulators. This is a genes and associated proteins that can affect the transcription of particular genes, and then can be on the top of signal network regulation. So in that manner. So we used network approaches and the transcription data from gene expression omnibus and upstream analysis, computational analysis to identify such master regulators. And we found several master regulators that were also included in our network analysis and were included in the signal pathways associated with the depression. And then we produced several more computational studies to find the potential mechanisms of rhabdomyolysis, since it was predicted at the highest with the highest probability to be induced by rilpivirine with some other antineoplastic agents. It is important because sometimes antiviral compounds can be used, in particular, rilpivirine can be used as a part of.
Dr. Olga, since of the time constraint, you have completed 20 minutes. Can you please summarize?
Okay.
Thank you.
Just a minute. So we produced the same work and found some targets that can be associated with the rhabdomyolysis for this combination. The same. So to summarize, we have developed a computational approach for predicting adverse drug reactions for individual pairs, for individual drugs and drug pairs. And we found that structure activity relationships, along with machine learning and artificial intelligence, can be successfully used for making such predictions. And network analysis coupled with the machine learning procedure can help us to identify potential targets for the adverse drug reactions and the drug reactions of uh, combinations. I would like to thank all our lab for taking part in this study, especially Dr. Sergey Ivanov, who was the main, who has the main role in this study. And thank you for your attention. Mhm.
Thank you, Dr. Olga. It was a nice presentation. So anyone having any questions from the audience? So we have 2 minutes. Quickly, we can ask 2, 3 questions. Any queries from the students?
Of the time, you have used computational model and machine learning to predict drug-drug interaction and adverse drug reaction. So something are known, there are adverse drug reaction, interaction checkers and all. How that is superior to this machine learning process? Can it predict in advance based upon the genetic makeup of the host and the drug taken and the possibility of interaction with other drug? There is a genetic makeup of the host. You prescribed a drug that has different mechanisms and different mechanisms of toxicity. That is first reaction. There is a possibility of adverse drug reaction or side effect. If he takes another drug, okay, now two drugs, same host, what will be the probability of predicting definite association of this adverse drug reaction?
If
You
I
have,
just
you have presented to some extent, but I want to. hear something.
Yes, thank you for the question. If I understand correctly, you are asking whether we can predict the new adverse drug reactions for drug pairs, right? Yes, we are focusing on such a purpose. So we want to predict new adverse drug reactions for drugs and drug combinations. To predict new adverse drug reactions from the drug structure, we use the only one structure of one drug. And to predict the drug adverse drug reactions for combinations, we use structure of two drugs and the same method for generating descriptor for MNA for one drug and then MNA for the second drug. And then we made all possible combinations. For instance, MNA1 for first drug, the first one MNA for two drug. So we concatenate all the descriptions, all combinations. So the first row with the first row, the first with the second, etc. Well, we have all the combinations of MNA for both drugs. So we have like a description of the total structure made from two composite drugs, right? So then we used the same algorithm as was here predict described. So we obtained the probability, the frequency of have adverse drug reactions for drug if it include particular MNA descriptors. And also we obtain the probability to have such an adverse drug reactions in general. So it is the number of chemical compounds that have adverse drug reactions and they do not have the particular adverse drug reaction. So it's a probability in general. Then we use the statistical approach to also we understand how many chemical compounds include the particular MNA descriptor. So we have these three probability, this is three frequencies, and then we made up the statistics and we have a kind of curve that describe the probability for a new compound with the similar characteristics, with the similar frequencies to have the particular adverse drug reactions. And if we have two compounds, then we have all possible MNA combinations, the same statistical procedure, the same curve, and the same, yes. Mm. Yes. For person, yes.
Yes, sir.
Yes.
We are running short. Okay.
Communicate. Yes. Yes, it's large scale of prediction. It doesn't include the personalization at this stage. Yeah, you're right.
So I have a very small question, Dr. Olga. It was a very nice presentation, and you have predicted the adverse effects for the combinations, right? So I was just thinking, so why did you choose the combination of antivirals with antineoplastics? Is there any specific reason for that? And my second question for the students is that which software you used for the all the predictions?
Mhm. Thank you very much. Why we analyzed antiviral and antineoplastics? First of all, it's a good example because sometimes antineoplastics can inhibit the same targets. For instance, it is known that some antineoplastics that inhibit some kinases can be also active as antivirals. So there is a chance to find some antivirals from antineoplastics. And then because sometimes antineoplastics is used in combinations to prevent some viral association cancers, such as, for instance, Kaposi cancer or some kind of that. So that was two reasons. There were two reasons. And another question we used PASS that is freely available on way2drug.com. And our, how to say, daughter daughter platform viruses is included in way2drug.com. So it is also to use it for prediction versus drug reactions for antiviral compounds. But this is the whole data set, so you can use not only antivirals here, you can use any compound because it's specific.
Okay, thank you so
Thank you.
much. Dr. Olga, please stay for a minute. Please come on the dais, please. Yeah, yeah, please
We
come on.
request the session chairs to kindly felicitate Dr. Olga. Ma'am, please come this side. आप इधर आओगे? अच्छा, ma'am, come in the front. इधर ही.
शांति नहीं करेगा
So moving ahead. Yeah. So our next speaker is Dr. Rajesh Goel. He is a distinguished pharmacologist with 29 years of teaching and research experience. He holds an M.Pharm pharmacology and a PhD in the year 1996 and is a fellow of Indian Pharmacological Society and a member of the National Academy of Science. His primary research interests include the neurobiology of epilepsy, comorbidities, pharmacogenomics, computer-aided drug discovery from medicinal plants, and network pharmacology. He has published over 110 research papers in reputed journals with a highest impact factor of 15.11, supervised more than 40 postgraduate and 10 PhD scholars, and successfully completed 11 research projects, including 3 international Indo-Russian collaborations. Dr. Goel has delivered over 100 invited talks in India and abroad, and he has held key leadership positions including Dean Faculty of Medicine, Dean of Faculty Pharmaceutical Sciences, Punjab University, and Director of IQAC, Coordinator of RUSA. He has also served as a president for APTI, Executive Member of Indian Pharmacological Society, and is an empaneled expert with NAC, AICT, PCI, PTU, etc. So, Dr. Goel, you can start. So one request is we have, due to time constraint, we have 10 to 12 minutes for you to present. Then we'll have 1 or 2 questions. Yeah. So Dr. Goel, please. Thank you.
Thank you. I can understand the time constraint is everywhere now. So uh, uh, we'll be talking a little bit less than the slides because time is already told to be utilized for 10 minutes for me. We are having another very big issue that when we are working in pharmacology, we have to depend on animal models to develop new drugs. And animal models, as we say, they are primary, they are the primary experimental tools with the pharmacologist to understand the efficacy and safety of the drugs. And when we are using these tools, we have to be sure that the tools are very, very appropriate. They are giving very reproducible results, and their results can be reliable in such a way that whenever we are testing certain drugs in the animals to show their, to you can say, evaluate their efficacy and toxicity, then we can understand that they can be useful for human beings also. Whatever the facts we are getting in the animals, they should also be in the human beings, because there is a lot of problems when we are going for new drug discovery. And nowadays the people believe in fast fail, you can say, theory, where we say that earlier we understand that the molecule is failing, more severe it is for the pharmaceutical industry to save time and money. So if we are supposed to, you can say, remove this particular factor, fast fail theory, then we have to understand that preclinical research should play a very important role in finding translatable, you can say, findings from the preclinical research. So when we talk about models, animal models, we say that animal models should have certain criteria to be fulfilled for use of evaluating new molecules for certain diseases. So these molecules can be, you can say, useful. And in 1948, Wilner gave a classification whereby he has classified, or he has, you can say, evaluated the model in a way that whether they are good for predictive, you can say, validity, or whether they are having good face validity, or they are having good construct validity. So if any of these is there, so based on that, the model will be having the best one is with the predictive validity. So this is the definition. So if anybody wants to deliberate more, we can talk later. So I will just present a case study where we developed a model for drug-resistant epilepsy. The epilepsy is a chronic, you can say, neurological disorder which is affecting 50 million to 60 million. That's data, varying data in the literature. And the available drugs are not able to treat more than 30% of the patients. They are not able to manage the seizures in 30% of patients. So those patients come under the category of drug resistant epilepsy patients. So these patients need a new treatment, novel treatment, which can manage them. So based on, you can say, this unmet need, we have to understand that whatever drugs we are making by using existing pharmacological animal models, they are not yielding new molecules because we are not knowing what are the different new mechanisms which may be leading to drug resistance. So if we have to understand what are these new mechanisms which are leading to drug resistance. If we can understand that, then we can definitely model those mechanisms in the animals to have new animal models which can be useful for discovery of new molecules for drug resistant epilepsy. So there are different theories which are revolving around how there will be drug-resistant epilepsy. There's pharmacokinetic, you can say, hypothesis. There is gene variant hypothesis, target hypothesis. So I'll not deliberate much in this. There are number 5, 6 hypotheses which are available already there, which are talking about that how the drug-resistant epilepsy can be there. So these are the most commonly used animal models which are available in the literature, which are useful for modeling drug-resistant epilepsy. And we are using two experimental models in the lab. One is lamotrigine pentylene tetrazole kindling, PTZ kindling. Pentylene tetrazole kindling is a very common model to induce chronic model of epilepsy. But when we supplement lamotrigine in low dose, in low, you can say, subtherapeutic dose, along with PTZ, the animal, when it is kindled, it shows resistance to many drugs, not all drugs, but many drugs. So at the same time, we have 6 hertz low frequency, low electric frequency seizure model, which is going to give you another kindling, you can say, kindled animal, which will be resistant to many drugs. So this is one of the model which we were talking about. So we have different, you can say, we have compared this model with the PTZ model, what is the resistance, and we found that this model is not, this is how the visual convulsive seizures look when we are giving the kindling procedure. This was, say, resistant to many drugs. If you see the graph, it was resistant to many drugs. We are not able to see the red one. I think, I don't know. There's a color issue or so. So there's another bar near to this, that is red bar, which is having this, what we say, SEM levels. So most of the drugs were showing resistance, but except few, 5 drugs which were not showing. And again, moreover, the combination of drugs was also not showing the resistance in this model. So we said that this model may not be true model of drug resistant epilepsy. We have to work on that. So for this, we use bioinformatic based approach to find out what can be the best construct validity to have new animal model. Okay, so this is these are some steps. If you see this particular figure, you'll find that we have used two approaches. We have tried to see what are the available drugs and which targets they are going to targets they are going to you can say occupy to show their effect. One thing is that, and still they are not showing the anticonvulsant effect or anti-epileptic effect. That means they are not touching some of the targets which may be involved in the drug resistance. The second part was showing us the total landscape of biological, you can say, information, which says that what can be the different targets which can be useful for treating epilepsy. Okay, so when we combine this, okay, I'll just skip this because this is very long presentation, so I'll skip this. We got this information from drug bank and we found that These are the different databases. 42 drugs were available by that time when we did this study. And then we tried to find out their targets. And we found the different targets. So each drug is having different target. You can see. So database was about 42 licensed drugs were available. And then we have 175 targets of these licensed drugs. But we got the information from genetic information or dysgenet. We found that there were about 601 gene associated with the epilepsy. And once we see okay, this procedure I'm skipping, you can come to me, or you can see my lab if you want to have more details of this procedure, how to informatic tools are being used to deduce this information. So we got a lot of information from this, that there are number of genes which are available in this, and then we found what are the common number of genes out of this. Okay, so the genes will be ultimately making proteins. So how these proteins are interacting with each other, we have to find out that. Then PPIN networks are made, and they are visualized in the cytoscape. And these PPI networks, they are further analyzed to find out the clusters and which are different pathways which are involved in that. And this is another way of doing it. Just now, I think Dr. Olga also mentioned about this algorithm, diamond algorithm, which we use for investigating the possible new interactions with the existing information of genes, which can be useful for treating or managing the disease. So here we found you have to write a code, and then that code will be, you can say, running this diamond algorithm. And here is the list of, again, 84 genes in addition to 601 genes. And out of that, 527 were the genes which are common. And those common genes were further subjected to diamond analysis. And we found that there are 84 genes that are present in drug resistant epilepsy related module. And when these genes were further analyzed, we found the higher the rank of this diamond algorithm, more important that gene is for modeling their disease. So we took the first one. The first one was related to mitochondrial complex 1 function. So we identify that mitochondrial complex 1 function is compromised, and then there's drug resistance. And then that was modeled in animals using rotenone. Rotenone is a specific mitochondrial complex 1 inhibitor. Now, instead of lamotrigine, now we use rotenone in addition to PTZ kindling first, and then also with the 6-Hz kindling. You see, the corneal kindling was again made with the same way. So this is a healthy mice day 1, and this is how we'll be leading out with the kindled mice. This is a simple procedure which we follow to kindle the animals. And then these kindled animals were subjected to monotherapy as well as combination therapy. We combined some drugs because, as a legal definition of drug resistant epilepsy, any patient who is not responding to monotherapy and combination therapy of at least 2 drugs is called as drug resistant to the epilepsy, anti-epileptic drugs. So that's why we have used both monotherapy as well as the combination therapy on these kindled animals. And then we have also evaluated mitochondrial complex 1 activity and correlated it with the degree of resistance which we have seen in these animals. So I'm just skipping it quickly. So this is how the development of kindling was there.
Hello,
You can see kindling
Dr.
was.
Rajesh.
Hanji.
Although it's very interesting and very elaborative, but you please.
Yeah, I'm just concluding
Thank
in
you.
2 minutes. So,
Thank you.
okay. So 35 days are taken by the normal kindling procedure. But in the case of 6-Hz corneal kindling with rotenone, we take only 15 days. So this is effectively more. And if you find the results of anti-epileptic drug effectiveness, most of the drugs, they were not showing their effect in this model, as well as their combinations were also not active. So this was the best model till date when we developed, which was showing the drug resistance through pharmacological manipulation. So this was mitochondrial complex 1 activity, which was well correlated with the drug resistance, which was shown. So in conclusion, if we see, the informatic tools really helped us to identify the best construct, you can say, approach to model the drug resistant epilepsy. And as a result, we have got 6 Hertz rotational colony kidney model. And this model has been published, and we have made many more studies to validate this model. I think till date we have made about 8 to 10 studies to validate different mechanisms which may be effective. Latest one was this, where we have talked about HMGB1 inhibition is also possible to take care of drug-resistant epilepsy. So with this, I'll stop, and thank you very much for your kind attention.
So thank you, Dr. Rajesh. And it was a wonderful presentation.
This is the team. I think Olga is there. So she was not in the photograph, but she was also in the same lab where we collaborated through DST RSF project. So Dr. Vladimir Poryakov was the leader. So uh, we had a good time and we had good research.
So you can have good time here also.
Yeah.
So thank you so much. It was a wonderful lecture. Quickly, if you have any queries, one question or something from the audience. But I was wondering that for every disease now we are having resistance
Resistance, yeah.
with all the drug molecules.
Yeah,
So we need to
definitely.
have a new line of drug molecules.
Resistance has to be there. It is a ever evolving process. You can't stop it.
Yes.
Maybe antibiotics, maybe anti-cancer drugs, maybe any drug. Even depression nowadays, it is resistant. Treatment resistant depression is available. So okay, I think.
Thank you so much.
There are no
Thank you.
questions.
Yeah.
Thank you.
So Dr. Shirsh Ojha, and he's from Department of Pharmacology and Therapeutics, Director, University Animal Research Facility, and he has come all the way from UAE. So we welcome you here. And Dr. Ojha is a pharmacologist and translational researcher with extensive experience spanning preclinical and clinical pharmacology in both academia and industry. So he earned his medical degree and research training from AIMS, New Delhi, followed by a postdoctoral fellowship at University of Arizona's Center for Translational Research, USA. He has also served in the Clinical Pharmacology and Drug Development Division of Ranbaxy Research Laboratories, and currently he coordinates Masters of Medical Sciences in Pharmacology and Therapeutics program. He is a fellow of Royal Society of Biology and a member of several international scientific societies, including the Society of Neuroscience USA and many others. So he has published over 300 peer-reviewed articles and reviews with more than 14,000 citations and H index of 66. So huge round of applause for Dr. Oja. And we are so privileged to have you here. He also ranked among the top 2% of scientists globally, and his research has been recognized with the Distinguished Research Award from the Chancellor of UAE University and multiple institutional awards with high impact publications. So we welcome you here, Dr. Oja. Please start with your presentation.
Thank you, Dr. Professor Ranju Bansal, and thank you, Professor Gaikwad, for chairing the session. I thank Professor Anil Kumar for providing me opportunity to present here my work, and I can see a good number of students around. I want to ask you something before I begin. Do you know the history of pharmacology? Students, do you know any drug which completed 100 years? I think Professor Bansal will witness this. The history of pharmacology is very new, but once we say herbal drugs, they are since ancient civilization, right? Ancient time. And once you say synthetic drugs, do you know the first synthetic drug? Penicillin was discovered in 1940, launched into market in 1944. Before that, sulfonamides were used as antimicrobial, right? 1935. Before that, we have no synthetic medicines. Morphine was the first compound isolated in 1804, right? Marketed by Merck in 1827. So in 2027, we will celebrate 200 years of morphine. And except morphine, so if you look into the history, we have no pure compounds into the field of pharmacology, right? So what makes you happy? You know some chemicals in your body which makes you happy? Morph Endorphins. Have you heard of dopamine, serotonin, and endorphins, right? Apart from endorphins, which makes you happy is cannabinoid, right? But don't get confused with psychotropic cannabinoids, which are fashion these days. No marijuana, no hemp. I'm not working on those, right? And I don't propagate those. So, talking about cannabinoids in liver disease, I have generated interest to work on non psychoactive, non psychotropic cannabinoids in health and disease. And this is working. So, I have to go like this, sir. So if you look into endocannabinoid system, once you do some exercise, once you walk, you feel happy, right? This is because of endorphins and cannabinoids. So we have endocannabinoid system which evolved into our body since 500 million years ago. Vertebrates, invertebrates, and human beings. We all are blessed with endocannabinoid system. And if you look into, there are two kind of receptors, cannabinoid 1 and cannabinoid 2, and we have two kind of metabolic enzymes which constitute ECS, endocannabinoid system. And we know cannabinoids since several million years ago, but CB1 receptors were cloned in 1990 and they are dubbed as central receptors. So they are mainly working on CNS level, while CB2 receptors, they were cloned in 1993 and they are dubbed as peripheral. So they work on all across the body, okay, except CNS. Few reports coming, they are working in CNS too. So CB2 receptors are mainly considered as immunomodulator. Why immunomodulator? Because they check the immune inflammatory axis and oxidative stress. So looking into these, we are focusing on cannabinoid compounds. If you look into, again, India is largely recognized for cannabinoid worldwide because of one connection I'll bring in that. So endocannabinoid system consists of CB1, CB2, and enzymes. And these cannabinoids often talk with other receptors, TRPV1 or PPAR gamma, which are used as insulin sensitizers. So coming on to the first endocannabinoid recognized in our body was anandamide. In Western English, we say anandamide, but if you talk like Hindi or Sanskrit, you say anandamide. Ananda is Anand, to be blessed. A group of Israeli scientists search all the way in Himalayan caves and find out none of the sannyasis, they are dying of cardiovascular and cancer diseases, right? And they thought how this is happening. This is all happening because of cannabis. Due that cannabis is psychotropic in nature because they are away from home, they want to be blessed. So the compound name derived is anandamide, to be pleased, to be happy, and to be blessed. So there are three sources of cannabinoids. We have endocannabinoids, phytocannabinoids, and synthetic cannabinoids. In past few years, enormous number of synthetic cannabinoids have been developed, but those are still adulterated and those still have psychotropic nature. Endocannabinoids, they are very short-lived. They cannot be made as a drug. So there's not much focus. The only way to develop endocannabinoids is to do exercise. So we are focusing on phytocannabinoids, and based on those, we have cannabis receptors in our body, right? So we are focusing mainly on CB2, the reason being the first most successful drug. Professor Gaikwad will witness it. The first successful drug for obesity was rimonabant, right? It was very good in terms of efficacy, but in terms of safety, it has been withdrawn because of hallucination and suicidal tendencies. So the whole focus on drug industries on cannabis 2 type CB2 receptors. So activation of CB2 receptors are considered good for human health. So we are focusing on cannabis type 2 receptors because CB2 receptors, they are devoid of abuse, tolerance, resistance, and dependence. So looking into CB2 receptor agonist, we were again exploring phytocannabinoids, and our lab focuses on phytocannabinoids other than cannabis. Okay. So we say we don't promote cannabis, but we can promote cannabinoid kind compounds which are lacking psychotropic effects. So we focus on one compound. It's known as beta caryophyllene. There are many other compounds isolated from ginger, echinacea, and they are known to consist of some phytocompounds which act like cannabis. If you talk about, I'm working outside, so I have this pleasure to connect everybody. Once you go Germany, the only herbal medicine, licorice. More than glycyrrhiza, what we say, glycyrrhiza glabra, everybody knows licorice. You might have heard of licorice candies, right? The old licorice candies has no licorice. If you go to US, cranberry juice, used as urinary antiseptic. Once you say any Westerner, I'm from India, they say curry, right? It's connected to curcumin, turmeric. So same like that, we are all connected to cannabis too somehow. In the past 5 6 years, we have a good number of cannabis-based drugs into the clinic. Professor Goel mentioned about epilepsy research. The most successful drug these days for refractory epilepsy, once resistant to all anti-epileptic drugs, whether lamotrigine, vigabatrin, or gabapentin, we have cannabidiol. And cannabidiol has been approved for pediatric patients. Okay. So that altogether, we have drugs for chemotherapy-induced nausea and vomiting. We have drugs for, say, multiple sclerosis, Sativex. So all based on cannabis. So in the past few years, we focused on cannabinoid type 2 receptors, and these are considered of activating MAP protein kinases, and they have no psychotropic effect. They are known to play a role in diabetic complications because of their pleiotropic nature, multiple pharmacological activities. So I'm not going in detail of these things, but altogether I can say oxidative stress, high glucose condition, hyperglycemia causes oxidative stress, inflammation, apoptosis, and autophagy. Whenever you talk about any pathogenic process of disease, these are the four events: oxidative stress, inflammation, apoptosis, and autophagy. Everything connected to these four. This is why we say Trinity of God: infection, inflammation, and immunity, right? Any disease associated to
Hello,
these three.
Dr. Oza.
So
Although your lecture is giving lots of anand to us,
I'm just
but
limiting it.
we,
So we worked on PCP, and we were looking into cannabinoid type 2 receptor activation. In liver function, we developed two models. One is we worked on BCP, and we worked on thiacetamide induced liver fibrosis as well as high fat diet induced liver fibrosis. And looking into high fat diet, we use high fat diet and streptozotocin to induce diabetic fatty liver. We say non-alcoholic fatty liver. And we look into majority of the parameters related to oxidative stress, inflammation, and apoptosis. We found BCP treatment in mesenteric staining reduced the fibrosis. In serious red staining, it reduced fibrosis. We did lot of Western blots to look into the fibrotic proteins, whether fibronectin, vimentin, cadherin, and alpha SMA. And we found BCP treatment reduced hepatic fibrosis by modulating these all proteins. So it also impacted inflammasome. Inflammasome is one of the trigger for the countering inflammation. So it activated by caspase-1 and interleukin 18 and NLRP3 genes. So we also looked into pro-inflammatory and anti-inflammatory cytokines and found BCP treatment was able to reduce inflammation. It also exerted antioxidant improvement by NRF2, KEEP, and HO1 pathway. We have very limited time, right? So it improved antioxidant enzymes, antioxidant status. It corrected the liver function parameters, whether ALT, AST, or ALP. And we also evaluated BCP in thiacetamide-induced liver fibrosis, where we use thiacetamide for 8 weeks. And we looked into the gross anatomy, we can say liver index, and we found that BCP treatment corrected the liver enzymes. It ameliorated oxidative stress, improved SOD, catalase and GPX, reduced MDA. It also attenuated inflammation by inhibiting inflammatory cytokines. And we did HNE, serious rate, and other staining protocols like MT, and we found reduced fibrotic pattern. We also looked into immunohistochemistry protocols for different fibrosis proteins, fibronectin, cadherin, and vimentin, and find a significant reduction in these proteins. We also looked into different kind of these are the densitometric analysis data, and we also looked into cell death pattern, longevity, and many apoptotic proteins. And we also found that these are able to fix matrix metalloproteinases, which are vital for collagen synthesis, which provides tissue structure, strength, and elasticity. We also looked into different collagenases. We've also found it was able to normalize the body weight, improve, reduce the blood glucose levels. It has an impact on hemodynamic parameters and improved the lipid profile. It also corrected toll-like receptors activation and P38 junk pathway. It improves the glucose tolerance and insulin resistance. It also restored the age range function. And we were looking into whether it can impact pancreatic insulin. It has impact on pancreatic insulin by immunofluorescence. And we, in the past few years, we published, I'll quickly go on. We published a good number of papers in standard journals, whether JPET or ACS Pharmacological Sciences. And I thank my collaborators and research team. I know plenty of bachelor students are here, but if somebody who is masters in pharmacy with a research publication, they are invited to approach me. We provide a very good fellowship, 5,000 dirham per month, which equates to 1.3 lakhs. Yeah. And a beautiful life in Dubai and Abu Dhabi. I'm when I moved from US, I thought I will there for 2 to 3 years, make some money and go back. But I'm there since 2000. So it's my 15th year in Dubai. And if you are a master's in pharmacy with a research publication, feel free to contact me. I say huge thanks to my collaborators, Professor Samir Goyal, Dr. Kapil Suchal. I have few collaborators from Singapore and US, and I can see Professor Butter here. I have been witness to his research many times. Thank you all.
Yeah, thank you, Dr. Oja. And I will
Sorry if I took more time, I cut down like
No,
anything.
absolutely fine. And I'll definitely convey your message
I
about
always
that
feel,
research fellowship to my students, and they would love to go to your place to do research on cannabinoids.
Thank you.
And thank you so much for presenting this extensive research on cannabinoids here. And it was really a Anandmai presentation. Thank you so much.
Don't get confused with Amrita Anandamai Ma.
Anyone having any questions? Quickly?
Any questions?
Yeah.
Yeah.
Thank you very much. You mentioned that we have four pathways that are essential for every disease.
Right.
Find these four pathways, and we think that there's because there are a lot of hubs around these pathways.
Right.
What about nature? If we don't think about bioinformatics in nature, are these four pathways essential for any diseases?
These are not four pathways. These are four pathogenic events. We need
Yes,
to
yes,
differentiate these
yes,
two.
yes, yes.
Oxidative stress, inflammation, apoptosis, and autophagy. These are four pathogenic events which takes place in the human body, and then the diseases appears. Whatever disease you talk about. There are three major culprits. You say infection, communicable diseases, inflammation, non-communicable diseases, right? It can happen in communicable diseases too, and immune dysregulation. That is why we say whatever disease we talk about, these are centered on these. So once you look into oxidative stress, there are enormous number of pathways to regulate oxidative stress at cell level, at cellular and molecular level. Enormous number of pathways to regulate inflammatory events, starting from vagus nerve stimulation to pro-inflammatory cytokines to complement activation. Once you come on to immune regulation, enormous number of pathways. The approach these days, what network pharmacology is using, there is a new word you might have heard about, immune inflammatory axis.
Mm-hmm.
So we often say, leave oxidative stress at one place and talk about, because ultimately oxidative stress is the reason for the generation of inflammatory cytokines
Of course.
and immune dysregulation. So immune dysregulation, immune dysfunction, immune inflammatory dysfunction is the main axis. You can, as you mentioned, what about nature? You can do network pharmacology for all these protein targets. But ultimately, then you have to go for the in silico, more of in vitro and in vivo conformations,
Mm-hmm.
right? So this is, I used to say sometime, these days we are refining our target so much. Once you refine the target so much, you will not get anything.
Yes, I agree with you.
You
That's
know,
why I'm asking.
when paracetamol was discovered, until now, what is written in the textbook? It is believed to inhibit prostaglandin synthesis. We don't know the exact mechanism. Ask somebody how propranolol work in migraine. It is believed to have an impact on cortical spreading depression. Once you refine the target so much, you will get nothing. This is the absolute fact. We refined sugar, we're getting obesity.
Mm-hmm. Thank you.
Absolutely true.
So be crude, go to nature, and explore them with a wide precision.
Thank you so
Thank
much.
you.
Thank you.
Okay, so we'll move to the last speaker of this session, uh, Dr. Debashish Hota. Dr. Debashish Hota, professor of pharmacology at AIMS Bhubaneswar. He has made notable contributions to both experimental and clinical pharmacology, reflected in his 76 peer-reviewed publication. His research span pain management, metabolic disorders, and drug-drug interactions. He has led important clinical studies, including a randomized double-blind trial comparing lamotrigine and amitriptyline for painful diabetic neuropathy and investigations on how piperine alters the pharmacokinetics of phenytoin. Dr. Hota has also explored the metabolic benefits of dietary oils, demonstrating the anti-diabetic and anti-hyperlipidemic effects of blended rice bran oil with sesame oil compared to soybean oil in the pre-diabetic and diabetic individuals. His additional work includes assessing urinary iodine status among autoimmune and non-autoimmune thyroid patients in coastal Odisha, contributing valuable regional endocrine health insights. So with this, we welcome Dr. Hota, and I'll request to please stick to the time of 10 minutes. Thank you.
Thank you. I'm basically a trained medical doctor having experience in clinical pharmacology. I'm basically a medical teacher. So apart from teaching, I also am involved in research work, but we primarily focus on research that is directly beneficial to the patient or to the human beings. Bench research is just a secondary aspect of our research. So now we are focusing on those services or researches which will help in improving the patient care in the hospital. So let us start with therapeutic drug monitoring. That actually it started with drugs like digitalis, phenytoin, lithium, where we know that these are the drugs having narrow therapeutic index. That means their therapeutic range is very, very narrow, below which there will be subtherapeutic, which will produce lack of efficacy, and above which it will produce more risk of toxicity. So they are narrow therapeutic index drugs. Then in the second phase, in 1980s, then standardization. Then we tried to correlate the pharmacokinetics and the dynamics. So PK PD correlation that started later, and later 2010 onwards, we tried to refine our skills further. So, pharmacogenomic, model based precision, and real time continuous monitoring. So, that will be more precise in controlling the symptoms. As we have been discussing about machine learning, artificial intelligence, drug resistance, resistant epilepsy. So, we will present some of our experiences in drug resistant epilepsy. So why these things happen despite maintaining a standard plasma concentration? We talk of certain things that this is the safe level, maybe for phenytoin 10 to 20 microgram per ml is the standard. That is what the textbook mentions. Many patients are well controlled below 10, and some patients even do not get adequate control above 20 microgram per ml. So that varies. And there is another interesting finding. People with stress, it can be traumatic stress or having serious this multi-organ dysfunction, the clearance of phenytoin becomes very fast. Despite getting intravenous phenytoin regularly, we that maintains a normal plasma concentration. In a patient who is having polytrauma or who is admitted in the ICU with multiple organ failure, the clearance becomes very fast. We still do not know why it happens in a patient under stress. So that is how Olga has actually given me some insight into it. We are trying to work on it on our lab. So pharmacogenomics. So that is the thing, we are coming to that. Then model information and precision dosing. So one size does not fit all. Maybe I am well controlled with a particular dose or dosing schedule that may not work in the index case. So that varies. So individualized therapy is the word I can say, that one size does not fit all. Then advanced biosensors, that is the upcoming area in therapeutic drug monitoring and estimation of drug level and its comparison with the dynamic response without increasing in the toxicity. So our purpose is to have a safe medication or medication dosing schedule which will have maximum efficacy and minimization of the adverse events. So we have some experience here that we published last year. So this is carbamazepine toxicity with concomitant levetiracetam. There is no report saying that there is an interaction. It is probably the first in the world which shows that levetiracetam can increase carbamazepine toxicity. There was no report. So here what we have found, levetiracetam is considered to be one of the safest of the anti-epileptic drugs. Having daily dose up to 6 gram is considered safe. At least in Indian patients, I can say neurologists are prescribing. But when a patient received carbamazepine not controlled, added levetiracetam, there was a problem. So this study actually focused on certain things that when you are prescribing a drug, because every patient is different, you have to be very, very vigilant. It may work, it may produce more toxicity, it may not work, may not even show any sign of toxicity. So levetiracetam is known to have no pharmacokinetic interaction. So this therapeutic drug monitoring clinic in our hospital so suggested a different reality prompting into more detailed investigation. So we studied the records of 158 patients receiving carbamazepine, though concentrations were comparable and between the two groups, but disproportionality was there, and carbamazepine levetiracetam interaction, the odds of having toxicity is 16.65 times, more than 16 times, where odds of this receiving toxicity or interaction, that is 2.25 times when combined with when not combined with levetiracetam. So mean carbamazepine concentration increased from 9.72 microgram per ml 11.1 when combined with levetiracetam. Does it mean that levetiracetam decreases the metabolism? We still don't know. More research has to be done, or decreases its clearance. So we still don't know. More studies are to be done. And this you can find that levetiracetam, when all other p-values are insignificant, when combined with levetiracetam, there is a significant interaction. So this is the first we reported, and it has got good citations also. And carbamazepine here, the critical threshold ratio is 1.86. That means when the ratio between carbamazepine and levetiracetam goes above 1.86, there is a risk of toxicity. This is the ROC curve. We show that 1.86 is the critical threshold. So do not exceed the dose ratio above 1.86. So levetiracetam should not be more than 2 times at least. We cannot have a precision dosing here. So never go beyond twice the dose of carbamazepine when you are using levetiracetam. So what happens? Therapeutic advancement is going on every year. New drugs are pouring into the market, new interactions, new indications, and mostly our aged population is a drug population. As the age increases, more and more disease burden comes and more drugs are prescribed. We forget about the possibly the beneficial effects of sleep, lack of stress, exercise, dietary habits, that we always look for an easy solution through medicine. Rather, we should go back to nature and rely on our instincts, how to minimize the drug usage. If we have to use a drug, we have to see that it is less toxic and more beneficial to the subject. So, real world TDM data revealed what controlled clinical trials missed so far. So, this is something we found from our records, that levetiracetam carbamazepine can be an unsafe combination. Then this is something very, very interesting. Clozapine forgiveness. This was published only few months back. But here, clozapine is also another narrow therapeutic index drug. It is typically prescribed in resistant schizophrenia, and our psychiatry department regularly uses. As we know that whenever multiple drugs are used, the problem of resistance gradually comes. So it is a reserved drug, this clozapine, and it is given. It's a very effective drug, but has a narrow therapeutic index. So what happens here? We have modeled that clozapine has a narrow therapeutic index, 0.35 to zero point 0.6. That is the acceptable therapeutic range. But initially, to reach that range, we have to give little higher dose to achieve that therapeutic concentration. Despite achieving the therapeutic concentration, the patient gets pharmacological response or the benefit of that drug maybe after few days to few weeks. We still do not know what are the dynamic changes happening in the brain. We focus on the dopamine 2 receptor, but we still do not know what are the changes happening in serotonin receptor, 5-HT2, or
Dr. Hota?
T4 receptor, or other receptors.
Dr. Hota?
Yes, sir.
Can you please summarize?
Yeah,
Right.
I'm just coming
Thank
to
you.
it, the last study. Then, so missed doses. So here, this is the interesting part. This is one dose missed. So there is a significant reduction in that level. 2 doses missed, further reduction in the level. 3 doses, further reduction. 4 doses. These are all models, computational models from the population data. And this is the 5 dose, the 6 dose missing. That means when you are miss a dose, or you see what are the changes happening. We stopped after this 8 doses. There is no point. That means you have to restart the therapy again. Then concentration time profile delay. First thing, we missed the dose, then we see what are the changes. Now we delayed the time. If the time is delayed by 1 hour, there is no significant change. 2 hours, no change. 3 hours, also no significant change in the therapeutic level. Then gradually you found that there is a change in the level. The level goes after missing 10 hours. This shows that missing time is not important. but missing dose is more important. And this is recently published in Neurology India, ordinarily differential equation based individualized PK stimulation and dose optimization in an infant. This infant was having seizure not controlled by receiving 4 different anti-epileptic drug categories. So this was something very, very interesting. So the children with epilepsy, so that you know that global developmental delay, that is one, and about 1% of the global population, it is 0.8 to 1.1, so we can predict approximately 1% of the global population has seizure disorders, and therapeutic drug monitoring is not always the answer, but it can answer certain questions like the dose level and the therapeutic response. But to predict the response is not always simple, and you have to do modeling. So here we modeled that 400 milligram valproate twice daily versus 240 milligram 4 hourly. So here you found that when the dose is more, the fluctuation is very high. Here 400 milligram twice daily. So here 240 milligram IV 4 hours. So here it shows there is a steady concentration maintained, because again valproate is also a drug of narrow therapeutic index. So when the frequency is increased, dose is reduced, the levels are maintained in a particular therapeutic range, which is beneficial to the subject. So patient became seizure free within 24 hours of dose optimization, and 7 drug regimens were de-escalated to 4 drug regimens. So that is a problem. Pediatricians are always very, very emotional about the children and the care of this and the parental pressure on them to control the seizure and all those things. So how modern TDM is helping us? So traditional TDM requires steady state. So Bayesian approach, so a priori knowledge of the pharmacokinetic parameters like clearance, volume of distribution, and variability in the response and the levels. Posterior estimation and individualized PK parameter, that is very, very crucial. That is what I was talking to Olga, whether every patient needs to be monitored. So that is a question. We have to have a population setup. Then infrastructure, that is definitely one-time investment, and computational resources. You have to have technical manpower, technology that is necessary. Then we have to have patients who receive the target drug. This is very, very crucial. And you have to have the therapeutic, this quality assurance, because machine to machine variation, drug to drug variation, and formulation to formulation variation in the level, that is also a question, and we have to assure that. So we can get a better response and we can better individualize the therapy. And I acknowledge my colleagues. She is Dr. Archana, who does all of the analytical work. She is MD and DM in clinical pharmacology. This is me. This is my colleague, Dr. Rituparna. This is Anand Srinivasan. We run the therapeutic drug monitoring lab in AIMS Bhubaneswar. We expect more to happen in near future. So let us see what time will tell. Thank you very much for your attention.
Thank you, Dr. Ota, for presenting such an interesting findings. So we'll take one question from audience if needed.
Please. Yeah, anyone. Please.
What about the drugs that you've presented, carbamazepine and another in combination in their pharmacogenomics? Are there studies that you used in your?
Any patient who received carbamazepine with levetiracetam, carbamazepine will go.
Yes.
Definitely that point is well taken. The genomic analysis has to be done.
Okay. Thank you very much.
Thank you. Thank you, Dr. Ota, for the presentation. Yeah, thank you.
So we request you to be here. Sure. Dr. Anju Bansal. चलो बेटा दूसरा भी दे डॉक्टर अनिल गायकवाड़ There's an announcement. A cab is available for the delegates who want to visit the lunch area, just outside the OD. We will meet exactly at 2 o'clock for the post-lunch session. Thank you, everyone.
Hello, 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 hello.
हाँ निकले तुम हो गया हाँ 2 बजे स्टार्ट हो गया तो 2 बजे ना एक मैसेज कॉल है कि सीधा
Hello, hello.
सीधा साथ आ रहे हैं सर कितना थे रात को पढ़ जाते स्टॉक बोल रहे हैं सीधा साथ भी चेयर आऊँ 2 बजे बच्चे को चेयर क्लास में तो दे सिस्टम में फिर स्टार्ट होएगा कुछ नहीं लेना टाइम का दिन है ठीक है पहले वेट शुरू करना पड़ेगा क्यों वहाँ पर लंच दिया था प्लेस तो 2 बजे स्टार्ट करते हैं हम लोग
1 से 2 का लंच का टाइम था
बाकी लेट हो रहा है कभी तो करेंगे कैंटीन का तरफ
सिन्हा सर कह रही है दूसरा आपको ये टेस्ट करनी होगा भी टाइम चेंज करनी है चलिए इसमें 1 से 2 क्यों है
गुड है
उसका लेट चल रहा, चलने थे हम क्या करें?
लंच ब्लॉक वाला कोई लेट है ना, नीचे शुरुआत होती है।
ये क्या
वो अपने अकॉर्डिंग ही चला, अपने अकॉर्डिंग चला लेना।
कौन है वहाँ बंद है?
ज्यादा तकलीफ कर देंगे, बच्चे बैठे हुए आराम। कल भी मैंने कल पीएससी स्टार्ट किया था।
नहीं मैं तब बेटे जाके आई हूँ।
नहीं हम भी मिलते हैं 9 बजे से करना शुरू किया। हमें प्लस करना तीनों टीचर्स वाला शेयर कर देते हैं। डेलीगेट्स का क्या अलग अलग प्रॉब्लम है, टेर और उनका दूसरे वाला हमें लेट ही मिलते हैं। ओके मैं साइड
हेलो हेलो ठीक है भाई। हाँ हाँ हेलो हेलो
कोडिंग चेक कर। हां वन
हेलो हेलो
हेलो हेलो बात कर माइक कर रहे हैं पहले तो तेरा कोडिंग का ये काम ये करना कितनी करनी है। हेलो हेलो वो चक्कर वो तो बंद हो जाता है, ये बंद नहीं होता है। उन पीपीटी भी करना ये पेस्ट करनी।
Well,
ये तो माइक पढ़ के बोलन भी Hello, hello, hello.
Hello. 1, 2, 3.
afternoon, everyone present here to the session. I have the honor of inviting our distinguished session chair, Professor V.R. Sinha and Dr. Yogesh Kulkarni, to graciously ascend the dais. And, sir, please. Welcome you, sir. Before starting the session, I would like to introduce our session chair, Professor V.R. Sinha. He is a distinguished pharmaceutical and senior professor at the UIPS Chandigarh. He is widely recognized for his pioneering work in novel drug delivery system, pharmaceutical technology, and formulation development. Professor Sinha has an outstanding research record with more than 70 peer-reviewed publications, over 6,900 citation, and impressive H-index of 34, reflecting his significant global research impact. He has mentored numerous PhD scholars who serve in academia and industry. His innovation include technology transferred formulation developed at UIPS, reflecting his strong linkage between research and translation. He has held major academic leadership positions, including chairperson UIPS, and has contributed to Punjab University academic ecosystem through curriculum development, research collaboration, and institutional quality enhancement. And second chair for Dr. Yogesh Kulkarni. He is a distinguished academic and researcher, serving as professor and associate dean at the School of Pharmacy and Technology Management, NIMS, University, Mumbai. He has over 2 decades of experience in pharmaceutical education, research, and mentorship. His expertise span pharmacognosy, pharmacology, natural products, and herbal drug research, with a strong focus on translational and applied research in drug discovery and development. He has been actively involved in numerous funded research projects from national and international agencies, and has been widely cited and presented at more than 100 national and international conferences. Beyond research, he has played a pivotal role in curriculum development, academic administration, and mentoring postgraduate and doctoral students, shaping the next generation of pharmaceutical professionals. We welcome both of you, sir. Thank you. I request now to start with the session.
Good afternoon, everyone. So, I would like to invite Dr. Vipan Parihar for this invited talk. Dr. Vipan Parihar is an associate professor and acting head of Department of Pharmacology and Toxicology, as well as regulatory toxicology at Naipar Hajipur. He earned his M.Pharm Pharmacology and PhD from Manipal University, where his doctoral research focused on irradiation-induced genomic instability. His research primarily centers on CNS disorders, investigating sex differences in brain function, cognitive deficit from diabetes or irradiation, and regenerative pharmacological interventions via stem cell strategies. He is also engaged in antimicrobial resistance research, working on novel diagnostic and therapies for antibiotic-resistant pathogens. Dr. Parihar has published widely on topics such as radiation-induced cognitive dysfunction, neurogenesis, and nanoparticle-based cancer therapies. His work on cabozantinib-loaded nanoparticles for hepatocellular carcinoma. A notable publication includes PNAS paper, Cranial Eradication Compromises Neuronal Architecture in the Hippocampus. His research is heavily supported by major grants such as SCRB funded projects on endocannabinoids in neurodegeneration. So I invite Dr. Vipan Parihar for his talk.
रिस्पॉन्स है ये अगर लेज़र चाहिए ना खुद से पॉइंट करना वहाँ पर ये है ना ठीक है So good afternoon, everyone. So first of all, I thanks to this committee to inviting me for this guest speaker, as well as the panel member, Dr. Kulkarni and Senior Professor in our department. So for these nice introductions. So my topic was broadly focused on the sex-specific difference in radiation-induced cognitive impairment, as well as mood and memory dysfunctions. So as you can see, the India is a hub for diabetes. And the diabetes is not only a disease, a multiple disease, and also the cause for multiple other diseases, which consequently lead to death of many people. Especially the brain disorders, where now the brain stroke has become very popular in India. People who have diabetes, who have nearly double the time of chances getting brain stroke and brain paralysis. Second is diabetic nephropathy, and also vascular disease. Highly all vascular organs clearly responsible for this. So I'm not going to more detail in this here. The current study will focus why the males are more sensitive to the hyperglycemia and cognitive impairment and other disease. So you know that in broadly speaking, hyperglycemia or chronic diabetes causes a structural and functional changes in the brains that eventually leads to the reduced neurogenesis, which is basically formation of new neurons. In a layman language, our brain adds nearly 5,000 neurons per year or so per day. If you are hyperglycemic, the density and quality of neurons drastically reduces. So whatever happen, you have facing a crisis in learning, retaining your memory, taking judgment, cognitive flexibility, and eventually leads to a cognitive rigid brain. That's why the many diabetes people have. What is the basically cause of that and how it impacts male and female differently is no one knows this. That's why my current study will focus on that. So recently I have published a series of papers on diabetic nephropathy and diabetic neuropathy. This is one of them. So where we have determined the hyperglycemia doesn't affect male and female equally. So this study we have done in intense by integrated proteomics, transcriptomics, metabolomics. We have analyzed nearly 5,000 genes. In this 26 gene we have identified that critically controls the metabolism of glucose, bioenergesis, oxidative phosphorylations, cytosolic as well as mitochondrial glycolysis. So this is my published paper recently, we have seen it here. And then we checked whether this gene expression has a roles on the making proteins, because gene expression has no importance unless it makes its associated proteins, it makes signals. So then we did proteomics, and finally we correlating this. The same protein has been also related, and further we've identified if this gene is expressed, protein is also expressed, whether this gene or protein is functional, is really taking parts of learning and memory. Even you go to the walk, your gene expression happens, protein expression happens, whether this proteins really turns to the learning or memory, we identify with the behavior. That's why our study focus on the gene to behavior study, entirely we do. So this is so now our hypothesis is that the male are relatively more sensitive to the hyperglycemia. Why? Because a certain part of genes, which is basically are immunogenic, they are more expressed on the hyperglycemia. Immunogenic response because female are really more adjusted or more primed to the inflammation, especially in the brain, because they are 30% of brain cells, not other than glia or neuron, they are microglia, they are autonomous cell in the brain. They don't take effects from the periphery of vascular system. It's like a microglia are the autonomic cell in the female brain. If any injury in the brain, they don't get inflamed. While the male brain usually get inflamed by the surroundings. And there's a special type of signal we have checked here. So to induce this, we have produced a type 2 diabetes induced models where we have produced high fat diets, and then we confirm the high fat diet almost 3 months. Following the 3 months, we have injected streptozotocin. You all know these models, but this only aim of this model was to produce a slightly increase in the hyperglycemia, persistent, also a little bit of insulin resistance. Why insulin resistance? If you cope with the human study in pathology of diabetes, 30 to 40% of people who had
normal insulin, even sometimes higher. So hyperglycemia is not only the cause of diabetes induced cognitive impairment, it's a basically insulin resistance is the cause. So coming to the points, this is our composition of glucose, composition of the high fat diet, and we see fasting glucose level. Day one, both of the genders, both of the sex has the same glucose level. As it is moved through 34 to 68 days, most of the animals, so means male and female, go so hyperglycemia. The severity of hyperglycemia little bit changes, but it's not a big changes because a little bit more high, because female doesn't only differ only the hormonal level, it also different lot of metabolites. So this is not a proof. So but we clearly says this model is pretty valid to produce consistent hyperglycemia. After that, we have tested whether the cognitive impairment happen at same level or different level in male and females. So we induced the cognitive hyperglycemia, and mice were subject to test called novel object recognition test. This test selectively depends on prefrontal cortex. Prefrontal cortex is the most important part of the brain, would control your judgment behavior, how you judge the events before and after, and also controls your higher order learning. So if you see in test, the animal were habituated on box, and 24 hours they are exposed to identical toys. And after 5 minutes, so this is the point. So animal are habituated here, and animal were habituated for 5 minutes, say, day 1 to day 3, and then day 3 they are exposed to identical toys, toy 1 and toy 2, and after 5 minutes, one object is replaced, means animal go to the home cage, it comes back. So what we will see, if animal has intact memory, so he will spend more time exploring this novel object. This is called the recency memory. This is called the, so tendency of every humans also, they will spend more time with here. So if they discriminate these two, just like if they spend 50 times here, there's no discriminations. If he spends more time with here, it's called discrimination, means he remembers, so you can see it here. So in the all these male mice, so the clear difference was there. I think color is not coming here. So this is a male. This is basically the male mice. These are the female mice. So my slide is saying something different. He is saying something different. This is a basically male. So male are able to, they are not able to discriminate it here. So means male has been impaired. Why female are completely able to discriminate. This is a new object. This is an old object. So we can say there's a clear discriminations between two, and animal of the male mice show a impaired recognition memory. Yeah. Then we observe the second test. This second test is called as object in place task. This is called as navigation memory as well as spatial reference. Spatial reference means how we human, we basically, the brain is the most smart. He basically take a reference memory. So like we walk this corridor, we take left and right, basically another body as a navigating tools. So how to take. So in this 4 object, animal were exposed to 4 identical toys, so different toys. And after 2, the 2 object has been replaced to their locations. So if animal has intact memory, he will really identify, so these 2 objects, and these 2 objects are interchanged. You can see these 2 objects are interchanged basically. So this one interchange. So animal spends more time on interchange of objects because you take reference. So this basically memory depends upon perennial cortex. Perennial cortex is part of the brain who decides the basically event on basis also previous experience. So this is also got impaired in the animals. So third test we have done the cognitive rigidity and FST test. So here animal was subjected to the 4 swim test, and what I will see, there is a helpless test. So more the time spent on floating, more is called depressive behavior. Just like if animal is depressive, person is different. If he fall in the water, he don't want to escape himself or herself. He want to die. The same test happen on the 4 swim test. Animal who are really depressed really spends less time spending in struggling. So here the time spent on floating goes just like it here, and is almost here in this. So it's clearly saying that the male mice, because there is no major differentiated here, but tendency was there. There's little bit male, so little bit less helplessness compared to the females. So this is the end of the test. We did the water maze test. So, and the water mass test we did, but here, I'm not going to detail it here, but overall, we see the animals show a clear difference, the gender or sex say clear difference. So male mice are really struggling a lot in finding the tendency. So next, we found what is the reason behind this. So we isolated the brains and we analyzed the neurotransmitter endocannabinoids. So we have published a series of papers where we found the endocannabinoids as greatly influence in the diabetes, because endocannabinoids basically is phospholipids, higher lipid order. So hyperglycemia completely reduces. This is called anandamide in 2AG. A is a short form from anandamide, called anandmay. So if so, it's clearly control a series of functions, especially microglia activations, inflammations, and learning and memory. So anandamide was significantly reduced on the, is more extensively reduced in the male mice compared to the female mice here. So it's more reduced, but if you see it here, also the another enzymes called 2AG. This is a natural endocannabinoids enzymes. This is basically like a marijuana. Marijuana basically bind to the endocannabinoid receptors, but they are endogenous endocannabinoids. So this is our mechanism we found why diabetes signifies chronic inflammation, headache, and also these two of the interleukins, two of the lipids has been responsible for a lot of neurological disorder in the brain in diabetic people. Second, so whether these endocannabinoids are not synthesized or degraded, we had done the enzymes was responsible for their synthesis and degradations. So why is a critical enzymes, this is called fatty acid amide hydroxylase. This is basically enzymes overexpression significantly reduces the endocannabinoid concentration. This enzyme is also responsible for maintaining all the endocannabinoids. That's why now the most common drug in market is the FAI inhibitors. They are coming for the anti-mood enhancing. So it's not it's a mood enhancing drugs. So FAI is enzyme. If you stop this FAI, your mood will be elevated. Now the recent article has come with all the most of the drugs who are antidepressants. This basically has a big role on this. Second is a Nepalese. These are another enzymes who basically control the degradation of anandamide in 2AG, but there's no significant change on this. Same as the DLG, we identify 3 enzymes, but that's why we say the over degradations of anandamides may be responsible for the by the diabetes or hyperglycemia induced cognitive impairments. This is most commonly finding we have. After this, other than that, we also identified 6 or 7 enzymes, but there was no change there. Particularly, we have only focused on one enzyme now. So, because my study is highly specific and cell-specific study, so we have analyzed the neuron, glia, and astrocytes. So these are the brain cells called microglia. So microglia is basically, so they're very immune cells. Brain doesn't have immunity. That's why the most important part of our body doesn't have immunity, then doesn't have its own immunity. So brain import immunity from the bone marrow brain cells. They are brain macrophages. They migrates and they basically controls all inflammations. So we selectively identify the macrophages. And what we found by CD6 state activations. But CD6, if any macroglia is activated, it clearly indicates the chronic neuroinflammation and there's no treatment for that. So we found out most of the microglia, the process has been changed here. See, this is it goes down in the male, while female we identified here. So this is a blue double markers. They are more, the cellular expression of microglia, more is activation has been happen. So this we found there's no, now we have identified the neuron also, glia also, astrocytes also, thrombocytes also. We identified 6 or 7 type of cells, but most of our finding major, we have identified that microglia heterogeneity is literally responsible for this. Now going to the next point to this. So this is again is microglia sensitivity. Here we identify the CB1 receptors. I think this something clear is not coming. Otherwise this hair is basically red color. So if you see it here, because there's a red color. Why red color? Because CB1 receptor basically present on microglia and mitochondria. So most of the mitochondria, CB1 receptor control is called the brain of our body. So cannabinoid receptor 1, there's a 2 type of receptor, cannabinoid receptor 1 and cannabinoid receptor 2. Cannabinoid receptor 1 control the energy, while cannabinoid receptor 2 control the immunological response. That's why the cannabinoid receptor 1 is selectively present on the brain in the mitochondria. And how we identify this? We did confocal imaging in very depth, and we double label the first whether it's neuron or whether it's astrocytes. We identified is glia because there's no change in neurons. Then we identify the mitochondria here, and after that we identify the mitochondria, what type of receptor. Then we found it is cannabinoid receptor 1 receptor. So finally we determine the phenobloid with cannabinoid receptor 1 is selectively get degraded in this, and this receptor has role on the basically the mitochondrial functions, autophagies, as well as controlling the mitochondrial subdivisions and inflammations. It's also a powerhouse of the cell because it controls the energy transmission. So we found significant difference here. So second part, we identify the brain mature neurons, because smart brain has a lot of neurons, basically neuron positive cells. And we found there is selective loss of neurodegenerations in the male mice. Because if you see, the estrogen has a big role in protecting all these females. Because usually the male are more stressed because there's a, if you know, polyglycol pathway. This is basically when your glucose goes up, the hexokinase, the glucokinase, in certain limits, they can engage the glucose. If 100 millimolar glucose go to body, the polyglycol pathway gets started. That's why the hyperglycemia, if 300, polyglycol get activated. This polyglycol get activated with a lot of mannitols, a lot of the sorbitols, and they are very selectively more in the male's brains. And that's the reasons they have caused more. The male mice goes very severely change on the all this type of neurodegeneration. So this is how it's going down. Although this is basically part of CA1. It's a part of the brain where the pre-mitochondrial neuron are there. So if they goes, they cause a lot of neurodegenerations. Coming to the next, so we have activated what is the pathway for this? This is TLR4. So TLR4 is also immunogenic neurotransmitters, so neurogenic pathway. So all the microglia, they are found positive for the TLR4. More the TLR4 is became active, more has been neuroinflammation has been linked. So here we have identified M1 and M2 basically. So M1 basically the microglia are defensive, and the M2 is basically pro-inflammatories. So this is the ratio has been disturbed. So this ratio has been disturbed, is more activated form has been observed, and the TLR4 is most critically responsible for activating this microglia. Shaking. Okay. So finally, the conclusions. So we finally conclude that males are basically more sensitive to the hyperglycemia, and the major pathway, the underlying mechanism is endocannabinoid signaling, leading to the degradation of neurotransmitter 2-AG and anandamide. And also there are associated enzymes, especially the FA, fasciacidamide hydroxylase, which is critical enzymes, play a role in the degradation of both the enzymes. So the overexpression of FA basically reduces the content of the anandamide in 2-AG, which play critical roles on the microglia activation, neurodegeneration, apoptosis. Second, we also have in the males show a lot of TLR4 overexpressions, and which is may be responsible for the neurodegeneration and the cell loss. I think thank you very much for your attention.
Nice talk, Dr. Pariyar. Okay. The session is open for question. We'll take only 2 questions. Yes, anyone?
Okay.
I would like to ask one question. This is a wonderful model which you have used, well-established model, diabetes, streptozotocin and high fat. This model is also done in rats.
Yes.
You have used mice in this particular experiment. So can you just comment if same model would have been done with the help of rats, what will be the effect?
Actually, yeah, the effect may be the same, a little bit different. There's no extensively rat is different because this high fat diet is pretty costly. You cannot take so much of high fat diets is 3 to 4 lakh per batch. If a rat will go more than 10-15, one is cost. Second, mice has a lot of option in transgenic mice. I'm using a lot of transgenic mice in my study where accelerated brain aging, Alzheimer, Parkinsonian. So right now, rat doesn't have that options. You want to compare with the, because now new model has come to the diabetes-induced cognitive impairment where the insulin resistance is created and mice came. That's why I prefer mice. Rat you can do, but expensive. Housing is very difficult for this. And mouse is more behavior-wise easy to do. You water maze, open arena tests, you can easily handle them. You can do faster also. That's the one thing. That's basically I have done. Okay. Okay. Thank you very much for this. Okay.
Nice talk. Now I request the chairperson to kindly present a token of appreciation to sir. Kindly give a round of applause.
Uh, the second speaker is Dr. Harmanjeet Singh. Dr. Harmanjeet Singh is MD, DM in Clinical Pharmacology, and he is Assistant Professor in Department of Pharmacology at the Government Medical College and Hospital, Sector 32, Chandigarh. He earned his MD in Pharmacology and later completed DM in Clinical Pharmacology, building a solid foundation in both basic and applied pharmacological sciences. Dr. Singh has made important scholarly contribution including publication on antimicrobial resistance, strain-related muscle symptoms, and the pharmacology of the neuropsychiatric cardiometabolic therapies. He has also presented work at national and international forums, notably on awareness of the pharmacovigilance and vitamin D statin interactions. Dr. Singh has published 9 papers, and in recognition of his professional achievement, he was elected a Fellow of British Pharmacological Society in 2023. Committed to evidence-based medicine and mentoring, Dr. Singh actively contributes to the pharmacology curriculum and training the future clinician as well as researchers. So let us welcome Dr. Harmanjeet Singh, and I will request you to complete your talk within 15 to 20 minutes so that we can have questions.
Good afternoon, everyone. I'm thankful to the chairpersons for a nice introduction, and I'm thankful to the organizing committee for giving this opportunity. So my topic is about, as you know, that GLP-1 receptor agonist and GIP receptor agonist, like semaglutide, tirzepatide, so they are widely used in the metabolic medicine. So they have extended their horizons from the management of type 2 diabetes mellitus to obesity, and they have now being used for cardiovascular protection as well as to metabolic associated steatohepatitis. So as far as their use is concerned, so there is widespread use. So with this widespread use, certain safety concerns are likely. So my talk is about the raising concerns about those safety concerns, and also so what is their status and what all are the regulatory changes, and so what should be done on the clinicians as well as on the patient side so that we can have a safe use of these agents. So this is the outline of my talk. So we'll be talking about evolution of GLP and GIP therapies, their clinical success, traditional safety concerns, real world challenges. So what are the emerging safety concerns with them? Also the public health message, and then finalizing with the summary. So this is the evolution of GLP-1 agonist and GIP therapy. So first of all, there was a discovery of extendin-4, so which was discovered from the Gila monster, a type of lizard. So then there was the discovery of exenatide, so which was the daily use exenatide. So then in 2010 and 13, we have more drugs, liraglutide, lixisenatide. So then 2014, we discovered once weekly exenatide. Then 2014 and 17, two more weekly agents were added, dulaglutide, semaglutide, and then we came up with oral semaglutide, so which is still in use. So then 2021 to following that, we discovered semaglutide for obesity. So then 2022-23, tirzepatide got 2 approvals for the type 2 diabetes mellitus as well as for obesity. So then their use were extended for cardiovascular protection and MASH. So this is their evolution. So coming to their clinical success, so they are highly effective in type 2 diabetes mellitus and obesity. So they are leading to HbA1c reduction of around 0.9 to 2. So there are many named trials: Sustain, Surpass, Step, Surmount, Leader trial, Sustain. There's a series of Sustain trial from 1 to 6 and onwards, and Select trials, Flow trials. So all have shown clinical success. So they are, leading to weight loss of 15 to 23%, which is way higher than the current available therapies. So then they have shown the cardiovascular benefits. So they are reducing the major adverse cardiac events in all these Leader, Sustain, 6 and Select trials. So then they also extended the renal benefits. So in Flow trial, so now they have been approved for the metabolic dysfunction associated steatohepatitis. This is the recent approval. So semaglutide here got accelerated FDA approval. So these are the different approvals as far as US FDA and Indian setup, CDSCO. So for type 2 diabetes, all those which I discussed in the last slide, all those drugs have been approved, plus in US FDA for the management of type 2 diabetes mellitus, along with tirzepatide. So then for chronic weight management, there are 3 drugs: semaglutide, liraglutide and tirzepatide. So their name is different. So the names are different as far as their indication for type 2 diabetes mellitus and obesity they are concerned. Then cardiovascular risk reduction, Wegovy, semaglutide. So then for MASH, again Wegovy, which is also semaglutide. So then India, most of the GLP-1 agonists are approved for type 2 diabetes mellitus, and only one drug is available for the treatment of obesity, semaglutide. This tirzepatide, although it has been shown by the media reports that it is approved for obesity and all, but it is not approved in India for the treatment of obesity, but they are extending the indication as off-label use. So that is backed up by various media reports which are not right. So then these are the different doses which are used for different different candidates of these two groups. So for semaglutide, we are mainly starting with 0.25. We are raising the therapy every 4 weeks, accelerating the therapy, and then we are reaching up to 1 milligram. So the 1 milligram is the highest dose. Then for this oral, we are starting with 3 milligram, and the 14 milligram is the maintenance therapy. Then obesity, we are starting with 0.25 milligram, but most commonly used doses are 1.7 and 2.4 milligram daily. So they are way higher than the dose which is used for the treatment of type 2 diabetes mellitus. So then there are dulaglutide, this liraglutide, they're less well used as of now, but tirzepatide is widely used for the treatment of type 2 diabetes mellitus under the brand name of Mounjaro, and for obesity as the brand name of Zepbound. So the dose is starting from 2.5 mg, we go to 15 mg per week. And obesity, same titration, but the most commonly used doses are 5 to 15 milligram weekly. So from this you can make that for obesity we are using relatively higher doses. So these are their mechanism. You all know that their receptors are widespread in the body, mainly in GIT, pancreas, hypothalamic areas, and renal and vascular tissue. So they are producing variety of beneficial effects. So they are enhancing glucose-dependent insulin secretion as the food enters your gut. So there is increase in blood glucose level. So this leads to stretching of the gastric mucosa, which sends a signal for the secretion of the GLP-1. So which leads to various beneficial effects: insulin secretion, suppressing of glucagon secretion. Also, so there is slowness of gastric emptying. So this is also responsible for most of their adverse effects. When there is slowing in gastric emptying, so there would be increase in nausea, vomiting potential. So also this is modulating satiety center. So these effects are also helpful in their anti-obesity action. So also hypothalamic appetite center, reducing hunger and energy intake. So there are two types of signals, or exogenic and anorexigenic. Or exogenic is by the neuropeptide by and a Gauty related peptide system. So which are suppressed in this case. Also, there are anorexigenic pathway, POM, FUT, pro-opium cortinin, and then there is cocaine and amphetamine related transcript. So they are anorexigenic, so they decrease food intake, they decrease the craving. So those are stimulated by these agents. So also they are having renal and vascular benefit. So they improve glycemia, they lead to weight loss, they reduce blood pressure and lipid, there is improvement in lipid content, also cardiovascular and renal protection. So then GIP receptor activation, apart from all those actions, so they are also present in adipose tissue in large quantity. So in adipose tissue, they have, they decrease the fat mass, so they lead to weight loss. So this is the additional action of GIP receptor agonist. So these agents are acting on mostly GLP-1 receptors, and tirzepatide is also acting on GIP receptor. So what are the traditional safety concerns? So these are well written with their, in every textbook, in every, drug package insert of every drug. So they are leading to nausea, vomiting, early satiety, decreased appetite. Also because most of them are subcutaneous injections, so they are leading to injection site reactions. So also there are some, their labels have been given more adverse effects like hypoglycemia only when they are used with hypoglycemic agents like insulin and sulfonylurea. Also pancreatitis because they are leading to ductal hyperplasia, and also there is more increase in secretion by stimulating the acinar cells. So pancreatitis, it is a rare risk. It has shown to be largely neutral with the long-term randomized controlled trials. Also, rodent-specific phenomena is their association with thyroid C-cell tumors. So this has not been seen in clinical setting. They have not been seen in most of the clinical trials. So coming to the emerging safety concern with them, so we have some key clinical concerns like diabetic retinopathy progression. So there are gallbladder and biliary disease, gastrointestinal hypomotility disorders, gastroparesis, ileus or obstruction. Then there is acute kidney injury, which is mostly dehydration related. As they are leading to gastric, they are decreasing the gastric emptying, so there is more of nausea and vomiting. So that can lead to dehydration. So that can lead to acute kidney injury in the pre-renal fashion. So then there are signals which are under ongoing evaluation. They are not of much concern, but again the caution is needed. So that is non-arthritic anterior ischemic optic neuropathy. Then there are low likelihood concerns like psychiatric events. So sometimes they were, sometimes back they were related to, they were shown to be related to depression and suicidality. Also the sarcopenia, where the body fat loss is more than the lean body mass. So these are the emerging safety concerns which we should know, and also we should, monitor them so that their actual frequency and actual damage should be known to the public. So why these emerging safety signals are now appearing? So because now we are using higher doses, as you have seen that for obesity related, this treatment, so there's likelihood that higher doses are being given. So they are leading to more adverse effects. Then rapid weight loss physiology. So they are having very potential effect on weight loss. So they are decreasing weight by, as compared to, significantly as compared to other agents. So this rapid weight loss is leading to some metabolic disturbances, like there is gallbladder stasis, metabolic shifts, ocular perfusion changes, also changes in patient population. So now you have seen even the patient who are not type 2 diabetic, or where the body, this BMI is not more than 30 or 27. So they are also using these drugs for cosmetic purposes, for the aesthetic purposes. You must have seen many this celebrities like this Karan Johar. So he's using this. He has been, I mean, there are some reports that he was using these drugs. So then non-diabetes physiology. So we have changed the patient population. So the selection of patient population that has been changed. So now more and more adverse effects are appearing. So they are of they're entirely new adverse effects which were not cited in the textbook or the previous articles. So now also there is off-label and unsupervised use. Online sources are there. Then we are using them in other than recommended doses. Also limited follow-up of all those patients who have been given these drugs. So then there is population scale exposure. Now millions of patients are using these drugs. Some are, actually requiring the drugs. Others are not requiring these drugs. And so that is why. So now these emerging safety concerns are appearing. So first, coming to the diabetic retinopathy progression. So first it was highlighted in the SUSTAIN 6 trials. SUSTAIN 6 trial where the semaglutide cardiovascular outcomes were seen. So it leads to diabetic retinopathy complication with hazard ratio of 1.76. So this was published in NEJM. So this is mainly seen in patients with pre-existing diabetic retinopathy when there is rapid change in or the rapid drop in HbA1c. So this leads to a phenomena which is known as rapid glycemic correction. So there is also contradictory evidence to this. So there is one leader trial which was for the liraglutide and Rewind trial. So they have detected no diabetic retinopathy signals. So also American Society of Retina Specialists in 2025, they have analyzed data about 160,000 patients. So there was no excess sight-threatening diabetic retinopathy. So this is a fresh report, but they have not given the proper reference where the studies, populations, what are the other aspects. So that is not there. Then the issue, this mostly be related to rapid glycemic correction rather than the direct retinal toxicity by these agents. Also, FDA has given warning regarding this relationship. So now this is the paper which was this is a study which was conducted by me and my team. So we also found increased risk with all these agents. Semaglutide had very strong risk, then followed by dulaglutide. And this tirzepatide and liraglutide showed a weaker but significant association. While lixisenatide, the most probable reason is that now it is not being used. Lixisenatide was withdrawn by the US FDA because of its low sales. So it was not, but because its competitors are more stronger, so they are having better sales. So it is not used. So we had very limited data for lixisenatide. So rest of all the agents were associated with the diabetic retinopathy. So this is the proposed mechanism for diabetic retinopathy. So there is rapid and large fall in HbA1c because they are very strong agents which are reducing this HbA1c. So with this, there is disruption of retinal autoregulation and perfusion. So this gets disturbed by the changes in HbA1c. So there is increased retinal capillary leakage, edema, and microaneurysm turnover. So this leads to transient worsening of diabetic retinopathy. This is the proposed mechanism. This is due to large and the rapid fall in HbA1c. Then gallbladder and biliary disease. So this association is also in discussion nowadays. So there are some, most of the reports are linking them with these gallbladder and biliary issues, and some reports are contradictory also. So a systematic review of and meta-analysis of 36, RCTs. It's a big meta-analysis. Around 100,000 of patients were included in this meta-analysis. So they have shown that this use of GLP-1 receptor agonist is associated with increased risk of gallbladder and biliary events with the relative risk of 1.37. So it has also been seen that in the same meta-analysis, the patients who were enrolled in obesity trials and were taking higher doses for a long duration, they were more associated with this gallbladder and biliary events. So you should consult the patient for any right upper quadrant pain, and early ultrasound should be done if patient is symptomatic. Also, the regulatory guideline has been given by the US FDA regarding the same. So this is our ongoing study. We have almost completed this study. This study was also presented by one of our senior residents in the same conference. So we are finding, very strong signals with semaglutide and tirzepatide, followed by exenatide and liraglutide. So these are all associated with gallbladder and biliary events. So these are the mechanisms. There are two type of mechanisms which are possible. One is hormonal pathway. As GLP-1, the receptor agonists, they are decreasing the cholecystokinin, which is the hormone which is required for gallbladder contractions. So with the gallbladder contractions, it increases the passage of bile. So now there is because this GLP-1, they decrease the function of cholecystokinin. So there is poor gallbladder emptying. So there is stagnation of bile. So there is propensity of stone formation. Also, there are metabolic pathway because there is rapid weight loss. So there is high cholesterol load which is presented to the liver and bile. So now bile gets supersaturated with cholesterol. So this is supersaturated bile. So there is stagnancy and also there is propensity to cause gallstones. So these are the probable mechanisms which are leading to gallbladder and biliary events. So now one more concern is about gastroparesis and ileus and the intestinal obstruction, very much related to their mechanism because they decrease gastric emptying. So that this decrease in gastric emptying can lead to gastroparesis like presentation. So also observational real world studies, they have shown their association with gastroparesis and this intestinal obstruction. The FAIRS data signals also, this FDA adverse event reporting system studies also detected these signals, and also there is a regulatory guideline for this. Then one important concern nowadays is acute kidney injury. This is related to their potential to cause dehydration because of their serious GI toxicity. So this leads to vomiting, dehydration, and it leads to pre-renal acute kidney injury. So also some case reports have shown that there is acute interstitial nephritis related with semaglutide. So there is also potential hypodipsic effect. This is also very important as they are known to cause dyspepsia. So they are decreasing the food intake, decreasing the cravings for food. Also they are decreasing the water intake, also craving for water intake. So this is the additional effect which can lead to more dehydration and can lead to this can be associated with more renal damage. So also FDA adverse event reporting system signals, they have shown AKI with semaglutide and liraglutide. Liraglutide was more strongly related. So but with the FLOW trial, this was conducted with semaglutide. So here semaglutide has shown contradictory effect. So it reduced major kidney outcomes and CV mortality, cardiovascular mortality. So also there is regulatory guideline by the US FDA regarding this safety concern. So then non-arthritic anterior ischemic optic neuropathy, which is related to, which is unrelated to the diabetic retinopathy, but this is a new concern. So there is optic nerve head perfusion vulnerability in patient with vascular risk factors. So there are rapid metabolic changes like weight loss. So there is altered perfusion reserve. Also the nocturnal hypotension, because most of the cases of neon, they are detected during the sleep only. I mean the most of the patient, they develop this neon during sleep. So nocturnal dip in blood pressure is also responsible for reducing the optic disc blood flow. So this neon is seen with these agents. So overall, this is a heterogeneous finding, and there are many confounders which could be there. So this is considered a signal under evaluation, no FDA warning, but this European Medicinal Agency has raised this concern, citing neon is a very rare side effect of semaglutide. So the clinical message is any sudden painless vision loss whenever the patient is taking these agents. So you can suspect the neon. Until proven otherwise, urgent ophthalmology referral should be made in this case. Then also sarcopenia, lean mass loss.
Dr. Singh, we are running short of time. Please
just 2
try
minutes. Just 2 minutes. So this is also because they are leading to decrease in body fat. So this also decreases the lean body mass, muscle mass also. So this is one of the toxicity related to these agents. So you have to ensure the adequate protein intake. You have to encourage the strength training exercises. Also, you should avoid the over-aggressive doses and monitor muscle strength in older adults. So these psychiatric adverse events, so they're not of much concern. So because the FDA has conducted an analysis, they have found that these are not likely to be related with them. So other concerns are there are skin and face changes. So now the term has come Ozempic faces. You can correlate it with Karan Johar and you can see what changes can be there. So these are the changes. There's a gaunt, hollow, old appearing face, loss of mid-face volume, skin looks looser and more wrinkled. So this is not a drug toxicity but result of fast weight lose. Okay, so you have to be very careful about the dietary changes and all, and taking protein, diet. Loss of libido was also found to be associated in some reports, but now they have negated this risk. So what are the real world challenges with these drugs? One is unsupervised use, compounded semaglutide. I will take 1 minute on this, please. This compounded semaglutide, it's not a crime because drug compounding can be done when patient is in a dire need of it. We can change certain ingredients, we can change the content, okay, when it is needed, but it should be reported to the regulatory authorities. But now what, there's a black market. With the black marketing, some companies are doing compounding and they are adding some peptides and other things which are not needed, so which are not approved by the FDA or any other regulatory authority, but they are being used in the in compounding these drugs. So that is also very dangerous. So we should avoid that. Then online and black market is there. For gym injections are available. Beauty parlors are supplying these. Some of the quacks are promoting these drugs. So and social media driven dosing is there. And rapid titration every 3 to 4 days. With the expectation of getting more weight loss. So some people are giving, they are titrating them every 3-4 days. Otherwise, they should not be titrated before 4 weeks. So there are system gaps also, inadequate patient selection, as I told you, no screening for the safety concerns, no hydration counseling, no follow-up, and limited cleaning training in obesity pharmacotherapy. So we should be careful while using them, while titrating the doses, and also we should do some screening for the safety concerns and the public safety messages. So they are generally safe, effective, and transformative, but more serious adverse events occur due to off-label use, non-medical dose escalation, compounded unregulated product, absence of monitoring, and dehydration plus vomiting. So you can read this FDA, guiding document. This is very important regarding the upcoming safety concerns. So generally, they are GLP-1 receptor agonists and tirzepatide. So they have transformed metabolic medicine. So RCTs plus real-world evidence, they are complementary. Regulatory agency should activate, update safety guidance, and with proper safety selection, monitoring, education, benefit-risk balance remains highly favorable. So we should avoid the misuse. We should avoid the inadvertent use. So with that, we can get rid of these adverse events. Thank you very much.
Yeah, thank you, Dr. Singh. And really, this was a talk which is of interest of many sitting in the audience, and it is concerned to everyone. So let us have some quick questions from the audience. Yeah.
Actually, so there are many, as type 2 diabetes has been shown to be very important risk factor for the Alzheimer's disease. Even Alzheimer's disease, most of the this in unregulated way, they're classifying as one of the type of diabetes also. So with the rapid reduction in their glycemic beneficial effects, decrease in control in glycemia, control of glycemia, and also so there are also I mean active in CNS, certain CNS pathways. So with that, they are not being used as of now, but the research is definitely ongoing. So we have to see for the long term, benefits. So as of now, they are not of any help, but with the more research and with the more data, with the more insight, we can reach to some conclusion. As far as Mike's research is concerned, I have not done this as of now.
Thank you. Ma'am, please.
Yes. So accelerated approval was granted for MASH, which is metabolic dysfunction associated steatohepatitis. So we have only one approved drug for that, resmetirom. So that is the only this FDA approved agent. So we need drugs, and in one of the trials, so they have shown Wegovy, which the semaglutide has shown very promising effects. So that is why it has been approved. Otherwise, we have very limited option for that disease.
Thank you, Dr. Singh. Any other questions? So, Dr. Singh, I have a question. For how long this therapy is usually long?
Sorry, sir?
For how long this therapy is usually recommended?
This is recommended, sir, lifelong.
Lifelong.
For type 2 diabetes mellitus.
No, for weight loss.
So, it is recently
shows
approved.
So many side effects. So what is the caution?
It is recently approved. Now they have, there are no clear-cut reports on this.
Is it a subcutaneous injection?
This is a subcutaneous
and
injection.
it is given
Weekly. Same doses.
So
2.4 doses have been, they have been
so
recommended. 2.4 milligram per week. It has been recommended for metabolic associated steatohepatitis.
So one.
But for the upper limit of there is no such,
So this subcutaneous injection, is it a long-acting one or is it a simple one?
This is a T-half of around 1 week, sir.
1 week T-half.
Yes.
Thank you. Thank you.
Any other question?
Thank you, Dr. Harmanjeet, for an impressive talk. Now I request our session chairs to kindly present a token of gratitude. Give a round of applause.
Next speaker for this session is Professor Dr. Sneha Bagle. She is an assistant professor in Department of Pharmacology at D.Y. Patil University School of Pharmacy, Navi Mumbai. With PhD in pharmacology, her research focuses on neurodegeneration, mitochondrial dysfunction, neuroprotective strategies, particularly in models of Parkinson's disease. She has published several peer-reviewed papers, including studies on ursolic acid's neuroprotective effects and antioxidant potential of dietary compounds. In her academic role, she also contributes to teaching, postgraduate mentoring, and curricular development at her institution. So we welcome you, ma'am, and I request you to start with your talk.
Thank you so much, sir. Thank you to both the chairpersons, Professor Sinha and Dr. Kulkarni. After 2 amazing sessions, this is the 3rd one. I hope everyone is awake. And it's a lot of pressure of me to complete this within 15-20 minutes. So I'll try and do my best completing it in 15 to 20 minutes. So to begin with, my title is Apocynin Intervention, Phytoactive Intervention for a Multifactorial Disease, Exploring Apocynin's Impact on Neurogenesis Beyond Neuroprotection in Alzheimer's. So I'll quickly go through the introduction. So the theme is Vikasit Bharat, We want empowering young minds to transform pharmacology for Vikasit Bharat. So what is exactly Vikasit Bharat? So we are aiming for, India is aiming for its vision for 2047, and in that vision we are saying that the GDP will go up, we'll have a good infrastructure so that people could commute easily from one place to other. Health is one of the priorities. We have education as on the priority list. And technology, we are shaking hands with the technology and making it tech friendly. Last but not the least is quality of life. What this presentation will focus on is mainly on these two aspects of Vikasit Bharat, that is health and quality of life. To begin with, I know everybody must be aware of the patient August Ditter, who was the first woman diagnosed with so-called Alzheimer's disease by the physician, Dr. Alois Alzheimer, who was the one who coined the term, or who was the one who discovered the disease. And the disease, two characteristics of this disease are we have extracellular plaques and intercellular tangles. So what are the causative factors? I'll quickly rush through. Aging, of course, brain injuries, lifestyle, reduced physical activities, a lot of stress. Even the young minds, younger generation is going through under a lot of stress. Change in food habits, change in lifestyle, reduced mental activities. We are scrolling throughout the day. Our screen time is almost 4 hours, 6 hours, which is bad. And the only genetic factor that is responsible for sporadic AD is apocynin or ApoE epsilon 4 genetic mutation. And then we have genetic factors like presenilin 1, mutations in presenilin 1, 2, and APP mutations, which lead to familial AD. So there are number of stages, or the disease goes through n number of stages of Alzheimer's. So stage 1, 2 will not have any clear-cut clinical diagnosis, but from stage 3, the patient will show the characteristic features of Alzheimer's, start to show characteristic features of Alzheimer's, and it can be clinically evaluated using brain imaging or CSF test. And progressively, it will go on progressing and try and engulf the entire brain region, starting from your medial temporal lobe towards the entire brain region. And at last, your stage 7, the person will lose his verbal ability, which is devastating at an age of 70, 75 or 80 years. He'll be incontinent, he'll require assistance for doing daily routine tasks, which is not good, or which is not, the elderly population will not be able to sustain that kind of lifestyle. So hence, we are targeting Alzheimer's disease. On the epidemiology front, if we see only Asia, the number of people suffering from dementia is quietly increasing, and it is progressively increasing, and 2050 will see 67.2 million of people suffering from dementia in Asia alone. And if we see the Indian perspective, the number of Alzheimer's cases is also increasing, and we are talking about only the reported cases. We are not talking about cases which go unreported. So we see the number of patients is increasing, and by 2050, we'll have 14.3 million cases. And if we see the data for 2030, and in 2050, it is almost twice as that of 2030. And in 2020, the number of people suffering from AD was 50 to 75%. Out of that, 5.3 million of people suffering from dementia. So it's a huge number, keeping in mind India being the one of the most populous countries. What Alzheimer's does on the economy, since we are aiming at a economy or GDP growth, if we compare both the urban and rural population, the financial burden is huge. It is almost 23,330 crores of rupees, which is a huge burden on our health economy. But then why, if we have so many increased number of patients, there is financial burden, why still there is a stigma associated with the AD, or what are the barriers to care for AD in India? First is, there is a lot of stigma associated. People do not like to go and see a neurologist, maybe citing that I do not have any brain related problems, but actually it is not a psychiatric problem, it is just normal aging, and because of that you are acquiring that disease. There is lack of awareness, because if you see the first clear-cut symptoms of Alzheimer's is forgetfulness, which is so very well associated with aging. So it goes undiagnosed, or even the family members will say that it is due to aging, that he's forgetting things, he's forgetting his keys, car keys, house keys, etc. There is low health-seeking behavior for memory problems, because it is part of normal aging, considered as part of normal aging. There is less specialist manpower to treat and care dementia. We are talking about caring for people suffering from dementia. The care that we give to patient is only from the family members, at least in India. And available services need upgradation. And there are lack of policy initiatives taken by the government of India to treat and tackle the problem of increasing AD patients. And as a researcher and all the young minds sitting here, the problem is lack of funds for training services and research. So, but still, we have a community which is called as Alzheimer's and Related Disease Disorders Society of India, which is ARDSI. They have come up with a 7 actionable plan. What is that actionable plan? Making dementia a national health and social care priority, taking that priority list to a higher rate so that people are aware. They do not think Alzheimer's is just an aging disorder. Creating awareness, risk reduction, and dementia prevention. How can you prevent dementia? Improve access to best medical care facilities, strengthen the standard treatment protocols, social support services, strengthen dementia disease surveillance system, and lastly, invest into research and development. Unless and until we have a good molecule that can at least stop the progression, forget preventing, but at least stopping the progression is much better so that the patient doesn't go to level 7 or stage 7 of Alzheimer's disease. Quickly run through the pathophysiology. We all know that it is a journey of a healthy neuron from a healthy lifestyle or a healthy status to a very debilitating state. So what happens in Alzheimer's exactly is you have aging, and there are many pathological concerns associated with increases the amyloidogenic processes of A beta, leading, so increase in amyloidogenic processes and decrease in clearance will lead to accumulation of amyloid beta plaques, increasing the production of amyloid beta. That will lead to A beta being imported inside the mitochondria and targeting mitochondria, leading to oxidative stress and mitochondrial dysfunction. Mitochondrial dysfunction in return can cause oxidative stress. You have increased A beta production. Mitochondrial dysfunction can increase A beta production. Also, oxidative stress can increase A beta production. Ultimately, when you say that it is being hampering your neuron, mitochondria is the heart of your neuron. So in mitochondria, you have increased oxidative stress, there is decreased antioxidant enzymes, you have abnormal dynamics, mitochondrial dynamics, decreased complex 1, complex 4, impaired biogenesis, bioenergetic deficits, and there is mitochondrial pore formation hampering your mitochondria, which is the energy currency of your neuron, and neurons need them much more than any other cell of your body. And there is induction of apoptosis and leading to increase in caspase activation. Along with all these factors, we have cell surface receptors that are expressed because there is increase in A beta production. This cell surface receptors, expression of these receptors leads to inflammatory or storm. We can say there is pro-inflammatory cytokines rise. This rise can increase your A beta production. A beta production, increase in A beta production can lead to increase in pro-inflammatory cytokines. Increase in A beta production can also have an effect on your neurotrophic factors. So neurogenesis would be hampered. Forget neurogenesis, your normal neuronal health will be hampered, leading to decreased neuronal survival, decreased synapses, and decreased synaptic plasticity. Also, increase in A beta production can cause an altered neurogenesis. All these factors ultimately culminate into neurodegeneration, which sets in a vicious circle. One leads to the other again leads to back to the first one, leading to AD. So if we see the slide, Alzheimer's is a multifactorial disease. So we need to target many aspects of the disease to curb or halt the progression. And we know the current treatment option. Lately or recently, we have 2 drugs that have been launched into the market, lenanemab and donanemab. Both remove A beta plaques. One was in 2023, one was in 2024. Both have been manufactured by ESI Biogen, and the one is manufactured by Eli Lilly. Both are IV infusions, so they have their own sets of disadvantage, and that disadvantage or side effect is amyloid-related imaging abnormalities, wherein we see brain swelling and bleeding leading to headaches. Donanemab has higher risk compared to the other, but both show ARIA-like side effects. When you talk about clinical trials, as of 2025, we have 138 drugs in 182 clinical trials. So there are n number of drugs in phase 1. We can see, refer the slide, in phase 1, phase 2, phase 3. We have recently FDA-approved drugs that came up in 2023, but that still needs some R&D to be going on because they still have side effects, and these side effects are huge or major side effects. So what is the current need? Because there is change in population demographics, we say that India is a younger country because we have younger population in the working age is higher in number. Saying that, we have geriatric population which accounts to 9.7% of total population, which is 138 million. It's a huge number to be taken care of. Last approved drug came in 2023, but still we are want to go deeper and dig deeper into R&D and find for a newer cure. Continuous failures in clinical trials, shortcomings of the existing treatment, increased AD patients and increased deaths associated with AD. That is why we require a drug pressing need for development of new treatment regimen or new drug with totally different pharmacological approach that would halt or slow down neurodegeneration. So we need an assistant drug that will at least halt the neurodegeneration, if not prevent it, but at least halt it. So we come up, or we have selected through a literature review, apocynin, which is a acetovanillone. It is methoxysubstituted catechol. It is isolated from Picotosa curoa, which is an Indian plant. Also from other varieties of plants, and it has been used traditionally in Ayurveda to treat fever, scorpion sting, upper respiratory tract disorders, etc. It is an NADPH oxidase inhibitor, so it can curb the inflammation, which is one of the most important parameters when we talk about pathology or pathophysiology of AD. Antioxidant, it has antioxidant, anti-inflammatory properties. It was seen effective in MPTP model of marmoset and rat, and the log P of 1.62. Based on this, we selected apocynin to be treated or to be shown effective in preclinical models. We did 2 models, one in zebrafish and the other in rat. So why zebrafish? Because the organization is similar to humans. Neurotransmitter pathways are mapped and they are present, so we can trace down the neurotransmitter levels. They can learn mazes like passive avoidance, conditioning mazes, etc. And lastly, neurobehavioral studies and neurological studies are easier to be performed. And it's a miniature platform. When you talk about zebrafish, it is a high throughput model. Miniaturized format testing. Treatment is easy because the animal is small and it is cost effective. When I did the study, there was no ethical approval required for a zebrafish, hence you'll not see any IAC approval here. So we did simply a simple assay, that is passive avoidance test. So this was the apparatus that was used, and you see a white compartment on your left side and a dark compartment on the right. So it's a simple test wherein you train the zebrafish first so that it acclimatizes in the white compartment. You raise the door, the sliding door which is there in the center, so that the zebrafish enters the dark compartment. Once it enters the dark compartment, you drop a marble in front of the head. So that is the aversive stimulus. So the zebrafish needs to be trained that he should not enter, or the fish should not enter on the dark side, because there is a danger. If zebrafish retains that, that means your drug or your molecule is working wonders. If it does not, there needs to be something done. So this was the syringe or used for intraperitoneal and peroral injections. These were the treatment groups that were selected. So we selected apocynin 10, 30 and 70 mg per kg, and there was no study done in zebrafish using apocynin. So we had to do maximum tolerated dose studies to arrive with this dose selection. So when we did the assay, or when we did the behavioral test, we found out that the control fishes were able to increase the latency of entering the dark compartment. On the other hand, which was reduced by scopolamine, which was used as the inducing agent that would hamper the memory. And apocynin, the lower 3 slides or lower 3 graphs depict that apocynin was able to retain the memory in zebrafish. When we compare all the doses, we found that 30 mg was better in comparison to 10, 30 and 70. We wanted also to understand what is the long-term retention. So it's the same trial. The trial was given on day 1, and only the test was repeated for the second day and the third day. And here we found out that, of course, scopolamine has its effect. It reduced the it reduced the latency so that the zebrafish memory was impaired. And apocynin was able to restore, those not significant, but apocynin 30 mg was able to restore that memory even for a day 3. We also wanted to quantify the oxidative species or redox species, and we saw that apocynin was able to, reduce reactive oxygen species at all the 3 doses, which was increased by scopolamine administration. We found out the glial proliferation or the inflammatory reaction in the zebrafish brain by doing HNE staining, and we saw that apocynin 10 and 30 mg were good for lowering down the inflammation in the zebrafish brain. So to conclude with part 1, scopolamine induces memory deficit, so it's a well-known model. Apocynin reverses scopolamine-induced memory deficit, and it shows neotropic effect. It has again a neotropic effect because it retains the long-term memory. It reduces ROS generation, and it curbs scopolamine-induced neuroinflammation, showing neuroprotective behavior, and can be used as an acute model. So it was a 3-day model, and it doesn't require much. So the cost or the investment is literally very less, but now you have ethics approvals, you have to take approval priorly. The model is very quick, and it is an effective preclinical tool, and this was published into a publication. Coming to the rat model, so rat model...
Hello, ma'am, we are running short of time.
I know, sir. Last 5 minutes, I'll try and complete.
5 to you can finish in 2 minutes.
Sure. I'll just quickly run down through the results. So we did Barnes maze, and we saw that apocynin was effective in, decreasing the latency to find out the box in Barnes maze test. We had antioxidant enzymes like SOD, GSH, catalase, and LPO, wherein apocynin was effective. Apart from that, we tried and estimated the expression of RAGE, and when we did that, this is basically the pathophysiology of how RAGE works. It increases the neuroinflammation and ultimately it causes oxidative stress. So RAGE, we saw that apocynin was able to reduce the reduction of RAGE expression. Caspase-3, we wanted to see the expression of caspase-3 because apoptosis is one of the causes of Alzheimer's. So apocynin was able to reduce caspase expression in hippocampus as well as the brain cortex. We did IHC staining to understand the A-beta levels, and we saw that apocynin reduced A-beta levels, but it was not significant. We did congruent staining. In congruent staining, we saw that the score was less for apocynin compared to amyloid beta. Similar results were obtained for Nissl staining and HNE staining. Coming to neurogenesis, we wanted to find out whether apocynin could trigger neurogenesis, so adult hippocampal neurogenesis, and we saw that apocynin increased BDNF levels. It increased the Ki67 levels, and also it had an effect on the DCX levels, which is involved in migration of neurons. So once the neurons migrate, once they go to the particular area, they will show proliferation and convert into mature neurons. And then lastly, we saw the neuronal nuclei, which is an adult neuron marker. Then we did mitobiogenesis, wherein we try to see the PGC1 alpha levels, which is a master regulator of mitochondrial biogenesis, that is increase in mitochondrial number, not only mitochondrial number, but healthy mitochondrial number. And we saw that apocynin was able to do so. To conclude with, it has nootropic effects, antioxidant effects, it reduces A-beta deposits, it has an anti-inflammatory effect, apoptotic, reduces apoptosis, it has neuroprotective effect, it has neurogenesis aspect, and lastly, it has mitobiogenesis. So the key learnings were we could have used a standard drug in our zebrafish model, which we did not do that. The model for rat could have been for a longer duration to see and understand the neurogenesis aspects or parameters or markers for neurogenesis. And more downstream markers could have been used for mitobiogenesis. We could have checked the protein expression, where what we did here was we checked the RT-PCR. Using RT-PCR, we have checked the expression of proteins. We could have and lastly, we could have done PK-PD correlation, which is ongoing process. And here are young people sitting here who are doing their UG PhD. I'll just take 2 minutes. So there should be resilience when you're doing, because there will be ups and downs. This task was not done. When we talk about that zebrafish model, which was 5 slides, concluded in 5 slides, but it took 2 years to just standardize that model. Literature survey will not only help you to design your study, but also to help you support your results and conclusion. Work with simple assays initially, have patience, work with a collaborative spirit, because only you working alone will not help you out. You should follow ethics in research work and research publication and research practices. You should always go for critical thinking. You might get negative results, but you can, show them, or using your literature, you can, reverse engineer that and show what exactly that effect could be because of. Excellent communication skills you need to have. Extract maximum from a single model. We did only a single rat model because it's expensive and we want to extract maximum what we can do. Time management is necessary. You should keep an open mind. Do not go that this is my hypothesis, so my drug should increase that particular protein. It might not increase that particular protein, which we saw when we did our RAGE assay. Be open in whatever results you get. So there is, and lastly, we have technology, we have network pharmacology. Please join hands with them. It is going to take you to places. This is too much. Lastly, don't forget to enjoy your life as a PhD scholar maybe, or as a research scholar. You might face with all these emotions each day, every day, or in 6 months. Throw them back and come back with a happy emotion. Eat. Do not forget to eat. Give your time to your family, friends, and have a social life. Enjoy the sport that you want to play. Enjoy each and every weather. And keep celebrating small achievements. And do not forget to rest. Rest is important. So, thank you.
Thank you, ma'am, for this nice presentation. We'll have only one question, running short of time. Yes, anyone? Thank you. Thank you, ma'am.
Thank you so much, sir.
Thank you, Dr. Sneha, for such an descriptive and informative talk. Now I request session chairs to kindly facilitate Dr. Sneha with a token of gratitude. Kindly give a round of applause.
So we are moving to the next speaker. I think probably the last speaker of this session, Dr. Rahul K. Verma. Please welcome him. Dr. Rahul K. Verma is Scientist E, Associate Professor and Head of the Unit of the Institute of Nanoscience and Technology, Mohali, that is just in vicinity of the Chandigarh. He received his PhD from CDRI, Lucknow, and his MS from the NIPER, Mohali. He also completed postdoctoral research at the University of Oklahoma and USA, and was a visiting researcher in the Bradford University, UK, with support from the Royal Society. Dr. Verma has authored over 85 peer-reviewed articles and holds H index of 28. His research is dedicated in designing safe and effective nanoparticulate delivery systems, microparticles, nanoparticles, porous aggregates for pulmonary, nasal, transdermal, intra-articular delivery. So please welcome him and let us enjoy his research for next 15 to 20 minutes.
Good afternoon, everyone. Thank you, sir, for the kind introduction. I would like to thank the organizing committee for inviting me in this conference to present my data. Today, I will give a brief overview of my recent research, that is fair responsive or inflammation responsive drug delivery for the rheumatoid arthritis. So before diving to my research, I would like to give a brief introduction of my organization. So Institute of Nanoscience is an autonomous institute of Ministry of Science and Technology, Government of India. It was established to boost research and development in the field of nanoscience and technology. And its aim is to generate the nanotechnology-based processes, technologies, devices directed toward the national priorities, especially in the sector of healthcare, agriculture, defense, and energy. So coming to the research, rheumatoid arthritis is a systemic autoimmune disease that primarily affects the joints. It not only damages the joints but also affects the other tissues like soft tissues and the other vital organs. According to a recent WHO report, 23 million people are affected with the RA globally. And in the India, it has high burden, 11 million people have rheumatoid arthritis, in which 75% are the women. So it takes the it affects the impacts the mortality of the patients. The mortality of the RA patient is 1.5 times higher than the general population. And the persistent inflammation, it causes the degradation of the cartilage, swollen inflamed synovial membrane, and the bone erosion, which cause the narrowing of the joint spaces. And this reduces the mobility and results the significant functional disability. So the common RA treatment includes the NSAIDs, DMARDs, biologics, and steroids. NSAIDs are helpful in reducing the pain and the inflammation, but it cannot be used for reducing the arthritis symptoms. And the DMARDs like methotrexate, sulfasalazine are the main first-line anti-rheumatic drugs. which are used for the treatment, which they act against the overactive immune response in the body. And the biologics includes the anti-pro-inflammatory and that inhibit the immune pathways in the body. And steroids are helpful in calming the flares. It can be used for reducing this inflammation in the synovial cavity, but they cannot be used for long term because of their side effects. So there are several challenges in the conventional drug delivery systems in the rheumatoid arthritis, like the poor joint targeting. If we deliver the drug systemically, then the very small fraction of the drug reaches to the synovial cavity. So and it causes the high systemic side effects that can affect the liver, kidney, and GIT. And most of the drugs, anti-rheumatic drugs, have the short half-lives. So it requires the repeated administration, and that causes the poor patient compliance. And in the most of the DMARD drugs, there is slow onset of action. Sometimes it takes weeks to months, and so the prolonged inflammation and comfort remain the same. And it causes the rapid clearance from the body. In spite of the intra-articular delivery, there is a rapid clearance from the intra-articular synovial membrane. And these conventional drug delivery systems have no response to the flares or the inflammation. The releases and the systemic dosing do not adjust with the fluctuating inflammation in the synovial cavity. So rheumatoid arthritis is a fluctuating disease which in which the patient have the different episodes of high flare or the inflammation and accompanied with the low activity periods. And our existing drug delivery systems release the drug continuously, and they are independent of the inflammation. So there are major two risks. That is, they can deliver the subtherapeutic level during the inflammation, and they can give the excess drug release during the remission or the slow period. So there is a need of the flare responsive, the inflammation responsive drug delivery system that can release the drug at the time of the inflammation and minimize the release during the remission periods. So due to these limitations of the conventional drug delivery systems, our formulation strategy is to develop the inflammation responsive dynamic moving microparticles that can sense the inflammation in the synovial cavity. Then they move towards the inflammation site and release the drug where there is more inflammation as compared to the normal condition. So our design concept for the inflammation responsive dynamic moving particles, the formulation particles that could move toward the inflammation and deliver the drug into deeper joint spaces and release the drug only when it is needed. So it includes three components: the drug-loaded microparticles that can release the drug on demand on the inflammation, and a catalyst that can propel toward the deeper tissues or where the inflammation is more. And this can be done using the several catalysts like manganese. So it can react with the hydrogen peroxide present in the inflammatory site, and it can give the propulsion by the generating the bubbles of the oxygen, and it give the targeted on-demand drug delivery. So the targeted, in our case, the targeted inflammatory markers present in the synovial cavity, we have targeted two biomarkers. One is the matrix metalloproteinase. This is the enzyme which is overexpressed in the synovial cavity in the case of the rheumatoid arthritis, and the hydrogen peroxide which is present in the inflammatory condition in the arthritis. And here we have used the JELMA microparticles which gradually degrades in the presence of the matrix metalloproteinases and release the drug accordingly. And here these JELMA microparticles were coated with the manganese catalyst. And when when there is inflammation, then there is an increased amount of the hydrogen peroxide in the synovial cavity, and this manganese react with the hydrogen peroxide and really give the oxygen bubbles that propels these particles to the deeper tissues. And when they reach to a deeper tissue, they find this matrix metalloprotein and they degrade and releases the drug. This is the basic concept of our study. So for this, we have designed this manganese-powered JELMA microparticles, and these were prepared by the this oil in water emulsion method, followed by the UV cross-linking in the presence of the photoinitiator under the controlled homogenization. After cross-linking, MnO2 was uniformly doped on the surface of the particle to impart the catalytic activity or to induce the propulsion. So these particles, the average size of these particles was 3.3 micron, and this electron microscopy image shows that they are spherical in shape, and the drug loading was around 21.6%. So the manganese deposition was validated by the EDEX method. This elemental analysis graph shows that this manganese is uniformly coated over the JELMA particles, and the quantitative analysis of the EDEX shows that there is an increase in the manganese content with the increase in the when we add the increased amount of the magnesium oxide to the system. So after that, we did the dynamic motion analysis of the manganese doped JELMA microparticles in the simulated synovial fluid using the single particle analysis using the fluorescence microscope equipped with a high-speed CCD camera. Here, we did this experiment in a specialized petri dish, which has two chambers. One chamber containing the synovial fluid, having the barrier, and other chamber we had hydrogen peroxide. And this hydrogen peroxide was supplied to the other chamber that contains the synovial fluid, and these microparticles were added to the synovial fluid. And when this H2O2 or hydrogen peroxide was supplied as an inflammatory biomarker, then there was a movement. We can see in the particle tracking videos, and this is the particle trajectory image. When there is no H2O2 in the system, then there is a minimal movement of these particles. But when we added this 5 millimolar of H2O2 or 10 millimolar of H2O2, there is a increase in the motion of these particles toward the inflammatory side. This is this indicates the inflammatory side where the higher concentration of H2O2 is there. And when this H2O2 comes inside this synovial fluid containing chamber, then there is a movement of the particles towards the higher inflammatory region. So we can conclude here that from this experiment, the active movement was observed in the presence of H2O2, while their motion was negligible when there was no H2O2 or there was no inflammation. So these particles move towards the inflammatory sites. So after that, we did this inflammatory responsive drug release study, in which we did in the normal condition as well as the inflammatory condition. Here the normal condition was the PBS, where there was a normal sustained release was observed, and there was no burst release, and there was diffusion mode release. But when we added this matrix metalloproteins, MMP2 and MMP9, there was increase in the drug release and because of the degradation of the matrix of the these microparticles by these enzymes. The gelma is the substrate of these matrix metalloproteinase enzymes. So the formulation remain stable during the remission, but accelerate the drug release during the inflammatory flares. Exactly the behavior needed for the precision therapy in the rheumatoid arthritis. So we also did the scratch assay and using the fibroblast-like cyanocytes. These were the activated fibroblast-like cyanocytes which were treated with these microparticles, and we found that the untreated control showed the normal growth in the of the cell membrane and the wound healing. But when we added this MN-doped matrix methotrexate containing gelma particles, then there was a reduction in the wound healing. So it means the formulation strongly inhibit the abnormal FLS migration. This is the essential driver for the synovial hyperplasia and the joint erosion in arthritis. By limiting this cell movement, it can help to protect the cartilage and reduce the structural damage and slow down the overall disease progression. So now coming to the therapeutic efficacy, here we did the therapeutic efficacy studies in the CFA immunized CFA arthritis model. Here we treated these arthritic rats using this by the intra-articular delivery of the microparticles, and we found there was high swelling in the case of the control or naive on the untreated animals. But when it was treated by the pure drug or with the dynamic particles, then there was a reduce in the edema and the swelling. And this formulation showed the strong dose-dependent therapeutic effect in the CFA arthritis model, restoring the paw morphology and significantly reducing the swelling. And X-ray and the morphological analysis confirmed the protection of the joint space and the bone structure. Now coming to the biodistribution and the joint retention studies using the animal imaging. Here we found that we injected these microparticles having this ICG dye. Here, actually it is not visible from here. There was a fluorescence of the dye till 42 days in the normal rats. And when we injected this formulation, our formulation in the arthritic rat, and this formulation fluorescent disappeared within the 28 days. Means there was a degradation of the formulation in the arthritic rat as compared to the normal rats. So in the normal rats, the fluorescent gone up to 42 days, while in the formulation having a formulation injected in the arthritic rat, it was up to 28 days. So in the histopathology and immunochemistry studies, we found that the formulation markedly restored the cartilage thickness, reduced the inflammatory infiltration, preserved the proteoglycan content, and prevented the joint destruction. And ISC immunohistochemistry further confirmed the strong restoration of the COL4A expression in the formulation, indicating the active cartilage repair in the system. So, and this formulation also decrease the inflammatory markers like TNF alpha, IL-6, MMP2 and MMP9 in the synovial homogenate or the synovial cavity and the serum. So it shows that these formulations reduce inflammatory markers locally as well as in the systemic circulation. So now coming to the conclusion of the study, that we developed a dynamic MnO2 GELMA microparticles that exhibited inflammation-triggered autonomous motion, showing rapid propulsion only in the presence of inflammation, and remain static under the physiological condition, normal physiological condition. And single particle tracking confirmed the clear chemotactic movement towards the higher inflammation conditions, mimicking the directional navigation towards the inflamed synovial region. MMP responsive GELMA degradation enables the synergistic enzyme-triggered drug release, ensuring the accelerated release, specifically under the RA-like enzymatic flare conditions. Overall, these dynamic moving particles represent the next generation intelligent therapeutic platform capable for targeting transport, on-demand release, and improve the management of rheumatoid arthritis. So I would like to thank my organization and my lab members for this immense effort in this study, and I would like to thank funding agency for their generous support, grant support. Thank you.
Thank you. Okay. We can have some questions from the audience. Okay, Dr. Rahul.
Yes, sir.
As you said that this is a system which is it, can we place it into a self-regulated type of delivery system?
Yeah, this is self-regulated.
On demand.
This is on demand actually. This is on demand means that when there is inflammation, then there will be release. If there is no inflammation there,
inflammation is not there, drug release will be stopped?
Not really. Not there will be lesser drug release. Lesser drug
Because
release.
in the case of the self-regulated system, it is dependent on the some triggering factor which increases the release, but as soon as that particular system decreases, say for example lowering of pH also, the release is stopped.
But here release doesn't stop. But there will be slower release and then higher release. In the normal or condition, there will be slower release, and in the inflammatory condition, there will be higher release.
And what is the degradation time of the GELMA?
Degradation time, like it was available until 42 days in the synovial cavity. 42 days. Yes, we have studied.
What's the method of the preparation of that?
The method of preparation. This microparticle I have given in the.
You can just tell the method.
Yeah. Yeah, we use this water in oil emulsion method with the UV cross-linking. Yeah, we use the cross-linker for that. Cross-linker, sir.
Thank you.
Okay, thank you, sir.
Question from
Yeah.
Yes,
Dr. Rahul, nice work. You've done animal study also, right?
Yes, sir.
It's always been a question of interest how we decide dose for all these nano drug delivery systems and how you have decided dose for your drug delivery system.
Sir, we decided on the basis of the drug loading. Actually, this is the drug loading basically, and we equilibrate this drug loading amount with the normal dose actually. So we have to give nearly 5% of this microparticles compared to drug in this case.
Okay, and do we have any guidelines for carrying out some studies related with these nano drug delivery systems in India? Because many scientists they are working on nano drug delivery systems.
Sir, it is in the actually these are in the formulating. These guidelines are being formulated by different agencies. Not really available right now. Thank you, sir.
Thank you, Dr. Rahul, for such a wonderful talk. Now I request session chairs to kindly facilitate Dr. Rahul with a token of gratitude. Now I request all the speakers to kindly on the dais to facilitate our chair. So please, all the speakers, if so, up here. Please give a round of applause. Dr. Yogesh Kulkarni. Thank you, sir. Thank you. Those participants, if anybody is interested in for tomorrow quiz, they can give their name at the registration counter. By today only?
If possible, today only.
Oh, by today only. the honor to I have the honor to distinguish Dr. Avijit Hazra. He is a professor of pharmacology in the Institute of Postgraduate and Medical Research, Kolkata, to gracious to please come on the dais. Welcome you, sir. Now I request sir to please start the session.
So, good afternoon, everybody. We are into symposium number 21 in IPSCON 2025. And we have 4 speakers, and I'm sure that you'll benefit by their presentation. So, are we ready to start? Can I call the first speaker? इसमें थोड़ा feedback ले.
Hello.
So it's my pleasure to invite and introduce the first speaker, Dr. Nitin Kothari. Nitin, can you come to the dais, please? Dr. Nitin is Professor of Pharmacology at Government Medical College, Dungarpur, Rajasthan. He is recognized for his contributions to public health pharmacology, particularly through his research on respiratory infections, as seen by his co-authorship of a number of clinical epidemiological profile studies on influenza A H1N1 variant in southern Rajasthan. He has also led research on pharmacovigilance and has publications on pharmacovigilance knowledge, attitude, and practices among nursing staff in a medical college hospital. Dr. Nitin is active in professional circles, serving on the editorial board of the Journal of Pharmacovigilance and Drug Safety, and he regularly participates in national and regional conferences on pharmacology. And today he'll be discussing antimicrobial resistance, which is a topic of renewed interest and concern. Through his dual role as an academician and clinician, he is bridging epidemiological research, drug safety, and medical education, particularly in the context of Rajasthan's healthcare system. So, Dr. Nitin, you can start.
Thank you, sir. Thank you for the nice introduction. At the outset, I first want to thank organizing team, Dr. Anil, for giving me opportunity to deliver a talk on this podium. My topic is National Action Plan for Antimicrobial Resistance, and in this session we will touch upon the few points, that is the NMC initiative, and NMC has released a document for prescriber last year only. As we have seen, every now and then the newspaper headline like that, resistant to last resort antibiotic growing in India, says ICMR report. The other newspaper headline was that 70% of antibiotic used not working on patient as per the expert panel. So with this background, we have witnessed the antibiotic resistance is now the silent pandemic nowadays. One pandemic we have seen in the previous years, the COVID-19 pandemic, and the coming year will be the antimicrobial resistance. And interestingly, the conference is going on from 20th to 22nd November, and in this 3 days, we are seeing this antimicrobial awareness week, which starts from the 18th November and will end on the 24th November. So with this background of the knowledge, I will start my session with these objectives. The objective of the sessions are antimicrobial resistance and its impact, global action plan to tackle the antimicrobial resistance, India's own action plan and role of National Medical Commission and prescriber module released by National Medical Commission last year only. First, we will see in the brief what is the antimicrobial resistant and its impact. Microorganism does not respond to an antimicrobial is called as antimicrobial resistance. It may be innate, in which the antimicrobial does not act upon from the beginning. For example, penicillin G is not active against gram-negative microorganism. This is the innate antimicrobial organism. Acquired is the antimicrobial was effective, but it is no longer effective against that organism. That acquired antimicrobial resistance is more dangerous as compared to the innate one. Because of this problem, trivial infection like sore throat, ear infection, urinary tract infection, and some more common infection in the endemic country like malaria and tuberculosis, they are becoming incurable and difficult to treat. So the longer treatment is required, hospital stay of the patient is increased, and it will increase the financial burden not only to the patient, but the whole of the nation is suffered. And because of the problem of AMR, higher chances of treatment failure and death may ensue. Antimicrobial resistance is directly responsible for 1.27 million deaths worldwide in 2019. It is the data of around 6-year-old, and it has contributed to around 4 million deaths. As per one estimation, death of 10 million people will occur in 2050 after 25 years, that means, and it will cost as much as US $100 trillion. So this is the impact of antimicrobial resistance. In the NMC document, the first chapter was dedicated to overview of antimicrobial resistance. What happened in the 2015? In the 2015, the 4 different bodies working at the international level, global level, they came together and they formulated antimicrobial resistance global action plan. These 4 bodies are World Health Organization, World Health Assembly, Food and Agriculture Organization, and the 4th body was World Organization of Animal Health. So you can see here, these 2 bodies are related to the human health, this is related to animal health, and this Food and Agriculture Organization is related to the use of antimicrobial or insecticide in crops. And this global action plan, they set the 5 core objectives. First objective was to improve the awareness and understanding about antimicrobial resistance. Second objective was to strengthen knowledge and evidence based. Third was to reduce the incidence of infection, that is the prophylactic measure, so the infection chance can be decreased, so the use of antimicrobial can be decreased. Fourth was to optimize use of antimicrobial medicine in human, animal and agriculture. So you can see here, the global action plan included the optimized use of antimicrobial medicine not only in human, but animal and agriculture were also included. And the last one was to develop economic cases for investment, diagnostic, vaccine and drug. So in the global action plan, the use of antimicrobial is reduced. On the other hand, the newer antimicrobial should be developed. It was included in its objective. And this was all done in 2015. What happened in 2017? WHO resolution urged its member state to align National Action Plan on antimicrobial resistance with GAP-MAR. What happened in India? In the same year, the core working group by Ministry of Health and Family Welfare, they devised National Action Plan on AMR involving constitution with various stakeholders, ministries and department at the national level. Their strategic objective of NAP-MAR are aligned with the Global Action Plan on national need and priorities. And they set up the 6 strategic priorities. And under each strategic priority were the specific objective was set up, and interventions what will be taken up, they were set up with the key outputs. So you can see here they set up these 6 objective or 6 different strategy, and the first 5 strategy are the same as the global action plan. You can see here the last one is unique to the national action plan. What is that? Strengthen India's leadership on antimicrobial resistance. So These are the 6 objectives set by the National Action Plan. What is the role of NMC? NMC is concerned with the objective number 1, this one, and objective number 4. First objective is to improve the awareness and understanding of antimicrobial resistance through effective communication, education, and training. This was the objective number 1 with the NMC is concerned. And NMC is equally concerned with the objective number 4. Optimize the use of antimicrobial agents in health of human only. NMC is not concerned with animal and food. So we will discuss the NMC role only in these two objectives, objective number 1 and 4. So later on, after the 2017 letter was circulated to 606 medical colleges that time, to dean, director or principal, and baseline data of antimicrobial resistance in their institute was gathered. They were asked to nominate a nodal officer, and out of the 606, 350 closed institute responded through Google form. Various online meetings of nodal officer were conducted. So we will now switch over the objective number or strategic priority 1 and 4. In strategic priority 1 was the improve awareness and understanding of antimicrobial resistance through effective education and training. This was the objective. That is the improve knowledge and capacity of key stakeholders regarding antimicrobial resistance and related topics by strengthening and consolidating AMR and related topics as core components of professional education and training. And target audience were all the stakeholders, including medical students, doctor, resident, faculty, medical officers, and others, allied health professional, including nurse, pharmacist, technician, and other allied health professional, and the administrators. So to obtain this objective, the different module, because the first objective was to improve awareness and understanding. So to achieve this target, this objective, the different modules have been developed or are in pipeline. This prescriber module was released by NMC in the May 2024, and the remaining two undergraduate module and the non-prescriber module are in pipeline. This is about strategy priority 4, optimize the use of antimicrobial agent in human health. NMC is not concerned with crop and animals. So objective was the improve knowledge and skill of prescriber, dispenser, and medical trainees. And intervention activity were included, developing structured and mandatory training programs on optimal antimicrobial use, and they collaborate with regulatory body to mandate periodic training and to optimize antibiotic use through pre-service and in-service training. So this is the front page, cover page of the National Action Plan on Antimicrobial Resistance. This is the module for prescriber that was released on 14 June 2024. This module was consisting of 175 pages. So it is not possible to cover all these 175 pages. We will touch upon this module, prescriber module says. This module was primarily for resident and faculty in medical colleges, but it can be used by any medical practitioner. Understanding of AMR and to importance in clinical practice and medical education has been stressed upon, and to promote rational prescription and antimicrobial stewardship. This document has been prepared in easy and understandable language, and in the last chapter of this module, PPT has been given, and this PPT can be used by a nodal officer to train medical professional in their institute. So total 9 chapters have been given in this module. First chapter was the overview of AMR, we have discussed already. Chapter number 2 is the clinical approach for prescribing antimicrobial, in which on the basis of history and signs symptoms, the disease is diagnosed and differential diagnosis is made. How it is made, it has been explained in this chapter. Chapter number 3 and chapter number 4, they are related with the diagnostic stewardship and mainly concerned with the microbiology department. Chapter number 3 is related to microbiological diagnostic stewardship, and chapter number 4, interpretation of antimicrobial sensitivity results. In chapter number 5, it is more like a theoretical one. It says about the antimicrobial resistance, principles and implication. Chapter number 6 and 7, they are related to more clinician. In chapter number 6, antimicrobial policy is stressed upon, and next chapter, antimicrobial stewardship in humans is discussed. Chapter number 8, in this chapter, how the use of antimicrobial can be discouraged with the use of infection control is given. In chapter number 9, there the PPTs are given in the form of that can be used by the nodal officer to educate their doctors. And it is named as prescriber toolkit. So chapter number 1, as we have discussed already, it has been mentioned on the chapter number 3 and PPT were given on the page number 59 to 67. We will switch over to chapter number 2, clinical approaches for prescribing antimicrobial. It has been described in these pages and these PPTs. And in this chapter, the medical officers, doctor are trained about to make the clinical diagnosis on the basis of history and clinical features. On the basis of history and clinical features, diagnosis is made after excluding the other possibilities with differential diagnosis. The clinician has to decide on the basis of history and clinical feature whether to start antimicrobial or not. Infective disorder can be bacterial, viral, fungal or parasitic. With the best possible reason, the empirical treatment is started. Common pattern of presentation of different diseases such as URTI, LRTI, urinary tract infection, meningitis, diarrhea, skin and soft tissue infection. These cases have been discussed in this chapter. So prescription of antimicrobial should be based on the following step. The step number 1 is making the clinical diagnosis based on the accurate history taking and thorough clinical examination. It helps in selecting the right test for the right patient. The sample must be collected before the start of antimicrobial. In step number 2, the empirical antibiotic therapy must be limited to seriously ill patient. This choice should be based upon institutional or local antibiogram. In the last step, choose the appropriate antibiotic that will be most focused on the narrow spectrum. It will be based on the clinical evaluation and most likely pathogen, keeping the antibiogram in mind. In the chapter number 3 and 4 are related to microbiology department, especially the chapter number 3 is microbiological stewardship, and this is described. Okay, sir. In the chapter number 3 is the content of this chapter is the definition of the diagnostic stewardship, and in this chapter sample collection techniques, precaution, how the sample is transported and when and how it is rejected, this common sample, it is explained. In the chapter number 4, antibiotic susceptibility testing, importance and interpretation has been explained in this chapter. One concept of antibiotic susceptibility is important, that is the cascade reporting. In report A, antibiotic sensitivity results of restricted antimicrobial or secondary agents only if the organism is resistant to the primary agent. If the microorganism or bacteria, you can say, is susceptible to the narrow spectrum antibiotic, the microbiologist should not report sensitivity to the broader spectrum. This principle should be followed. That is the concept and logic behind the cascade reporting. The chapter number 5 is related to antibiotic resistance principle and implication. In this chapter, that is the more theoretical, as I have already said. In this chapter, the antimicrobial and antibiotic difference has been explained with the driver of resistance, global epidemiological and clinical and economic impact have been explained. In chapter number 6, antimicrobial policy, how it is formed, what is its importance, it has been explained. This policy must define prophylaxis, empirical treatment and definitive therapy. The antimicrobial policy should be based upon the following factor. It should be on the prevalent local antibiogram. It will be given by the microbiology department. A spectrum of antimicrobial activity. You will have to consider the pharmacokinetic and pharmacodynamics of antimicrobial. You will have to consider the adverse effect and potential to select resistance of antimicrobial. And the last but not the least, you will have to consider the cost of therapy as well. This is the team to develop antibiotic policy. You can see here from the microbiologist, pharmacologist, physician, medical director, management members, and infection committee, all are involved to develop antibiotic policy. In the 7th chapter, antimicrobial stewardship in human has been discussed with these contents. You can see here
core and supplementary intervention we will touch upon. In the antibiotic stewardship, these are the core intervention has been taken, like prospective audit, antimicrobial timeout, antimicrobial consumption data is taken, formulary is restricted. Not all physician can prescribe all of the antimicrobial, that is the restriction of formulary. A standard treatment guideline has been developed on the hospital. These are the core intervention. Supplementary interventions are the dose optimization and combination therapy. You have to de-escalate the therapy after obtaining the antibiotic sensitivity result. You have to convert the parental antimicrobial to oral when the patient is suitable for that. And laboratory surveillance and feedback and information education and communication, these are all the supplemental intervention. The chapter number 8 is related to infection control, and the standard precaution is followed, and these are the elements of standard precaution, starting with the hand hygiene, hand wash and hand rub, personal protection equipment, safe injection practices, sharp management, spill management, patient care equipment, environmental control, that is linen, respiratory hygiene, and proper disposal of biomedical waste. These are the steps through which the infection can be controlled, and when the infection is controlled, the use of the antimicrobial can be discouraged, can be reduced. So in the last chapter, chapter number 8, the PPT of the that nodal officer to educate the medical officer, doctor and faculty member have been given. So this ends with this session. And so it is our responsibility to educate and to our level to doctors, medical faculty, and we have to save the antimicrobial to fall down and to save all of us from the silent pandemic. Thank you.
Thank you, Dr. Nitin, for giving us a broad overview of antimicrobial resistance and stewardship in the Indian context and the measures mandated by NMC. Since time is short, I'll allow only one question from the audience if it's there. Anyone? Okay then. Thank you, Dr. Nitin.
Thank you.
So may I now call upon the second speaker in this?
Excuse 1 minute, just we have to facilitate.
We can actually felicitate at the end. It will save some time.
You are here only?
I'm here.
So we'll felicitate at the end.
Okay.
So may I now call upon the second speaker for this session, Dr. Vivek Vidyadhar Bhosle. Dr. Bhosle is an MD pharmacologist serving as the senior principal scientist in the Department of Toxicology and Experimental Medicine at CDRI Lucknow. He has extensive experience in both preclinical and clinical pharmacology, overseeing research on novel therapeutic compounds. His group is involved in clinical trials for CDRI-developed molecules, including antimalarials, anti-osteoporotic, anti-thrombotic, and anti-hyperglycemic agents. He has several publications, both journal articles and books, with a current H-index of 17. His work supports translational drug discovery, bridging laboratory toxicology studies with first-in-human clinical evaluation, and helping drive the pipeline at CDRI's IND candidates. So over to you, Dr. Bhosle.
Thank you, Chairperson. So it is my pleasure to participate in this 55th annual conference of IPS. So today I will discuss about navigating the clinical trial approvals for phytopharmaceutical drugs, insights from the DCGI framework. So we know that there is a lot of research going on in pharmacology on plant medicines, and we know also that Ayurveda is a rich source of knowledge. And but if you go to any physician who is MBBS doctor or MD or DM, he says that, and if you ask any question about can I take this medicine, he says that no, I am not an Ayurvedic physician, you can't take this. So because he says, because there is nothing in his curriculum, as well as he cannot say, because he don't have any experience on that, he don't have any data. So NDCT, now Drug Controller General of India, is taking care of that, and they are saying that we will bring all these plant medicine into our regular medicines. So they have derived a new guidelines called phytopharmaceutical guidelines, in which the plant medicine will be considered as a drugs and will will be approved by the DCGI, so that all registered medical practitioner can now prescribe these medicines. So in this lecture, I will discuss about the regulatory definition of a phytopharmaceuticals. Then we will also discuss about some US botanical drug approvals and experiences, the successes got from these plant medicines. Then what is the current situation in India about the trials, and then CSR mission about these phytopharmaceutical drugs. And we will also discuss about some practical aspects, that how to deal with these applications and taking care of clinical trials. So definition of a phytopharmaceutical drugs is given in NDCT rule 2019. So as per the Drug and Cosmetic Act rule 110(2)E, the phytopharmaceutical should is has at least 4 biomarkers. So it is a purified and standardized fraction of a medicinal plant containing minimum 4 bioactive or phytochemical compounds meant for non-parental use, intended for human or animal use. So you can see that the first 2 letters, purified and standardized fraction, has been used. That means the crude extracts are not considered as a phytopharmaceuticals. So there should be some enrichment, it should be there, some enhancement in the fraction should be there. Second is that 4 bioactive or phytochemical compounds has been said. So if you see that in multiple in plant medicine, you can see multiple compounds, but you can also study all the compounds, but minimum 4 are required for standardization. So these 4 compounds should not be all bioactive, only 1 bioactive is necessary, but for at least they for analytical work, the 4 compounds should be there. Then we can also say that phytochemicals should be used for diagnosis, treatment, mitigation or prevention of disease or disorder. So it means that they are the drugs, now they can be classified as drugs, and they should have a defined composition. So the biomarker should be quantitatively and qualitatively assessed. So quantitative assessment means that if there are 4 compounds, we should know about their concentration, means how much percentage there. So if any plant medicine has a compound A, compound B, then the A should be 20%, B should be 30%, like that we should know the composition. So it is quantitative assessment. And qualitative assessment, we should know the structure of that compound, so that we can what is whether it is toxic or not, etc. Then it also, all these drugs should have must quality, safety and efficacy data like a new drug. So there are very few waivers given. All the toxic studies should be same as the new drug. And here the clear exclusion is there, that Ayurvedic, Siddha, Unani formulations are out of scope of phytopharmaceutical. If they are standardized, if there is a work, analytical work done, then only they can be converted into phytopharmaceutical. Second is that these phytopharmaceutical drugs should be for external and internal use only. The parenteral drug should not be cannot be considered as a phytopharmaceutical. So these phytopharmaceutical, though is came word used in country, but in US the botanical drugs are word is used, and they have a separate guideline for botanical drug approvals. Actually the US regulations are very stringent for impurities, etc., and Indians are little bit relaxed. But even though US has approved the botanicals first before the India, even because they are little bit innovative. So there are some examples that Veregan is a product available in their market, which is a green tea extract, and it has been approved for external genital and perineal warts, and it is the first botanical drug approved by US FDA in 2006. Another is a Mytesi, it is a Chromphalerum, that is a croton lechery extract, and it is approved for HIV related diarrhea, and it has demonstrated standardization complex plant extract. Another is Filsuvase, that is a birch tritepans. It is approved for epidermolysis bullosa wounds. You can see this is a cream, and Nexobrid is a Anacalyus bromelain extract, and it is approved for ischial removal in severe burns, and it is an enzymatic botanical with surgical precision. So you can see that botanical drugs are in the US market, and they're also doing well and taking a lot of profits. So key lesson for this, that if there is a rigorous quality control and batch-to-batch reproducibility and robust clinical evidence, then these we can get a regulator success even in US market also. So in India, what is the situation? So in India, the guidelines for phytopharmaceutical came in November 2015, and but there was a little confusion about, and then it was incorporated in 2019. So since then the work is going on phytopharmaceutical. So first success came for AQCH extract. It was a collaboration of IIM Jammu, CSR Institute and Sun Pharma. So Sun Pharma did the IND filing and did the clinical trial. So it is a Cocculus iristus standardized extract, and it was meant for COVID-19 management. And they did the phase 2 clinical trial, and it was found that there is a patient positive outcome, that it is demonstrated faster symptomatic recovery as well as there is enhanced viral clearances. It was a multi-center randomized double-blind trial. But the trial was successful. They approached the USA to the DCGI, but the DCGI, and they asked for the emergency approval, but they did not get emergency approval. They said that now you have to do phase 3 trial, but now COVID has been gone, so phase 3 trial could not be done, and so the drug is not marketed. But you can see they have 4 phytochemicals: cyanocolin, then magnoflorin, macisterone, and 20-hydroxydisone. So they filed the phytopharmaceutical application because they worked on these 4 phyto. If you read their patent, you will see that they have claimed that these 4 markers, even though they are in aqueous extract, but if you see the natural aqueous extract, the proportion as well as quantity is not equal to the whatever available in nature. They have enhanced it. So this drug has been enhanced, formulation has been enhanced. Then CSR is also working on multiple phytopharmaceutical product, and they have launched a mission for phytopharmaceutical drugs, in which there are multiple components. First is that there is sustainable cultivation. That is some institute like IIM Jammu has been led by the CSI CDRI. Then Moria Coinji is given by CSR ISB. Then Dioxolicium binefactorium is research is going at CSR IIM Jammu. Then Picoriza is by ISBT and as well as CDRI. Then Glycyrrhiza garba rich extract is for the CFR CMAP, and Cisampylus pyrea is for ISBT. Then Tinospora cordiphilia by CDRI, and Berginissa silista for IM. So these all are undergoing rigorous analytical procedure development as well as IND enabling studies. So we got success from this mission, and we got the IND approval. This is the first clinical trial approved by the DCGI for phase 3 trial, which is approved by the DCGI, and it is a multi-center, and we are doing as a hepatoprotic agent for the Picrolue as a phytopharmaceutical mode. So the Picrolue, which is available as a Ayurvedic medicine, will be available as a standard medicine, and it will be prescribed by the gastroenterologist, etc. So the trial is going on at 6 hospitals now, at all these hospitals, and it is a double-blind and parallel study. So 2:1 allocation ratio has been done. So this is the actual approval. Actually, we initially got in 2022, but as phytopharmaceutical has a lot of challenges, we have to manufacture the drug by captive cultivation. Picoriza is a endangered species declared, so we have to manufacture as a captive cultivation under the permission from the state government, etc. So it was done by IHBT, and that's why we could not initiate the trial immediately. Also, we have to give contract for GMP manufacturing by tendering process. It also took required. So ultimately, after a lapse of 2 years, we again filed IND, and we got in April 2024, we got final approval. So this trial is going at 6 hospital at AIIMS Delhi. Dr. Shalimar is taking care of. He has enrolled 46.
Dr. Bhosle, can you speed up a bit?
Okay, so IL-based Delhi has recruited 27. Then KGMU Lucknow has recruited 14. KM Mumbai has recruited 9. PJ Chandigarh at 15, and NIMS Hyderabad at 30. So all the leading practitioners you can see in the list are taking care of this trial. So we have registered this trial in the clinical trial registry, and also we have taken insurance from the National Insurance Company. So the trial is ongoing. So if anybody is interested, if you are suffering from fatty liver, etc., you can approach these investigators and can enroll the trial also. So I will now discuss about the regulatory pathways overview. So you have to do the first pre-IND work, and then you can also discuss with the DCGI. And now NDCT rule has a provision that you can directly go to the DCGI, file application, and discuss the pathway for the regulatory application. But it is many people are not doing because they are charging 5 lakh fees for that. But even though unofficially also you can approach, and have a consultation with the drug controller, so that there will not be a wastage of money as well as time. Then you have to prepare the CMC dossier, which has a comprehensive chemistry manufacture control documentation. Then you have to study the toxicity on GLP in GLP laboratories only, which has a GLP certification. The other toxicity studies are done at without GLP are not acceptable for the regulatory filing, and all the toxicity studies should be same as the synthetic drugs also. Then you have to file the application and then do CTRI registration, do phase 2 trial, and then you can do in so filing the dossiers, we have to give lot of data. So these are the we have to give data into form part 1 and part 2. In part 1 is that data which is already available, and part 2 which is data is generated by the applicant. So part 1 which has we know about the botanical identity, traditional literature, etc. we give, while in part 2 we have to give quality specification, ash, heavy metals, microbes, etc., and raw material authentication. The taxonomic certificate is also important, that taxonomy should sign the certificate that this is the actual species we are using. And these are the other requirement, that the data should be generated, and pharmacological studies as well GLP toxic studies are must for the phytopharmaceutical. So there are some common pitfalls, and we have some practical tips that we have already gone through these exercises. So we found that their raw material is very seasonal, so there is a lot of variation from batch to batch. But even though if you keep the raw material same, like we did the captive cultivation, and the raw material became the consistent, then standardization also are not doing properly, because the bioactive compounds are very small in a compound. There is a instrumentation issue, but even though you have to do it, then incomplete toxicity data and weak clinical design. So these are the some issues phytopharmaceutical drugs are facing. That's why the products are not coming to the market. But if you are engage with the DCJ early, you will get a clear path. Then you can also use the CSIR labs, etc., for analytical work, and you can also do meticulous documentation, and also you can for trial also you can engage CRO early. If you take this path, then there will be no stoppage. You can directly get approval for the phytopharmaceuticals. So there are various challenges that reproducibility is the most commonly faced challenges. Also IP there is, but phytopharmaceutical you can get a patent. So private companies are can get attracted due to the patent, because phytopharmaceuticals are not the traditional medicine, so you can get patent. Then the development cost is also high, because we have to do all that as a drug. So these are also challenges, but even though you can manage that, and but if you see the synthetic drugs are very few, and the chances of success are very few, but we have already biodiversity advantage, and the products are already showing efficacy. So there is a lot of opportunity to go into this space, and now the regulatory clarity is also there. In 2019 has clear path how to go ahead. There is no ambiguity left, and we can, with the help of phytopharmaceutical, we can go for Atmanirbhar Bharat. So this is last slide, that we have now clear regulatory path, and we can export these drugs also. We have institutional supports, and main thing for phytopharmaceutical is data, that it is the most that quality, comprehensive, safety, and convincing efficacy data is needed, and so that we can now blend our old knowledge with the modern knowledge, and we can bridge our ancient knowledge with the science, and which has bridges this tradition. So thank you. I hope that people will get some molecule into the market soon.
Thank you, Dr. Bhosle, for giving us an overview of this phytopharmaceutical development pathway, particularly in the context of CDRI, CCRI efforts in bringing this particular product, Victoriza, to the market. So, one or two quick questions. If there are no questions, thank you, Dr. Bhosle. Please be around. We'll do the felicitation at the end. So now I invite the next speaker in this session, Dr. Ashish Kakkar. Dr. Ashish is from the Department of Pharmacology at PGIMR, Chandigarh. He is active in both teaching and research, including a number of collaborative research studies under the ICMR Intent Scheme. His research expertise spans clinical trials and drug development, both from early phase development to later multi-center studies. He also has interest in infectious diseases, engaging in research related to safe and effective use of antimicrobials, neuropharmacology, and pharmacovigilance and drug safety. He is also active in the area of health policy and drug pricing, and he has written extensively on pharmaceutical price regulation and innovations. He has 42 publications as of date with over 1,000 citations. He has been elected as a fellow of the British Pharmacological Society in 2024 for his contributions to clinical pharmacology. He is active also other ways. He is on the editorial board of the peer review panel of multiple journals. He has supervised several students, research scholars in pharmacological research, both experimental and clinical. And his work on antimicrobial stewardship and optimization has contributed to curbing of antimicrobial resistance in the hospital setting. So, as a mentor and a teacher at PGIMR Chandigarh, he has fostered capacity building in pharmacology research with a focus on clinical pharmacology. Dr. Kakkar, over to you.
Thank you, Professor Hazra, sir, for that kind introduction. So today I'll be discussing about, mainly I'll be talking about neuroinflammation and what role does it have in various neurological disorders. So I'll be focusing mainly on the neurodegenerative disorders, and I'll see why it is not so straightforward to go around and suppress inflammation as we think it could be. So I'll be mainly focusing on what neuroinflammation is, whether it's our friend or whether it is an enemy that needs to be considered. What is its role? What evidence do we have with respect to various neurological disorders, primarily focusing on Alzheimer's disease, Parkinson's, and multiple sclerosis? And what are the current challenges in targeting neuroinflammation, and how can we possibly overcome these challenges? So as the name indicates, neuroinflammation is nothing. It's an inflammatory response that persists in the brain and the spinal cord. It's a response which is mediated by various cytokines, chemokines, reactive oxygen species. The cells which are involved can be the resident cells within the brain, which could be microglia and astrocytes. It could be endothelial cells, or it can be even the peripheral immune cells. So this question was posed about a decade ago in this famous editorial was published in Science Translational Medicine, where they asked whether neuroinflammation is a friend or a foe as far as Alzheimer's disease is concerned. So this reason was actually, till this time, it was thought that neuroinflammation needs to be suppressed so as to treat Alzheimer's disease. But at that time, there were two independent groups that were working, and they did studies with IL-10, which is an anti-inflammatory cytokine, and they thought that if they treat these animals who were models of Alzheimer's disease with IL-10, they should benefit. But what they found was, to their surprise, that actually it made matters worse, right? And they found that there is actually a complex relationship as far as the neuroinflammation and Alzheimer's disease is concerned, and we have to focus not just suppressing neuroinflammation, but there are other drivers which needs to be kept in mind as well. So neuroinflammation can have both positive as well as negative aspects, so as is indicated in this figure here. So if it's transient and it's low-grade inflammation, especially in the early stages of the injury, it's going to be beneficial, right? As we see, it can have various benefits. It can, so here we have microbes which lead to neuroinflammation, which leads to reorganization of host priorities and leads to immune surveillance following the infection. It can also aid in memory and learning with the help of IL-1 and IL-4 following traumatic brain injury. It can lead to tissue repair and also neuroprotection. But once it becomes chronic, and especially if it is high grade, for example, following traumatic brain injury, it can lead to collateral damage in other areas of the brain. In animal stress models, it has been shown to cause anxiety and depression. It has been shown to cause cognitive impairment, especially with aging and traumatic brain injury. And in the neurodegenerative disorders, it has been associated with neurological damage. So the effect of neuroinflammation will depend upon the context, it will depend upon the duration, and also it will depend upon the regulatory pathways which are existing along with when the neuroinflammation is happening. So these are the major neuroinflammatory disorders that we'll be talking about. So I'll be focusing in this talk just on 3 of them: the Alzheimer's disease, the Parkinson's disease, and the multiple sclerosis, and what are the differences in terms of neuroinflammation in these 3 disorders. Coming first to the Alzheimer's disease. So this is just epidemiological information for you that from 2019 to 2050, the numbers are going to be 3x people suffering from dementia, and the cost of management of dementia is going to double. Alzheimer's disease represents the most common form of dementia, responsible for approximately 70% of all the cases, approximately 10% of the people above ages of 60, and 1/3 of people above 85 will be diagnosed with this condition. In India, recent epidemiological studies have pegged this number as around 8%, making a total count of nearly 9 million Indians. And this was an interesting study which was published recently, which found which looked at the epidemiology of dementia in India, and what they found was that it's much more common in females. I'm sorry, you can't appreciate the colors here, but it's much more common in the upper curve represents the women. The lower one is for the men, so it's much more common in women than men. Also more common in people who have no education as compared to those who have primary and middle education. And this is the urban-rural divide, and it's also much more common in rural areas as compared to the urban areas. So those are some of the risk factors for dementia. And Alzheimer's disease, as you would know as students of pharmacology, they are characterized by abnormal accumulation of amyloid beta plaques and neurofibrillary tau tangles. So these are the major hypotheses of AD. So just like when we don't know an answer to a problem, we give 5 answers. In your exam, we have 5 hypotheses for AD because we don't have a definitive answer as to what is underlying AD. Right? So by far the most common ones is the amyloid cascade hypothesis, which is characterized by amyloid precursor protein leading to ultimately formation of fibrils and amyloid plaques. And to target this amyloid, various anti-amyloid antibodies have been developed. 3 of them have been recently approved. They goes by the name of aducanumab, lecanemab, and donanemab. So these have been approved for early stages of AD, typically mild cognitive impairment. Their benefit is debatable. It is modest at best, and they have adverse effects which can limit their use in these patients. So they are not very successful therapies, just like the existing anticholinergic therapies, anticholinesterase therapies that we have that aim at enhancing the acetylcholine levels in the brain. So this is the other hypothesis is the tau hypothesis, where there is hyperphosphorylation of tau in the brain, which leads to detachment of these tau proteins from microtubules, where they are normally associated with, and this leads to impaired neuronal function and cell death. And then we have the well-known cholinergic hypothesis, where there is degradation of cholinergic neurons, and there is reduction in acetylcholine levels, which leads to impairment of learning and memory. And these are the cholinesterase inhibitors, donepezil, rivastigmine, and galantamine, which have been used for several years for managing patients with Alzheimer's disease. Coming to finally the neuroinflammation hypothesis. So what happens in neuroinflammation hypothesis is there is accumulation of amyloid beta, which leads to activation of these microglia. So once the microglia get activated, they produce these chemokines, cytokines, and reactive oxygen species. And another important function of these amyloid beta activation of microglia is that microglia are involved in clearance of these amyloid beta plaques as well. So not only are microglia causing neuroinflammation, but they are also involved in the clearance of amyloid beta. So there is phagocytosis of amyloid beta plaques by microglia, which leads to microglial cell proliferation. And ultimately, however, when this process becomes chronic, there is burnout of microglia, and the process therefore goes on unabated. So that is why suppressing neuroinflammation will also lead to suppression of phagocytosis of amyloid beta by microglia. So that is why answering just by suppressing neuroinflammation is not going to solve our problem. So these are the various kinds of pathways which exist. So this is the physiological pathway by which the amyloid precursor protein is cleaved by alpha secretase and gamma secretase, and this is the amyloid digenic pathway where it gets cleaved by beta secretase, leading to formation of A beta fibrils and plaques. And this we have already discussed. Microglia, once they get activated, they release inflammatory cytokines, which leads to promotion of tau pathology as well as neurodegeneration. But at the same time, these microglia, they also engulf and they result in clearance of amyloid beta. And when there is microglial burnout because of neuroinflammation, it leads to reduced amyloid beta clearance. So what we did in one of our experimental studies is we tried to work on this hypothesis and see if we can inhibit neuroinflammation and what effect it has on the collection of features of cognitive decline in the animal model, and also what it leads to accumulation of amyloid beta. So we thought that would benefit by reducing neuroinflammation and will improve cognitive function in the animal models of Alzheimer's disease. But at the same time, there was a concern that there might be excessive accumulation of amyloid beta because the microglia were going to be inhibited. So what was the target was, so this interaction between microglia and amyloid beta is mediated through a number of receptors such as CD14, CD36, CD47, integrin and RAGE and toll-like receptors. And therefore, if you inhibit these receptors, you might suppress neuroinflammation, but at the same time, it will reduce your clearance of amyloid beta. So what we found in our experiment was there was improvement in the cognitive function and there was no increase in the accumulation of amyloid beta. So the results are under preparation and we'll be publishing it shortly. So these are the various drugs which have been approved for the management of Alzheimer's disease. I think we can skip that in the interest of time. So what is the clinical evidence? So we know that neuroinflammation is promoting Alzheimer's disease. So what are the epidemiological studies saying in people who use anti-inflammatory drugs? So do the use of anti-inflammatory drugs lead to improvement of cognitive functions in people who suffer from Alzheimer's disease, or is the long-term use of these NSAIDs associated with reduced occurrence of Alzheimer's disease, right? So there are not many studies which have supported this notion. So there is only one small clinical trial of indomethacin which is available, whereas a number of other NSAIDs, they have not shown much benefit in various clinical studies. So this was one of the studies which compared NSAID users with non-NSAID users, and they found that there was a significantly reduced relative risk among NSAID users as compared to the total cohort, right? But when they controlled it for various factors, this result was no longer significant. So there was only a minor benefit which could be appreciated in such conditions. So this was the trial I was talking about. So this was done with indomethacin. It was done long time ago in 1993. So they took patients with mild to moderate Alzheimer's disease who were treated with indomethacin for 6 months, and they found that people who were treated with indomethacin, their cognitive function slightly improved, whereas those who were treated with placebo, it declined significantly, leading to a significant difference between the two groups. But of course, it was a very small study, and therefore the results could not be translated to clinics just based on this trial. So a number of large trials have also been initiated after this study, but the results were largely negative, thus indicating that merely suppressing or globally suppressing inflammation is not going to work in these patients. And similarly, we also have epidemiological studies based on about 55 million patients where they looked at the effect of TNF-alpha blockers. And they found that in patients who have these inflammatory disorders, rheumatoid arthritis, ankylosing spondylitis, psoriasis, psoriatic arthritis, the incidence of dementia was much higher, thus indicating that an inflammatory phenotype is associated with higher risk of dementia. And in people who were using TNF-alpha blockers, this risk was much lower as compared to people or patients who were not using these drugs, thus indicating the protecting effect of these TNF-alpha blockers in these patients with these inflammatory disorders. So if we look at a number of 80 drugs in the pipeline, so there are as many as 25 drugs being currently evaluated, majority of them in being phase 1 and phase 2. And about 23% of the drugs are targeting inflammation, which are being developed for Alzheimer's disease. I think, sir, how much time do you have?
Not much, if you can speed up.
Yeah, okay. So I'll just take 2 to 3 more minutes. So that was about Alzheimer's disease. Similarly, in Parkinson's disease as well. So you will be, many of you would be aware that's majorly consisting of motor and non-motor symptoms. And there is progressive dopamine neuron loss as the disease progresses. And there is characteristically presence of Lewy bodies which move from rostral to caudal, from the caudal to the rostral direction, and they lead to worsening of symptoms of Parkinson's disease. And there is an increase in the inflammatory cells as well as inflammatory mediators as well in the brain as the disease progresses. And so there is also talk about the gut-brain axis being involved in Alzheimer's disease. So there are intestinal microbes, for example, one of which is being investigated, and also we did a trial on that was with Helicobacter pylori, which leads to production of pro-inflammatory cytokines which enter the circulation and then they promote the neuroinflammation in the brain. And similarly, currently we have mainly the symptomatic therapy, but we are trying to develop a disease-modifying therapy for PD as well. So these can be skipped because these are some of the well-known textbook examples of drugs which are being used for Parkinson's disease. And similarly, there are studies which have shown that anti-inflammatory drugs, particularly non-aspirin NSAIDs and ibuprofen, they have showed benefits in patients with Parkinson's disease. And the risk of Parkinson's disease was less in people who were using ibuprofen. So the future outlook, again, we have many anti-inflammatory therapies being developed for Parkinson's disease. Again, these are examples of some of the drugs. Sargamostrim is there, pentoxifylline is also being developed, and montelukast is also being evaluated in these patients. So this is one of the articles that we published where we looked at drug being repurposed for Parkinson's disease. So one were GLP-1 receptor agonists and DPP-4 inhibitors, which are classic anti-diabetic drugs, but they are also known to target neuroinflammation in the PD brain. And similarly, simvastatin is also known to have pro-inflammatory pleiotropic effects, and also it was being repurposed for patients with Parkinson's disease. Finally, coming to multiple sclerosis. So multiple sclerosis is a well-known neuroinflammatory disorders, and how this differs is that in contrast to Alzheimer's disease, where mainly the innate inflammatory system is, or the neuroinflammation is mainly because of the cells present within the central nervous system, the microglia and the astrocytes. Here we have the autoreactive peripheral B cells and T cells, which are getting reactivated within the brain and which are promoting activation of the resident cells, microglia, astrocytes, and ultimately leading to axonal dissection, and B cells also getting activated, and ultimately they lead to demyelination and oxidative stress and mitochondrial injury, ultimately leading to atrophy and degeneration as a ultimate result of neuroinflammation. So these are various drugs which have been approved for multiple sclerosis, and if you, in the interest of time, will not be covering them all, but if you see clearly, majority of them are targeting the proliferation of B cells and T cells, thus targeting neuroinflammation. So this is another one of the drug which is being repurposed, that is minocycline, which is an antibiotic of the class tetracycline, which has been shown to benefit these patients in some small studies, but yet we need more robust evidence. It has been shown to suppress the migratory inflammatory cells, which from migrating from the periphery to the central nervous system, also modulation of the peripheral immune response and inhibition of microglial activation in the CNS. So finally, coming to what are the challenges in targeting neuroinflammation. So you know that suppressing neuroinflammation is not a straightforward thing, which we can consider for many of these neurodegenerative disorders. So one is mechanical complexity. So the microglia can have two states, M1 state which is pro-inflammatory and M2 state which is anti-inflammatory. So widespread suppression of inflammation will need to be done carefully because we don't want to suppress the anti-inflammatory states. Also, drug delivery across the blood-brain barrier has been a challenge, and also if you give them systemically, there are going to be adverse effects of these agents, such as NSAIDs that we saw at number of epidemiological studies. Of course, there is heterogeneity of inflammation, early versus late stages. There can be variability in individual responses. There are safety concerns with long-term immunosuppression as far as cancer and infections are concerned. How do we monitor efficacy and safety of these agents? We need to have some robust biomarkers for that. Of course, there are translational challenges of what may work in animal models, may not prove beneficial in the clinics. And finally, what is our window of opportunity? When should we actually intervene? So we know early stages it is beneficial, but in the later stages it will be detrimental, neuroinflammation. So what is our window of opportunity of when to act? So finally, we can have targeted therapies for specific pathways of neuroinflammation. We can have advanced drug delivery system for blood-brain barrier. We need biomarkers which can help us in assessing the response to these therapies. We can have combination of anti-inflammatory, which are symptomatic, and disease-modifying therapies. We need to do clinical studies to find our window of opportunity. And also, importantly, we need to have more population from our settings, from lower middle income countries in these studies for various therapies which target neuroinflammation. So thank you for your valuable time.
So thank you, Dr. Ashish. You started off with the question, neuroinflammation, friend or foe.
Right, sir.
And so far as these 3 neurodegenerative disorders are concerned, we have established that there are probably foes that we need to target with suitable drugs.
Yeah.
And it would be interesting to see many of the drugs which are being repurposed, if they actually enter in clinical practice as standard treatment.
Yeah.
I hope the future will bring us closer to some
More
effective
options,
remedies.
yes.
So thank you.
Thank you, sir.
So we move over to Dr. Bushra Parveen. She'd be the winding up speaker for this symposium. Dr. Parveen is Assistant Professor in the Department of Pharmacology at the Center of Excellence in Unani Medicine, Pharmacognosy and Pharmacology, School of Pharmaceutical Education and Research, Jamia Hamdard, New Delhi. She holds a PhD in this discipline and has been contributing actively to both teaching and research in Unani pharmacology. Her work aligns with her center's focus on integrating traditional Unani medicine with modern scientific methodologies, and she is listed as part of the scientific team in the Bioactive Natural Product Laboratory. Dr. Parveen has also been playing a leadership role in academic events. She has served on the editorial team for conference abstract books at the Center of Excellence and has evidence of engagement in scholarly and organizational activities. So over to you, Dr. Parveen.
Thank you so much, sir. Good evening, all of you. So I would be talking about reverse pharmacology today. So I'll begin with how reverse pharmacology differs from conventional drug discovery approach, and then what is the scope of herbal drug development, and then the future possibilities in the reverse pharmacology approach. So we see that the conventional drug discovery process, traditionally it followed a linear and laboratory-driven pathway where potential molecules, they are identified, they are screened, they are tested into several phases like preclinical and then several clinical phases, and then they are translated for the clinical use drug. And this conventional drug discovery pathway traditionally has landed up into discovery of several breakthrough therapies over the years of decades, but yet it comes with several shortcomings, or you can say limitations, as it involves a lot of investment and a lengthy time schedule that leads from discovery to development of the drug. Another important challenge that is associated with this conventional drug discovery process is the high rate of attrition. So you can see that as we move from preclinical to phase 1 and then to phase 2, you can see the success rate, it drops preferentially. Especially the bottleneck appears to be phase 2, where we see 17% success rate only. And this is the phase basically where the drug is first time actually tested in the target population for its efficacy and safety. So we see when the drug, it fails over this later stage, when it has passed 2 to 3 years in the preclinical phase and 1 to 2 years in phase 1, and then in phase 2, and then it fails in phase 2, then it it brings about a loss of resources, investment, as well as cost that is associated. And it is not only the scientific barrier of the drug discovery, but also an economic barrier as well. So over this approach, what reverse pharmacology differs from it, you see reverse pharmacology is the reverse of the conventional drug discovery process, because it begins with the molecules that have been clinically used in humans. in the Ayush system, or you can say traditional system of medicine. So the reverse pharmacology approach, it begins with the human evidence, and then it translates them. It is then tested and verified and validated. That scientific claim, you can say therapeutic claim, basically, it is validated and supported by the scientific proofs in the reverse pharmacology pathway. So it is a flip model, and this process, it gives, it reduces the time cost, it reduces the time, it reduces the cost, and also there is high chances of clinical success, because we are using the molecules that are being traditionally used in humans. So the risk of hit and screen phase is almost nil in case of reverse pharmacology approach as compared to the conventional drug discovery process. Now we see that the reverse pharmacology process is not new, but it was pioneered in India, with the key figures being Sir Ramnath Chopra and Ganathan Sen, who began to investigate the medicinal plant role for the therapeutic use. And first of this claim was known to be rezapine, that was developed from Rauwolfia serpentina, and it was used for antihypertensive and tranquilizing effect. Later on, several drugs they came into market, like artemisinin, then gugolipid, that was used as a lipid-lowering agent. And now in the 20th decade, reverse pharmacology has integrated well with the state-of-art facilities for developing the drugs that aligns with the network pharmacology, system biology, as well as integration with omics and AI tools. And you see, when we talk of reverse pharmacology, then we talk of the plant sources that are being used in development of the drug candidate through reverse pharmacology approach. So this plant source, it can land up into multiple products depending upon the type of the country involved and the type of the regulatory body that is involved. So a plant source can either become a traditional herbal medicine product if it is followed by the European guidelines, that is EMA in European Union states, or it may come out to be a dietary supplement and it is regulated and approved by FDA and FSSAI, while it can also be a phytopharmaceutical which requires rigorous standardization as well as clinical trial and testing, and it is regulated by CDSCO. Then it can also land up to the classical Ayurvedic formulation and proprietary medicines that covers and under the purview of the Ministry of Ayush under National Ayush Mission. So these are the several possibilities that a herbal drug can become into a regulated product. And while the reverse pharmacology approach has been in place in the last decade, the government of India has also supported well for the development of herbal drug market and herbal medicine as such. And these are several government initiatives that have been undertaken, like, for example, Advanced Center for Reverse Pharmacology was established by Indian Council of Medical Research. Then several, a center of research was established at University of Mississippi, USA, that was called the Indo-US Center for Research in Indian System of Medicine, that basically focuses on scientific validation and dissemination of Ayush product to meet the global meat demand. Then also there was translation of the Ayurvedic as well as the traditional text into traditional knowledge digital libraries, that is developing the e-copies of the Ayurvedic and the traditional text, and also development of WHO Global Center for Traditional Medicine that is situated at Jamnagar, India, and it is also serving the encouraging innovative approaches in drug discovery, including the reverse pharmacology. Several other initiatives that have been taken by Government of India, namely under Ayush Swasth Yojana, several centers of excellence were set up in different parts of the nation, like Center of Excellence of Unani Medicine in Pharmacognosy and Pharmacology, from where I belong, that is situated at Jamia Hamdard. Similarly, there is also a Center of Excellence for Ayurvedic Medicine as well. And apart from that, several MOUs have been also signed for undertaking joint research in Ayush system of medicine. And also academic Ayush chairs have also been set up at various foreign institutes and academic institutes in the worldwide, so for the promotion of traditional medicine and herbal research. So on account of these government initiatives, how it has reflected in the numbers, we see that these government initiatives and these concreted efforts have resulted into upsurge of global herbal medicine market. And you can see over the globe that There's a steady growth reported in herbal and plant-based therapeutics that is supported by aging population and interest in complementary and integrative medicine. And the dominant area remains Asia Pacific, where you can see the market share is about 41% and with a growth rate of about 10.2%. And this is because of the increasing regulatory acceptance of standardized herbal products and evidence from clinical and reverse pharmacology approaches. So that is why this segment, this sector is, is moving closer to the mainstream. And the projections, they indicate that the global herbal medicine market could surpass $125 billion by 2030. And India is, would be taking the center stage in this growth rate. And if you see the India's herbal drug market, that is one of the fastest growing herbal or Ayush market that is driven by a rising preference for natural therapies and chronic disease management specifically. So the Indian herbal drug market size was valued at about $2.6 billion in 2024, you can see, and it is estimated to grow to $5.3 billion by 2030, and with a growth rate of about 11.2%. And this growth rate can be attributed to increasing consumer preference for natural and plant-based therapeutics, including and supported by India's rich biodiversity and long-standing Ayurvedic and herbal medicine tradition that we have. Also, if we see the number of publications in the area of research pharmacology over the years, then we can see there is an upsurge. From 2007, there were only 53 publications on reverse pharmacology approaches, and which have increased to more than 1,000 in 2025. So you can see this, all these government initiatives and the growth in herbal drug market has been in pace and it's aligned with increased publications that have been reported in several scientific databases as well. So here are some of the glimpses. I would not go into the detail of the studies owing to the paucity of the time that we have. So here are some of the glimpses of the studies that have been taken at our research center. For example, we have initiated development of phytopharmaceuticals and synergy-based combination of Punarnava, that is Bohria diffusa, and Tinospora cardifolia for chronic disease, that is a chronic kidney disease, basically using in vitro, in vivo, and metabolomic approaches. So we landed up developing a synergy-based combination that was helpful in elevating CKD by exerting immunomodulation, antioxidant, and renoprotective effects against diclofenac-induced nephrotoxicity. This was another study that included extracts of Punica granitum and Piper cubeba. So we developed a phytopharmaceutical also using this, using a synergy-based approach. And similarly, that was also directed against chronic kidney disease management. Now, with all these efforts and owing to the, aligning with the theme of the conference, that is empowering the young minds. So the young minds that are sitting here, this slide is for you all, that what is the future scope of reverse pharmacology and how you can contribute towards it. So a lot has been done in this area, but still needs to be done. So what we can do is we can integrate the omic technology with with developing the precision herbotherapy, that is precision phytotherapy. So precision or personalized concept has been there in the herbal medicine from quite long, and owing to the consideration of the doshas in Ayurvedic medicine and misaj or temperament therapy, or the modality that they consider while advising the treatment. That is the misaj or temperament of the patient, as well as considering the temperament of the drug as well. So the need of the hour is that now we need to quantify and integrate those doshas and misaj temperament with the gene bank and with genomics, and we need to come with a standard sequence of gene sequencing sort of things, so that we can correlate them with the state-of-art technologies in present world, and then that can be widely accepted globally and nationally. Another area that needs to be addressed is the herbal drug safety. We talk a lot about that herbal drugs are quite safer and they are with less side effects. It is there, but we need evidence against that. So there is a need to develop a proper pathway and network of herbovigilance system. Just like we have pharmacovigilance of the modern drugs, similarly there is a need to develop, although there is a herbovigilance system existing, but there is need to strengthen that system more, so that we can document the safety and or maybe the toxicity or the adverse effect that are associated with these herbal drugs. Apart from that, there is also a need for reformulation and dose metering in herbal drug medicine. So, for example, some of the herbal, traditional medicines, they are sugar-based. So there is a need to develop sugar-free, formulations and dosage forms of such drugs so that it is majorly acceptable for, everybody. Those who cannot take sugar-based things, so it is widely accepted to everybody over the globe. So with this, I would like to conclude that reverse pharmacology, it converts centuries of traditional and folk wisdom into validated modern medicines. And through reverse pharmacology approaches, validated herbal drugs that are developed can open global markets, can attract investment and increase export potential. And this, as a result, would strengthen India's leadership in traditional medicine and would transform cultural heritage into scientific and economic assets. And this would support the vision of Atmanirbhar Bharat, Made in India campaign, and would also help to resonate and achieve the vision of Viksit Bharat, as is the theme of the conference too. Thank you.
So thank you, Dr. Parveen, for giving us an overview of the potential and challenges of reverse pharmacology, particularly in the context of traditional Indian medicine. So I'll request all the 4 speakers of this symposium, Dr. Nitin, Dr. Ashish, Dr. Bhosle, and Dr. Parveen is already here, to come to the stage, and I'll hand over the mic to the organizers.
Thank you to all the speakers for a wonderful talk. Now, I request session chair to kindly facilitate all the speakers. Dr. Nitin, give a round of applause. Then Dr. Vivek. Please give a round of applause. Dr. Ashish. Dr. Ashish. Dr. Bushra. Now I request all the speakers to kindly facilitate our chairperson also for the session. Thank you, sir. Kindly give a round of applause. our next session of Symposia 25. I request, I have the honor of inviting Dr. Sukant Garg for the session of chair to kindly ascend the dais. Dr. Sukant Garg. Before starting the session, I would like to introduce our chair for the session. Dr. Sukant Garg is Associate Professor in the General Pathology Department of the Dr. Harvansh Singh Judge Institute of Dental Sciences and Hospital, Punjab University, Chandigarh. He is a part of faculty of a premier dental institute which integrate basal medical sciences and dental educations to promote both oral healthcare and research. Dr. Garg contributes to the foundational pathology education of dental students, bridging essential clinical and theoretical knowledge. In his capacity as a faculty member, he is connected to the broader mission of the institute, which not only provides high quality dental education but also carries out clinical research, community outreach and diagnostic. So, sir, I welcome you as a chair. Thank you, sir. Now I request chair to kindly introduce the speakers.
Good evening, everyone. Thank you, organizers, for providing me this opportunity. Our first speaker for this session is Dr. C.K. Kishan. He is a professional associated with Lela Nutraceuticals, Vijayawada, a prominent company specializing in life sciences, nutraceuticals, drug discovery, innovation, and manufacturing. With a focus on advancing health and wellness through scientifically backed natural products, Dr. Kishan contributes to the research and development efforts in pharmacology and nutraceutical sciences. His work supports the company's mission to create innovative health solutions, integrating traditional knowledge with modern scientific approaches to enhance the efficacy and safety of nutraceutical products. Dr. Kishan plays a key role in bridging pharmacological research with commercial product development in the nutraceutical sector at this Lala Nutraceuticals in Vijayawada. I welcome you, sir, Dr. C.K. Kishan.
Thank you so much, sir, for nice introduction. And thanks the organizers of IPSCON and the Punjab University for hosting me. And dear students, good evening. So today's my topic is Next Gen Pharmacology, Shaping the Future of Vikasit Bharat Through Young Innovators. So I'll be touching upon what I meant by Next Gen Pharmacology, what is Vikasit Bharat, and what is innovation, and some case studies, and how you people, young innovators, can drive it, and I'll conclude. So what is Next Gen Pharmacology? So a lot of recent techniques that have influenced the discovery and development of new drug molecules, that some of them I'll try to give a glimpse of them. Some advances like pharmacogenomics, AI and ML in drug discovery, organ-on-chip technology, 3D cell culture and bioprinting, CRISPR-Cas9 gene editing, digital therapeutics and mobile health, optogenetics in neuropharmacology, and so on. There are so many, but I'll try to give a glimpse of some of them. So briefly, what is pharmacogenomics? So for this, I would like to ask the students, do you know warfarin? So what is warfarin? It's an anticoagulant, right? So it is vitamin K antagonist, as we study in B-Pharm and all, right? So vitamin K, once it gets oxidized, it becomes inactive, and there is an enzyme in our body which is called vitamin K epoxide reductase, which regenerates vitamin K. So if a person is having VKRC1 mutation or something like that, so then to them warfarin, originally discovered as rat poison, but then based on the mechanism it became anticoagulant. Now in those people it might as well work like a rat poison. So you need to look into those genetic polymorphisms whatsoever, and accordingly you need to adjust the dose. So study of how genes affect the drug response is pharmacogenomics briefly, and warfarin is one example. And another example is you also might be knowing the drug by name trastuzumab, which is a breast cancer medication, and it is given to people diagnosed with breast cancer and overexpression of HER2 receptor. Okay, so we have a case where we are looking at the genes of a patient, whether our drug should be reduced in dose or not with respect to toxicity, and in another case we are seeing a patient's gene pool wherein if there is an overexpression of a particular gene, we are prescribing a particular drug. Moving on to next, AI and ML. So artificial intelligence and machine learning, ये तो आपका ज़माना का है, तो आप ही ज़्यादा जानते हैं, मैं थोड़ा बहुत बताते रहता हूँ. और AI is artificial intelligence and ML is machine learning. Simply put, whatever Alexa, Siri and all, they are AI. And ML, whenever you see those are YouTube videos, the next day the YouTube will recommend those videos which you might like. So the machine is learning what you like and what you don't like, and accordingly it is giving the videos to you. So that is the difference between AI and ML. And so how many of you know Lipinski's rule of 5? Anybody? अच्छा, okay, बताइए. Okay. Okay, so that is the implications of it, but actual rule of 5 is actually 4. Yeah, bolo. Yeah, okay, what are those 4? Log P and molecular weight. Very good. I owe you a chocolate. I'll give it in the end of the meeting. Okay, so Lipinski's rule of 5, what he did is he looked at all the drugs that were successful in the market, and he looked at the characteristics of each of them, pooled them, and brought out this hypothesis, right? So based on this hypothesis, we will, by looking at any new molecule, we will come to know whether it is bioavailable, whether it has a possibility of being successful in clinical trials and come into the market. Same thing if AI does it. Okay, that's what was done by benevolent AI, which looked into set of molecules, set of drugs that are useful against COVID-19, and it has found out baricitinib. Baricitinib is a JAK1 and JAK2 inhibitor, which is found to be useful in COVID-19 patients, and it predicted it will be useful, and later it was found to be useful. Moving on to next technique, organ on chip and 3D cell culture, both are almost similar. So wherein now our drug discovery path goes from hit to lead, and also in vitro test will be done, followed by in vivo test. There are some drugs the molecules that work in vitro test, but they might fail in vivo test. The major barrier is because in vitro test we are looking at 2D picture, a single layered cell wall and well, and also a drug might work there, but it may not work in the animal. There are other factors also. So if we can somehow make the in vitro test as equivalent to in vivo test, then that is what is organ on chip technology. For now, lung on chip is used for discovering or screening anti-asthmatic molecules. Then CRISPR and Cas9 gene editing and digital therapeutics. CRISPR and Cas9 is like in 90s, around 90s and all, 80s and 90s, there is a discovery of PCR, polymerase chain reaction, which is considered as the discovery of the century. And with that scale, CRISPR-Cas9 also can be considered as discovery of this century, 21st century, because you can pinpointly edit genes, and accordingly you can design the disease models so that a lot of time will be saved with respect to money and animals and investment. So example of it is like people are generating APP and PSEN1 knocked out mice for treatment of Alzheimer's disease. And then digital therapeutics. So for treatment of disease, you have a lot of ways of drugs like synthetic molecules, herbals, Unani, all these things. So for the treatment of drug, if the physician prescribes you a video game or an app. Okay, so that is digital therapeutics. Example is Reset app. It is a FDA approved app for substance use disorder. So then optogenetics in neuropharmacology. So where you will try to interfere with light for different outcomes, and light activated dopamine release for study of addiction pathways is one of the applications of it. And others like high throughput screening and all you must be knowing already. So now I move on to my next part of my presentation, that is Viksit Bharat. Actually the government aimed at Viksit Bharat by 2047. Hopefully we'll look into it more positively on or before 2047. So current status for India with respect to clinical research, we are top 3 global destinations for clinical research. That is good, behind only to America and China. And regulatory reforms, the government is proactively putting all these reforms so that the approval time and all gets reduced as much. And in the last year we have 18,000 clinical trials, which is very good. And we have also developed indigenous therapies like NextCare-19 for Hodgkin's lymphoma and other blood cancers, and Zyco-D, the first DNA-based vaccine indigenously developed for COVID. So looks like sub-Changa, right? Then we look into preclinical and biotech divisions. So here, as one of my predecessors suggested, like CAGR, that is compounded annual growth rate of 11.4%. So any business with a compounded growth rate of 5 to 8% is considered as good business. So it is currently a very good business, preclinical research, and we have added 60 new GLP facilities in the past 10 years, and our biotech business has boomed from 10 billion to 165 billion in the past decade, and we have startups growing from 50 to 10,000 in the past decade. Again, sub-Changa, right? But still, there is unmet medical need where, so out of every year, number of snake bite deaths are around 1 lakh, out of which close to 60,000 are from India. So India is worst affected with respect to snake bite. Then, so India every day faces 10,000 dog bites and around 5,700 rabies deaths. This is again 1/3 of global deaths annually, more than 1/3. And India has highest TB burden in the world. So I remember now old movie called Border, in which a Fauzi receives a letter from his Dharmapatni. उसमें ऐसा लिखा होता है कि this disaster happened, बाकी सब ठीक है. This disaster happened, बाकी सब ठीक है. This disaster happened, बाकी सब ठीक है. So till now whatever we have seen is sub-Changa. So बाकी sub-Changa, right? So moving on to what we should do from now on. Whatever we have seen the positives and we have seen the negatives of whatever is happening in our country. Now, there are 3 terms that I want you to understand. One is discoverer, innovator, and entrepreneur. So discoverer, by bookish translation, is someone who has discovered something that is already there in the world. An innovator is he has discovered something which is not there in the world previously. So one funny way of understanding it is, there is a saying, there is a sentence, my father discovered my mother and invented me. Okay, so that is the difference between discoverer and innovator. And so either by discovery or innovation, you have brought something new into the world, which if that idea is put into business, that is entrepreneur. Okay, and of course successful. So with that aspect, I want the students to be proactive in recognizing these people. You will be handsomely rewarded. So can anyone recognize this person? He is an eminent person which you should be knowing. No. Okay. Second person, third person, and fourth person. Okay. Right. Now, fifth, sixth, seventh. Subject knowledge ज़्यादा होकर local knowledge पूरा हो गया. Anyway, so I'll be explaining because whether you know it or not, you should be remembering these people as they will inspire you in your career. So first eminent person, he is Sri Prafulla Chandra Ray. Okay, he is the Indian chemist, and he is considered as father of Indian chemistry, and he is the first entrepreneur. Means he is the first person to establish a pharmaceutical lab in India. Bengal had chemicals and pharmaceuticals. And of course, as like a scientist, he has a lot of publications as well. So he is a scientist as well as entrepreneur. Then Rai Bahadur Sir Upendranath Brahmachari. Okay, so he is again a physician and a scientist. He discovered dermal leishmaniasis. You must have studied about it in the intermediate. Kala azar, Leishmania donovani, and Delhi boils are oriental source, Leishmania tropica. These are two microorganisms which cause these diseases. He has identified that organism, and he has also identified that cure to it, carbostilbamate. Okay, he was nominated for Nobel Prize for close to 7 times, but because of various other reasons, he couldn't get one. He is also scientist, but not an entrepreneur. Then this person is Dr. Yellapragada Subbarao. He is a physician and a scientist, and he discovered folic acid, the role of folic acid, methotrexate, which is a folic acid antimetabolite used as anti-cancer drug, diethyl carbamazine citrate, also called as heterozan, and famous class of antibiotics called tetracyclines. So he also discovered the role of phosphocreatinine and ATP biochemical pathway along with other scientists, not alone. And he also got nominated for 4 times but couldn't get one. But the Nobel Fellows, they felt bad that this guy didn't get a Nobel Prize, and they kept his portrait in the Karolinska Institute. So he is a scientist but not an entrepreneur. But I think this you should be knowing. One journalist, while writing his obituary, you have probably never heard of Dr. Yellapragada Subbarao, yet because he lived, you may be alive and well today, and because he lived, you may live longer. That's the obituary that was given to him. Okay? Now, eminent person 4, she is actually a student of the first person, Prafulla Chandra Ray. Okay? Asima Chatterjee, she is the first DSc from India, organic chemist, and she invented Ayush 56, means that time there is no synthetic chemistry in India, so using a lot of plants, she made a polyherbal formulation, Ayush 56 for epilepsy and Ayush 64 for malaria, and she also isolated coumarin glycosides from bale fruit, and she is the first female recipient of Shanti Swarup Bhatnagar Award in 1961, Padma Bhushan, Rajya Sabha MP and all. She is also scientist but not an entrepreneur. We have seen 4 scientists, out of which only one was entrepreneur. Rest all are scientists only. Now we are looking, so this is the Google Doodle for her 100th anniversary. Now, he is Dr. Saket Barman. You all must be knowing Dabur, right? So, he is the founder of Dabur. And he is also a scientist as well as entrepreneur. What he did is, whatever herbal products that were compounded in a local way, he made it into a bigger way, corporate way. Okay, that's how Dabur was found and is successful. So second entrepreneur, Kala Manjireddy. You all must be knowing the Reddy's Labs. He is the founder of it. And what he did was, he actually has worked as a scientist in IDPL. so he could have had his normal life, but he has seen some sufferings in the people where the affordable medicines is a big problem that time in India. So initially he started with the idea of getting quality medicines at affordable rates, and accordingly he succeeded, and because of his vision, today this one person's vision translated into a 28,000 crore empire and with close to 25,000 employees. So he's a successful scientist as well as entrepreneur. Last but never the least, he is a B Pharmacy graduate, like many of you, right? And he worked for a brief time in a small laboratory in Vijayawada, and then he founded Laila Nutraceuticals some 50 years ago. 50 years ago, imagine, nobody would have thought of the word nutraceutical, right? So that time itself, he standardized, civilized the plant extracts, different compositions and all, and made it into a successful company. So he's also a scientist as well as entrepreneur. From all these people, what can we learn? So they took some decisions, they thought differently and acted on their ideas, which they contributed to their families, their communities, as well as overall India, development of India. So now we have two choices, right? Whether to innovate or whether to discover. Innovation is one thing that we know, like synthetic chemistry, all these molecules, monoclonal antibodies and all. And in discovery is where we go back to our roots, the roots of some plants or stems, all those things, and we'll work on it. Why? Why I put this discovery much more? Yeah. Okay. Yeah. So these are some of the books. Recently I came across a book which wrote about the last books of Ayurveda in my local language, that is Telugu. Okay. So, and you all must be knowing a movie called Aparachit, sometime some 20 years ago, right? Where they mention about Garuda Purana. Garuda Purana is, as we know, is whatever sins we do and whatever punishments they are. That's what we know. But in that Purana also, there is mention of a lot of herbs. Okay? And there are, these are all Google books only. Okay? There are a lot of books that we don't know, but they have a lot of knowledge. So, what we can do for Vikasit Bharat, because you are the future, and either you can discover or innovate, and if not by your own, you can work in those companies which discover or innovate, and if you have financial muscle and wherewithal to think as a leader, you can become an entrepreneur as well, but always with a philanthropic attitude, and together with this you can achieve the goal of Vikasit Bharat. So I'll conclude here, and you have any questions, I'll be more than happy to answer.
Thank you, Dr. Krishan, for such an informative and interesting lecture. You covered such a huge topic in such a short time, I know.
Thank you.
Thank you very much. Any questions? No.
Any questions?
Okay. Thank you again.
Okay.
Now we have our second speaker, Dr. Rajbir Bhatti. Now, Dr. Bhatti is an associate professor and head of the Department of Pharmaceutical Sciences at Guru Nanak Dev University, Amritsar.
Yes, it works.
Dr. Bhatti earned his PhD in Pharmaceutical Sciences from GNDU in 2015 after securing a gold medal in his M.Pharma Pharmacology from Punjabi University, Patiala, with over more than 75 publications, including around 61 peer-reviewed research papers, 7 book chapters, 4 conference proceedings, and 1 patent. He is highly active in pharmacological research. His expertise lies in CNS pharmacology, especially in analgesic, anti-inflammatory agents, and natural molecules targeting effective pain. Dr. Bhatti also mentors students, about 3 PhD, 39 M.Pharma, and his research has secured 3 completed projects. Additionally, he holds administrative responsibilities like Deputy Director of GNDU's UGC plus HRDC organizing multiple faculty induction programs. Welcome, Dr. Rajbir Bhatti.
Thank you, Dr. Garg, for such a charming introduction. Only
Sorry.
she. And I was sitting here. My students are also here. Some of them who have passed out. She was asking me whether, ma'am, आपको डर लग रहा है? And I must say, I am really very afraid because it is. And most of you want to wind up soon, so I'll just rush through. What happened? So the topic that I have chosen is assessing the burden of adverse events in Indian scenario, and I have taken insights from the Global Burden of Disease study. So I always believe as a pharmacologist in this quotation by Kamneski, there is clearly no safe biologically active drug, which all the pharmacologists will agree. There are only safe physicians. It's the way we use the drugs that will matter. ADRs, as all of us know here, they are unwanted drug effects, which may include toxic effects or side effects, maybe dose-related, maybe non-dose-related. I am skipping the jargon of all these, which you have studied already. And adverse event is a broad descriptive category, which includes all these things which are adverse, which are unwanted, that may occur as a consequence of a disease or an adverse drug reaction. So history is not was. Actually, it is. Why I have included this slide is, have we really learned our lessons? We, I think, very conveniently forgot the catastrophe of 1937, which the students here, I mean, even I was not born at that time, but I've read it as a part of medical history in pharmacology, elixir of sulfonilamide disaster. But look at the state that we are facing here in India. The same diethylene glycol, which killed more than 100 children way back in 1937, the same things are coming up. The Coal Rift tragedy is the latest. It happened in 2025, this one, in MP. And we have gone as far as Gambia, then Uzbekistan, where more than 100 children died because of the spurious syrups supplied by our country. I'll like to differ with my previous colleague, the speaker. सब कुछ अच्छा नहीं है. So the WHO has flagged the Indian drug market, which we were monopolizing in the formulation, manufacture of generic drugs, but now we are in a very, very precarious situation. Of course, the government is doing its bit, and the new norms, they say, will be effective by December 31st this year. And the Union Ministry has also issued advisories against using these cough syrups for children less than 5 years of age. But then, how can we forget the disaster that shook the entire pharmacology world by a storm? That was the thalidomide disaster. All of us know the details of it. And this is how the exhaustive testing in the animals before taking it to humans and exhaustive clinical trials came into being. And the last tail ending of these survivors, they are less than 3,000 people. Still, they are there in all over the world. But literature evidence says we don't have these people in India. Although in India, this drug was repurposed for leprosy and also for treatment of certain kinds of tumors. Leprosy was in 1968 to prevent the severe leprosa, the nodosome reaction in the patients of leprosy, and we have the largest number of leprosy patients in India. It might be not holding to that extent now. Bangladesh has superseded us because we've taken a lot of measures. So the WHO says an adverse reaction is a drug-related event that is noxious, unintended, occurs at doses used in humans for prophylaxis, diagnosis, therapy of a disease. And then we have NCC MERP index, which is an international index developed to assess these errors, and also it standardizes the categorization of the errors. Let's look at the categories that they have formed. So here we have categories from A to I. It's not working. Anyhow. So A to D are not of much problem because they are errors, but they do not amount to harm in a patient. Whereas E is error, which causes reversible harm. F is again an error, little higher degree harm, but this is again reversible, temporary. But G is a permanent harm to a patient. H is where it is near death event in a patient, such as shock, allergic shock, or cardiac arrest. Then I is the error, which is the fatal error, which leads to the death in patients. So according to the statistics, WHO says around 1 in every 10 patients is harmed in healthcare, and more than 3 million deaths occur annually due to unsafe care of patients. The heartening thing is that 80% of these deaths can be prevented, can be avoided if proper health care is taken care of. Then the investment in reducing patient harm can lead to a significant financial savings at the global level, and more importantly, better patient outcomes. An example of a good return on investment is patient engagement, which if done well can reduce the harm by up to 15%, if not more. The common sources of patient harm include medical errors, surgical errors, patient misidentifications, then unsafe injection practices, healthcare associated infections, and the Swiss cheese model is very frequently used to explain the various causes of adverse events. For instance, I've taken an example of surgical errors here. It's not working. So the surgical errors may involve a human factor, which can be a tired surgeon. In Indian context, this is very, very applicable, where the number of surgeons as compared to the number of patients is very, very less. We have a lot of population, but qualified surgeons are few. Then it can be a system failure, where incomplete pre-operative checklist was there. The people might not be trained or might not have taken proper patient history. Technical failure, which is due to the equipment. Then it can be cultural failure, where the junior staff who might have noticed something happening, or some, say, the patient history of having some allergy, but he's afraid of the senior surgeons and fails to report that. So all these errors lead to lack of patient's faith, and profoundly affect the drug compliance at the end of the patients. So these are the studies from various journals. Let's skip these and let's come to the point in question, the global burden of disease, that is GBD, popularly called as. It's a global health metric, which is a comprehensive scientific effort to make some standardized measurements for various diseases, disorders, which lead to loss of health, loss of lives, and it has some metrics such as the disability adjusted life years, which is life years lost by the patient, then years of life lost, and years lived with disability in addition to death. These are few parameters on the basis of which we can categorize, quantify, or qualify the healthcare that we are providing. So the key purpose of GBD studies is to provide a powerful evidence-based picture of what makes us sick and die, and enabling informed health policy and priority setting. So the important limitations of the GBD studies remain that the estimates that we make are modeled. They have very high-end statistics involved. As sir was saying, AI is used, and models are used. You need a very qualified statistician and epidemiologist to work with you. And then there are data gaps. The quality of data may be poor from a country, say, like Afghanistan, where the collection of data itself is a big challenge. Then even in India, we are not very advanced data-wise. And GBD has a lot of interest in India. Why? It is because we are the highest populated country, and population is the raw material for the GBD studies. So if we develop a stringent system for collecting data by way of pharmacoepidemiological studies, will be into the game of various disorders and deciding what to develop as a new drug in a big way. So let's come to the point in question which I want to discuss with regard to the healthcare in India. First of all, the data available. Obtaining a precise national figure is a challenge due to limitations in death registration data and widespread spread underreporting of ADEs. So then there is unrecognized priority. The adverse effects might not be quantified significantly, especially in the rural regions where the healthcare is not very well developed. Then the foundational needs addressing this issue should begin at the specific geographical regions, not only be limited to the urban areas, they should go to the villages and the rural India also. The existing systems that we have in India, government has done its bit also and it's doing. We have National Accreditation Board for Hospitals, that is NABH, which helps to improve the healthcare quality. Then we have surveillance systems for the adverse events, especially for immunization. We have done it well. And also we have Pharmacovigilance Program of India. PGI is participating very actively in this program. And this is also a program to monitor drug safety and vaccine safety. The critical gaps that remain, despite these systems, are notable shortage of research that specifically measures the overall burden of the adverse events in Indian population, availability of data sources. We do have a national patient safety implementation framework, which was developed by the Ministry of Health and Family Welfare in 2018. And the goal was by 2025 we'll achieve this. And by 2030 we'll have good health for all as a part of the WHO health. There are some goals that the WHO sets on priority. They have set this 21 to 30 as the patient safety decade. So in consonance with that, we developed this framework. At the global context, the GBD studies provide a model. It aggregates data from various sources, including vital statistics, published literature, to estimate the impact of different health conditions, including the AEMT by country-wise. This is the paper that I have relied much on. It is in the Indian scenario by our researchers only, but the data they have taken is from the GBD. It's available freely for all. So India has a vast and diverse population with varying degree of access to tertiary healthcare. The AEMT accounts for only a small percentage of death, which is itself a big, cause of concern. If a country like USA reports a very high rate of deaths due to adverse events, it could not happen that in India we have less. It's only the underreporting of these deaths that is to be taken care of. So taking you through the methodology that we follow, we have data sources in the form of censuses, insurance claims, then non-reference methodology, literature surveys done, population-based studies, hospital records, and large-scale surveys. Then the data collected is subjected to Bayesian models. The DISMOD MR 2.1 is the most popular these days in the GBD studies. And then we quantify the harm in terms of DALY, that is disability adjusted life years, years lived with, years of life lost. Then we also categorize the data gender-wise, that is male is to female ratio. Age-wise categorization is also done, and so is the disease-wise categorization. So if we compare the data available in 2019, this paper was although published in 2024 only, it's a recent paper. So the data available is for 2019 only. This is India and this is the USA. So the number of deaths in India is 32,464 as compared to 5,016 from the adverse drug reactions. The death rate in our country is 2.33, whereas in the US it is 1.53. The percentage change in death rate in India is negative, which is again a questionable thing. Have we really reduced the number of deaths? Whereas in USA it is 1.21% still. So the paper that we have, the paper evaluates the incidence rate in AEMT in India from 2010 to 2019. We see there is an increase in the adverse drug events from 140 to 160, and then the corresponding number of deaths in 2019 were also higher in terms of death rate. It was 2.34 as compared to 2.33. Then the Delhi rate was also higher in 2019 as compared to 2010. This is the age-wise burden of adverse drug events. We see the people more than 50 years of age are more likely to have the adverse drug effects. So this is 45 as compared to 0 to 9, it is only 8.7. If we see the percentage of deaths due to adverse events due to drugs. Then the Delhi is also high in the population more than 50 years of age in 50 to 74 group and excuse me, 75 plus group. So it is reiterating the facts from the table only. If we come to gender-wise categorization, here again it is evident, although in the USA, more males suffer from adverse drug reactions as compared to females. In our country, the females are more likely to have adverse drug effects. It's, the death rate is 2.77 due to adverse drug reactions as compared to 1.92 in the males. So Delhi rate is also higher in females as compared to males. So the burden of adverse events, this is again reiterating the same. The females constituted 58% of the total number of deaths due to adverse events in 2019. And the Delhi in males decreased in numbers, in terms of total numbers. And in the females, it was 62,050 as against 61,000 and something. So although the death rates did not decrease, a downward trend was observed in Delhi rates in both the genders. So the key highlights that we carry from this study is the overall burden is relatively small as depicted by the data which is available. Again, we need to work on the data, and I think the national framework that they have decided upon, 25 was the last year, will continue with this, and this is going to increase. It cannot be less than 0.01%, when at the global scale it is much, much higher. So the reported fatal ADEs from Gujarat revealed 0.53% of these were serious, and 1.1% in pediatric patient from Maharashtra. Again, many states do not have the availability of data. Maharashtra and Gujarat are few states where the studies are very advanced. Even Karnataka is in this category. So these figures are from localized studies and cannot be generalized at a nationwide level. Then the high-risk medications are mainly the high-risk drugs, that is the cardiovascular drugs, which are the most implicated. This is at the global level only. And the antibiotics having a very narrow margin, that is ceftriaxone. Then NSAID inflicted damage is also a leading cause of serious ADRs in populations at the world level, not only in India. So the vulnerable groups remain the elderly patients, and also this is mostly above 80 years, deaths are more, but above 50 years, the ADRs are more. This could be due to polypharmacy because as we age, the patients tend to have multiple problems. It could be hypertension along with hyperlipidemia, along with cardiac problems and so on. So polypharmacy is common in these age groups. This could be one reason for ADRs. Then the systemic issues, that is the quality control, as we saw in the cough syrups, which has brought a lot of bad limelight to India. So understanding the challenges, the significant underreporting is the primary culprit, which we need to tackle over the years. We need to come up. So if ADRs in USA is 43% and we are reporting only 2% while having the maximum population in the world is underrated. Then the data quality issues, we should go to all the states, should be included, not only the more developed like Gujarat, Karnataka, Maharashtra won't work. We should go to the other states also. The rural belt also has to come up. The regulatory oversights, this is mainly dealing with the spurious drugs that are available from the Indian market. Many firms have been shut down of late. So the 72nd WHO Assembly in May 2019 decided to observe September 17 every year as the Patient Safety Day. This year's theme was Safe Care for Every Newborn Child, and the slogan was Patient Safety from the Start. So the WHO has taken the patient safety very seriously, and they have in their convention, which occurred on August 3rd, 2021, they have come up with a safety action plan for a decade, 2021 to 2030. And along with this plan, the key milestones that we hope to achieve is the assessing of the landscape, then securing strong commitment from political and organizational leadership, then establishing a sustainable mechanism for implementation, aligning with national contexts and priorities, decide on design and model for change of implementation if required. Then the 7 strategic objectives of this plan are the policies which will eliminate the avoidable harm in healthcare, then developing high reliability systems, safety of clinical process, patient and family engagement will help in a big way, the education to the health workers, skill and safety development in the health workers, information research and risk management, synergy partnership and solidarity. Because there is no standard way to investigate patient deaths for safety issues, there is ongoing disagreement about whether the estimated deaths or mortalities should be used as a reliable indicator of patient care or of adverse events or not. The current tracking methods for fatal adverse events may have flaws, such as relying on small samples, then subjectivity. But since death is the most severe outcome, it cannot be undone once it happens. Understanding the real-world frequency and causes of these events is fundamental to making healthcare system safer. So, in conclusion, the patient safety is the target goal, and it should be everyone for patient safety, all healthcare workers, including the physicians, patients, families, pharmacologists, everyone. So thank you for your patient listening. If there are any questions, I'll be happy to answer.
Thank you, Dr. Rajbir Bhatti, for enlightening us with this lecture. And I completely agree with you that there must be significant underreporting of all these adverse events in India. And also there's no formal training with the residents, I think, in majority of the medical colleges, dental colleges, to report these events. It should be included in their curriculum. It should be monitored by someone senior, whether they are reporting it or not. Only then we can get the complete data, as you mentioned. What about Punjab?
So we don't have much data.
No data in Punjab?
No. In Indian scenario, if you see the GBD, once they published, I am a part of the Global Burden of Disease Studies. I am working as a senior collaborator with these people. So they collect data from all sources that are available. In India, however, only the metropolitan cities are the ones that are giving.
I think big institutions like PGI, AIIMS, they must be doing it. But not tier 2, tier 3 cities, small hospitals.
And we came up in a big way during COVID. ICMR was a big player there, and they really gave good data to the GBD. They contributed it during that time, and after COVID, I think there is an upsurge in studies, and eventually we'll have. And in our country, still the doctors are also reluctant because we don't protect the doctors as we should. So that is also one of the aims of the various goals that have been, various categorizations that have been made. They have this goal that you should first give this confidence to the physician, that if they report a particular drug reaction, there will not be much litigation because errors can happen, otherwise they'll never be.
That could be the reason.
Yeah.
One want to hide these things.
Yes,
and another thing which you mentioned is, you are correct, that substandard drugs. I think in what I have seen in European countries or in US, there are only 2 or 3 major drug companies. But in India, there are so many.
Yes, sir. Now, especially in the last decade, there has been mushrooming of very small. These are all very small drug manufacturing houses. No big companies involved. I went through the literature that was there for the cough syrup. No big pharma firm was involved except for one, that is Glenmark. They came up with a lisinopril formulation, sustained release, but it was not actually sustained release. So it because releasing the drug very fast, they have withdrawn already. Otherwise, no big pharmaceutical houses involved in this.
Involved. Then government should have tight regulations.
Yeah, they are doing it now, and we hope that they'll do
Strict
more.
implementation.
Yeah, yeah.
Thank you all the speakers for such an exhaustive lecture. Now I request our chairperson to kindly facilitate the speakers. Please, all the speakers, can you please come on the dais? Thank you, Dr. C.K. Krish. Kindly give a round of applause. Now, I request chairperson to kindly facilitate Dr. Rajveer. Thank you, doctor. Now I request the speakers to kindly facilitate our chairperson for the session. Thank you, sir. Kindly give a round of applause. Now we conclude for today's second day of the session IPSCON 2025. We'll see you tomorrow morning at 8:30. Thank you, everyone.
सारी रात होती बैठी ड्यूटी लो वाली मर जाना सी अपन